Morning again, everybody. Can we settle down to start, please? So, welcome in Kruger and welcome here at our Savanna Science Network meeting. Our first speaker today is Kathy Griever. She's the General Manager of the Savanna Research Unit and our boss. So, we are extremely happy and pleased to announce her for the opening talk. Thanks, Kathy, for agreeing to do this. Thank you.
Morning, everyone. Just give me a few seconds just to log into my laptop. All right, so it doesn't matter how much we plan to get things to start at 8. There's always little gremlins that creep in to the system. Just bear with me. All right. Once again, good morning, everyone, and welcome to the 23rd Savanna Science Network meeting. Yeah, it's nice to see all the familiar faces in the audience, and I've noticed that there's quite a few new faces. So hopefully the next few days of the Savanna Science Network meeting is going to allow for some opportunity for us to get to know each other a little better. But also a warm welcome to everyone that's joining us online via our streaming platform. And once again, apologies for the delay because of the gremlins. But as the name of the Savanna Science Network meeting indicates, it is a networking meeting. It continues to play a crucial role in catalyzing collaborative research whilst we try and foster co-learning in sandparks. And it also maintains the scientific integration nationally and internationally. So yeah, welcome. Let's see if this is going to work. All right. So the Savanna Science Network meeting has come a long way. Last night, a group of us were actually just talking and reminiscing on the good old days and where we all came from and where we sort of linked into the Savanna Science Network meeting. But anyway. Okay, so it has come a long way, and it started way back in the 2000s, where a small group of scientists met at Shingwetsi Rest Camp in the north of Kruger National Park to discuss Savanna River-related issues. This was following the Kruger 2000 flood event. And that initial meeting led to the conceptualization of the International Savanna Science Network meeting as we know it today. Through the vision and the insight of our founding members, 3 of whom I'm showing on the slide, it also initially catalyzed by the Mellon Foundation through the enormous support by William Robertson, who showed quite a lot of support for this initiative. And the meeting aimed to bring together local and international scientists to discuss the latest research relating to biodiversity, conservation, and management of protected areas. This was initially within the context and focus on research conducted in Kruger, but later it expanded to include research and savanna systems from around the world. with savanna ecology from all continents often represented at this meeting. The meeting was supported by the Mellon Foundation grant until 2013, but then slowly with some adjustments, the last adjustment of the fee structure took place in 2014, and the meeting has been self-funded from 2015. So in March 2003, the first official Savanna Science Network meeting was held at the Goldfields Auditorium here in Skukuza. It's a building just across the road from here. But over the years, the meeting has grown into a successful, well-organized, and highly sought-after international Savanna conference. And despite its success and popularity, it has retained its original character and that familiar flavor, remaining true to its name as a network hub. As you can see, that was basically the first program way back in 2003. And as you can see, the team has over the years tried to just zhuzh up the program by putting in quite a few different front covers, etc., just to try and align with the meeting for that particular year. But Sandbox is very proud to have initiated and to host this annual symposium-style meeting, which has become a well-established platform for sharing knowledge, scientific knowledge, and practical experience. So yeah, it's not just the program that changed over time. The team also had to think of innovative ways on how to just, provide different mugs or coffee mugs basically over the years. And as you can see, you've got a new one this year. So it does take a lot of initiative to try and keep that flair going. Okay, let's see if this is working. So this year we have about 228 delegates attending the meeting, including 125 representatives from Africa. Among the international contingent, the USA, England, Germany, and France have been particularly well represented over time, and we're still seeing it this year as well. Currently, the meeting is attended by about equal numbers from South Africa and international institutions. So we are honored to bring together such a diverse group of remarkable individuals as yourselves, which are basically individuals from leading scientists to researchers, conservationists, and policymakers from around the globe. The meeting has a highly diverse and dynamic delegate attendance, which has grown both in numbers and origin over the years. During the discussions last night with one of the individuals that's sitting in, in the audience today, that individual was part of the Savanna Science Network meeting, even before it became the Science Network meeting. And yeah, he actually did indicate that the network meeting and just the way it's grown over the years has become one of the most sort of sought after networking meetings or conferences because it is so inclusive, right? But I had a look at the delegate information from 2008 onwards, and it shows that representatives from more than 340 unique institutions have participated since then. Primarily from sandparks and Southern African universities, but also featuring delegates from between 16 to 19 countries each year. So that photo in the top left basically shows where we came from. No one can actually tell me what year that photo was taken, but it is quite interesting. Over the years, the group photo has become quite a special significance or an event during the Science Network meeting. And we are planning something special for this year as well, but watch this space. So we have a full program, which indicates that there is a lot of great research happening in the Kruger and also beyond. The program will center on 9 themes that reflect the pressing challenges faced by savanna ecosystems in Southern Africa and beyond. But it will also allow for opportunities to uncover new insights and to improve our understanding of the challenges confronting savanna ecosystems and the communities coexisting with the nature. You would have seen these themes on the program as well. So it is basically the headings of the different sessions. The program also directly or indirectly reflects our 10 research themes, which is encouraging because for years we have been working towards having more research focusing on the understudied themes. But before I chat about our research themes, just a bit of background on how we got to those research themes. Some of you might remember in 2018, a horizon scanning exercise was conducted with scientists, park managers, and senior managers across all functions and scales in sandparks to provide context for developing key research themes for sandparks. This identified key factors influencing sandparks and the delivery of its mandate at both the organizational and park level scales, especially in the fast-changing context of sandparks, conservation in general, and then also South Africa and globally. So back then, after a lot of deliberation and debate, as we usually do in sandparks, the scientists and the non-scientists all agreed that the top 5 influences facing sandparks in future were obviously climate change, which includes the large infrequent disturbances, human population growth and resource scarcity. Then was also focused on the national, socio-political and economic processes, including land restitution, and then the illegal activities inside and outside parks. But then the changing societal expectations of what parks are about and the relevance of conservation was also flagged. So that came up as the top 5 influences. But then during 2024, the articulation of Vision 2040 has added additional impetus to better understand these influences at multiple scales, and to assist in identifying opportunities for leverage and action across mega living land and seascapes, especially seeing that we've got our marine protected areas included as well. But it was also to assess the socio-ecological acceptability and the effectiveness thereof. In particular, the concept of mega living land and seascapes within Vision 2040 represents a deliberate move from managing protected areas as isolated entities, as we did back in the days, to working with people in wider scale social ecological living landscapes for ecological sustainability and socio-economic upliftment. All right, so having an institutionally embedded research capacity in sandparks is important, and it was recognized back in 1950 already, when that guy over there, Dr. Nell, was appointed as the first biologist in Kruger. In spearheading the research section, Dr. Nell already listed 4 issues that were regarded as research priorities. All right. I'm not sure if you guys can see it there at the back because it is just a printout from one of the many books we had to go through during this process. So it mainly included population studies with the emphasis on surveys of these animals. Then also pasture research with the emphasis on felt burning. Then it was matters related to boundaries, basically all our fences, etc. And then also the ecological factors and influences, for instance, tourism. So those were the ones that Dr. Nell already listed in 1950. But 70 years later, our first research strategy was approved in 2020. with a 5-year revision cycle. We are currently in the final stages of the revision process, but the research strategy basically aims to enable research and monitoring in sandparks, and it articulates the broad research directions to focus attention rather than define specific shorter-term research needs or questions. From the sandbox context, we have now highlighted 10 research themes which emerged, but it covers in a cross-cutting manner the various organizational strategy or the strategic focus areas, which I've listed on the right-hand side of the slide there. It's basically the conservation areas, the management of those, and then the inclusive economic growth, regenerative tourism. Someone mentioned yesterday that, yeah, we have moved towards regenerative tourism, social license and legitimacy. Then it's also to try and make sandparks agile, caring, and also a capable organization. And then obviously there's also sustainability and resilience, which are given additional emphasis by Vision 2040. So these research themes are pertinent to the next 5 to 10 years. So unlike the 1950s and 60s, scientists today also have to compete with social media and AI influences. So we need to find interesting ways to get our science out there and to make our stories more compelling. In 2018 with our horizon scanning, climate change and the large infrequent disturbances was identified as the top influences facing sandparks. And we weren't far off because we've had flood impacts or flood events in the park already twice since then. So I won't go into much detail with regard to climate change and the effect of the recent flood. There will be a talk on Tuesday covering the various aspects from the recent flood event, which had damaging impacts on our infrastructure in Kruger National Park and left a path of devastation in some areas neighboring the park. For this year's conference, we have partnered with our colleagues from Socioeconomic Transformation. I was trying to see where Connie is, but I haven't seen her yet. So we have identified households affected by the recent flood event, and we will thus contribute towards rebuilding initiatives in the flood-affected areas. If you feel an urge to make additional contributions or donations to this little project of ours, please contact Jackie Deacon in the front for further details. So then she will just explain the process going forward. But in all fairness, the park has been through natural disasters before, and we always rebuild and we also persevere. And that's why this year, the Savanna Science Network meeting reaches yet another milestone, and it holds special significance as it coincides with Kruger National Park's Centenary in 2026, later this year in May. So it's marking 100 years of conservation and science excellence in one of the world's most iconic protected areas. We thought we'll just put some nice photos from back in the day, just things that we got from the archives. And I mean, everyone knows the big old clock that's parked here in the rest camp as we come in. But then there's also, the photo doesn't show clearly, but it's obviously, there wasn't high definition back in the day. But there's a guy standing on a rock, and he's busy with some other telemetry type equipment from what Judith explained to me. But yeah, so there was quite a lot of things that happened over the years and changed over the years, including our game capture techniques, etc. So a lot has happened in the park over the past 100 years, and it makes it rather special. But just for some background, for those who might not have been here or know the full history of the Kruger National Park proclamation, the park was proclaimed as a Sabi Game Reserve in 1898, which is basically that area between the Crocodile River and the Sabi River. But during the ensuing Anglo-Boer War in South Africa, the proclamation was nullified. So in 1902, after the war, that same area was reproclaimed as a Sabi Game Reserve. But then there were further extensions that happened in 1903 when the Shingwedzi Game Reserve was proclaimed, that portion up in the north of Kruger. So it's basically between the Letaba and the Limpopo rivers. In 1914, the area between the Olifants and Letaba was included. So it's that little white section there. And then the process continued. In 1916, the Sabi and the Shingwedzi Game Reserve were then consolidated to basically form the park boundaries as we know it. On May 31st of May in 1926, our parliament passed the National Parks Act, and the reserve was renamed the Kruger National Park as we know it today. But since then, we haven't stopped establishing the park. We've been dropping fences. We expanded by dropping the fences. We basically expanded the landscape to incorporate different landscape users, with further inclusion of community and other conservation compatible areas adjacent to the park. All of these adjustments were guided by the SANParks land acquisition policy and the Kruger National Park Land Inclusion Protocol, which provides clear biodiversity, that's a whole long list, it provides clear biodiversity, land use, socioeconomic, management, governance, and risk criteria. So it's not an easy job, but we're getting it done. So yeah, we haven't stopped establishing the park, and I think we are spearheading ahead with our Mega Living Landscapes concept to actually expand the park boundaries further. So this marks 100 years of inspirational leadership in conservation, biodiversity, tourism, and hospitality. It also shapes the responsibility and the continuous learning, which is so powerful, and what makes the Kruger National Park system so strong. Long-term stewardship of the Kruger National Park is also vital, as the park needs to be here for generations to come. So there is a sense of strong governance and conservation, because places like Kruger National Park survives when they are managed properly. So when stewardship fails, we may lose an incredible asset such as Kruger National Park. And that's why SANParks is spearheading this process to sort of just ensure that Kruger stays the flagship park that it is. And we've really come a long way through inclusive growth from a park system which was completely isolated. It was us and them with the fence around us, but we have brought the park closer to the people. It's a more open park, working with communities within and around Kruger National Park. And we continue to find very good ways to manage that relationship, which benefits both the park system and the communities. I just want to reflect on the bridge. Right. I'm not going to say show of hands, but it was interesting just to speak to some of the people last night at the meet and greet. Some people had not actually seen the bridge just as it is there, but they also know now that there is a train on the bridge in Skukuza, because that's where we are currently. But it was very interesting just to talk to the different people that have attended the network meeting over time, because they have also seen the changes that has happened over time. All right, so yes, in closing, we look forward to hearing from you during the next few days and hope that this is one of many more happy returns to the annual Savanna Science Network meeting. I know there's a challenge happening between Greg. There's a few of our people that have been attending the network meeting over the years, but now there's a challenge because one individual is wearing a crown and he's going to try and attend the meeting for as long as he can so that he can retain or maintain that crown. But he's got high competition behind him. So yeah, we'll have to wait and see how that little competition unfolds. It's between 3 people in our delegation. But as usual, the program includes opportunities for reflective learning. So I encourage you to make the most of the social portions of the network program, basically during tea time, lunchtime, even after the network meeting at the end of the day, to engage in discussions and to reflect on some of the research that you have done, because that's the only way that we actually learn from each other and we can actually improve what we are doing. And the network meeting has showcased that over the years. So I also want you to make new friends and possibly take up new research projects so that we can really add value to our continued efforts to learn by doing. So with that, I don't want to waffle on too long before Judith comes and stops me. I would like to officially open this year's Savanna Science Network meeting. Enjoy it. Thank you.
Thanks, Kathy, for that inspirational talk, and especially around the background that you gave us, how Kruger actually established itself and grew bigger. I think a lot of members in the audience didn't know all of that detail, so thanks very much for that. So we're going to continue on. Samantha is going to do the housekeeping for us, and once she's done with that, we're going to hand over to Llewellyn, who's then going to start the first session. Thank you.
Good morning, everybody. So we'll just do a bit of housekeeping quickly. The program has been shared with everybody, and I will share it again on the WhatsApp group. If you're not on the WhatsApp group, please just go see Jackie in the front. There is a notice board there where you can scan a QR code and join it. The reason why we need everybody to join the WhatsApp group is because all announcements as they happen will be shared on the group, so it will make sure that you are aware of any changes in the program or anything as and when it happens. There is Wi-Fi in the facility, and we will actually share that on the WhatsApp group, and we'll put it up in the front, so everybody should be connected. The program and the whole conference is being live streamed, and so the link has been shared on the WhatsApp group, and we will reshare it as well. And so because we are live streaming, we had asked people that if you do not want your talk to be live streamed, to please let us know so that we either move you to the top of the program or the end. So please, again, if you have changed your mind, please come let us know before the session starts every morning. And with that as well, we do share the presentations on our website at the end of the conference. And if you do not want your presentation to be shared there, please do let us know and we will make a note of that. The sessions will start at 8 o'clock promptly each day. Today we just had a bit of technical issues, and that's why we started slightly late. And in terms of your presentations, the program this year has presentations set at 10 minutes each. So if you do want time for questions, please make sure that your presentation is done within that allocated time period. We do have a pointer with us here. Very easy, 3 buttons. Red is back, green is forward, the laser is white. No fidgeting around, please. We do have a screen for you here, and it is on presenter mode, so you will be able to see your notes. And for what we have this year is that we have a timer, that electronic timer that is set up on top of the screen. So as you are talking, you will be counted down from 10 all the way to time up. The chairpersons who are sitting here also have a timer, so they are also looking out. So you should be able, whilst you're standing here, to see your notes, your presentation, as well as the timer, and also what the rest of us are seeing. We do have a poster session tomorrow for those that are doing posters. So that is at the end of the day tomorrow. In terms of our meals, so all the teas will be happening outside in the foyer. There will be coffee set out throughout the day. So in the morning when you do come through, there will be something for you out there, and throughout the day as the day goes. As you know, we do not serve breakfast at the conference, only lunches, and there are events in the evenings that are planned that do have some meals associated with them. And with that, for the program, all the lunches will be at the lodge. And at the lodge, what we've tried to do this year is that we have set up a number of feeding stations. So there'll be 2 inside, and there'll be 1 station set up outside where we had the meet and greet. So if we can just disperse ourselves throughout the various feeding stations to ensure that the queues move and we come back to start the sessions after lunch on time. And for those with special dietary requirements, on their tags, they've got a little pink sticker on it. You will have your own serving station as well, including even during tea time. You'll have a different session just close to Jackie here, and then everybody else will be served in the other stations. And so please, if you do not have a special dietary requirement sticker, do not go eat at the special dietary people's table. Please also ensure that you have your tags on you at all times. This is so that we are able to identify who you are, and that the people who are assisting us throughout know that you are actually part of the conference. What else do I have to say? We do have various activities that we have planned out throughout the day. So we've got a really nice little session at the end of today's session later in the evening. So please do come join us. It's the poster session tomorrow. And then on Wednesday, we've got our little fancy gala dinner, which is themed. And I hope that everybody's got their vintage outfits together for that. For Thursday, there is no planned dinner, but we are hosting a memorial for the late William Bond, and we had sent out a Google form so that people can RSVP if they'd want to join us. We will resend the form again, and so please, if you are planning on joining us for the memorial service, please RSVP on that form, because we are working on numbers and we want to make sure that everybody is covered. On top of us live streaming the events, everything will be shared on the SANParks social media platforms. So we do have accounts on everywhere, but on Blue Sky. We don't have a Blue Sky account as yet, but we are on X, we're on Instagram, we are on TikTok, we are on Facebook. So if you do have a social media account, please do share your handle with us, so that if we do take photos and we're posting there, that we can actually tag you and your institution. So yes, they will be taking photos of you as you're presenting, and photos of everybody as we move around. If for whatever reason you do not want your photos to be shared anyway, please do let us know, because in South Africa we do have rules and regulations, and because of Poppy, we need to seek your permission before we can share anything concerning you online. So our hashtags for that are hashtag LiveYourWild and hashtag SSNM 2026. So do use that, do tag us at SANParks in all the major platforms. And we will be taking a photo today just before tea. So instructions, after the last speaker for the morning session, there will be instructions to guide us on where we're taking the photo and what needs to happen there. So please make sure everybody's got your tag, because your tag has a little colored sticker on there, and that is very important and crucial for the photo. So please don't disappear before tea, because we would like to have everybody possible to take the group photo. And lastly, for the speakers as well, please make sure that you come and you sit on that chair so that you can get mic'd up before you come through. So every speaker who's going to come, once the person is on stage, if you are next on the program, just come have a seat there, and you guys should be good. Thank you so much.
Morning, everyone, and welcome to the Invasive Alien Species session. I just want to make a note, the talk by Martin Treckles has been withdrawn, so we'll just carry on through the program and the talks will come through a bit earlier. So to kick off our program, our first presenter is Julie Coetzee, and it's on the mass rearing and inundative release of biocontrol agents to manage invasive alien species. Okay, thanks, Julie.
Ready? Well, it's lovely to be here. My first meeting was in 2004, so it's great to be here at the 2026 meeting. And my talk's not, it's not on savanna ecological processes, but more the implementation of years, decades of research into managing what threatens probably a particularly important component of savannas, and that's aquatic ecosystems. Do I just hit forward? Oh, there we go. Okay. So just to acknowledge my co-authors here, it's Llewellyn Foxcroft and Zanele Jele from Scientific Services here at Sand Parks. And I'll be talking about the strategic strategic mass rearing and release for invasive species management, focusing on biological control of aquatic species, but this is very relevant to the management of terrestrial invasives as well. And for those of you, the top of the presentation is cut off, but there are many, many problems associated with invasive species. And here I've got a picture of Makatsi Spruit as it leads into the Engelhart Dam, covered in water hyacinth. And you can see these hippos appear to be having a good time, but it's probably not that great considering the multiple cascading impacts that this kind of invasion would have. You can obviously think of the biodiversity loss, water security threats both within and outside the park, livelihood disruptions, probably more relevant for outside the park than inside the park, and then the economic burden for protected areas and, of course, rural communities. So these impacts are many and they are diverse. So what can we do about this? And I work on releasing small host-specific insects that we use as nature-based solutions to control invasive species. They cannot attack anything other than the target plant, and we get very frequently asked the same question. So if anyone wants any information on the science behind biological control, please come and talk to me. But essentially, we release little armies of insects against invasive species to end up with beautiful before and after pictures. And you can imagine the array of biodiversity benefits that are returned and the ecosystem functioning that's returned following control such as this. So this is a savanna up in Kenya, obviously, but this didn't start up in Kenya. This actually started right here in Kruger Park in the 1980s. So just if you take a drive down to Lower Sabi at Sunset Dam, 1985, this is what the system looked like. The little weevil was released shortly after 1985, and within a couple of years, we could get to another one of these beautiful before and after pictures. I did not take these. I wasn't really a scientist at that time. Similarly, Orpin Dam, if you're on the way up to Satara, back in the 2000s, you would have seen pictures like this. And following releases of these agents, we can get nice, clear pictures. And then some of you might say, hey, but it comes and goes. And this is largely due to the dynamics of the seed banks of these plants, which I'll touch on. So in a nutshell, biocontrol significantly reduces the spread of these aquatic plants, well, any invasive plant, decreases our reliance on herbicides. And I think that's a key point that we have to take here, especially when you drive around and see lots of blue plants at the moment lining a lot of the roads. We restore ecosystems, and we can do this across sub-Saharan Africa. But there are a number of challenges. These include eutrophication, which is the increased nutrient input into these systems. South Africa has a really bad rap at the moment for our loss or our lack of really good treatment of wastewater throughout the country, and this would obviously be particularly important for water entering the park. Oh, sorry, just go back. We face cold winters where the insects might die or where the plants might die. We face floods, we face drought, and we also face uncoordinated management. And I'm going to put this here as an example of uncoordinated management at a highly eutrophic site. So there we've got a gent pulling out water lettuce, and as fast as he's pulling it out, those plants are growing again. So we needed a paradigm shift from this classical approach where we develop an agent, release it into the system, and say, good luck, guys, hope you can face all those factors affecting your success, to an inundative process. And at Rhodes University, we've developed a program called the Sisonke program at the Center for Biological Control, where we mass rear insects by people living with disabilities from the local community who can now be a part of nature-based solutions, not just for South Africa, but we've sent these agents north into Southern Africa, we've sent them to the US, and we've sent them to India most recently. And we can collect these insects, so we essentially farm them in huge greenhouse tunnels. We package them up in very high-tech, sophisticated containers that you can buy at Crazy Plastics. And what this does is it offers us, between classical biocontrol and inundative biocontrol, we get these complementary strategies to maximize their ecological benefits. It's cost-effective. They're environmentally safe alternatives, again, as I say, in comparison to herbicidal control. And potentially a problem is that this inundative strategy relies on a lot more coordination. And we've had fantastic success with this at Hartbeespoort Dam. If anyone's frequents Hartbeespoort Dam, you might tell me I'm lying right now because there's a huge mat of water hyacinth there at the moment. But through this inundative release strategy, where we can release tens of thousands of insects a month onto the system, we can break that permanent presence of this mat. I put the slide up because we have a lot of data, and this is a fierce ecological meeting with lots of graphs and stuff. I don't want to go into this too much because I'm running out of time, but essentially we can reduce mats of water hyacinth. So all that green is water hyacinth. This is December 2020, when the plants were at 42%, to March 2021, where we got that cover down to 7% at the peak of summer. No herbicides, no other management strategy necessary, and that's purely through these inundative releases. What you'll see here are cycles, and those cycles are associated with germination in springtime from a very, very long-lived seed bank. And we have a couple of components that we need to ensure for this type of strategy, and this is something that we're working on with sand parks at the moment. We have to have effective mass rearing infrastructure, and this is the Skakuza mass rearing facility that's currently being re-established. We need excellent stakeholder collaboration, and this is Nele with my colleague Kelby English at Hartbeespoort Dam, learning how to implement this entire strategy. And we have to have strategic release timing, which I'll get into largely because of these seeds. And that's a water lettuce seed, that's a water hyacinth seed. So here's a picture of the Rand water mass rearing facility that we've recently we've recently set up in order to combat the huge invasions on the Vaal River that started in 2024. So it's the operational backbone for biocontrol. It allows us to respond rapidly to new infestations. We can adjust our research and our protocols, and it allows for a lot of training as well as sustainability. Stakeholder collaboration, and here's Anele with her team on the Sabi, down near Lower Sabi, that's all water hyacinth in the background. It previously was water lettuce, so we get these changes in the dynamics of these plants. And this allows us multi-sector participation across government, institutions, conservation institutions, civil society. We can share our knowledge. Outside of the park, we have a lot of community engagement benefits, and of course, we can share our resources to make sure that this is cost effective. And most importantly, probably is strategic release timing. So as I said in Harderbeersport here, I showed you this beautiful picture, 7% in 2021, but October, there we start seeing the green stuff, and that's coming back from seeds. So we really have to align our releases with disturbances, and most of the invasions here in Kruger are in rivers. I don't know if that's an AI image. I got it off Facebook, and that's the Letaba River apparently in January. So you can imagine all those plants being washed out of the system, and we're back at square one. So we really have to coordinate our releases. And all of this this fits savanna landscapes because of this dynamic hydrology. And this is just where, this is the whole biocontrol pipeline. Where to from here? This isn't new to Kruger. This has been going on, Opuntia biocontrol in the early 90s with mass releases that cleared large amounts of Opuntia. And what about new targets? Parthenium. Here we've got guys wearing backpacks, spraying these when there are very effective biocontrol agents available that we should look at scaling up. 10 minutes on the dot. Thank you very much.
Time for 2 questions. 1 question. Corley?
Thank you, Julie. That was very interesting. I was just wondering, we do have a massive problem with Parthenium, and why are we not using biological control? You make it sound easy, but what are the hurdles? Like, how can we actually move forward?
People are the problem. Communication is the problem. We can set this up. It's very easy to do so. We've got the agents. It's just now talking between, it's not an insect that my organization works on. So we need to work with the right people, which is at plant health protection. Bringing them on board. Funding is a huge issue, but it's certainly, we shouldn't let that stop it because of the huge benefits that these little insects can provide.
Okay, thanks, Julie.
Thanks.
Okay, and we move on to the next presentation by Londiwe Makuna, and it's also nature-based solutions, but the use of large herbivores in managing invasive plants.
Good morning, everyone. My name is Londiwe Mokwena, and I am very excited to share some of my work from my PhD chapter. I first attended this conference doing my master's where I was speaking about grasses and C4 grasses and fire, and now I'm back again talking about invasion biology and mega herbivores. So it's been quite a shift for me. But when it comes to invasion ecology, we can all agree that not all introduced plants are problematic. But we can't ignore the fact that some do actually spread. They spread aggressively and extensively. This results in our native species declining, with changes in plant dynamics, changes in plant compositions, and overall reduction in biodiversity. And over time, these changes alter habitats in ways that are more difficult or sometimes very much impossible to reverse. And managing managing alien plant dominance, as we've heard from Julie as well, can be costly and often requires repeated interventions. Mechanical and chemical controlled approaches can be effective at local scales. However, they're sometimes difficult to sustain at a landscape scale. And going through my research, a very critical question that has come up is that why are some alien species more dominant as compared to others? And there's several mechanisms that have been proposed, but from my reading and research, what came up the most was the enemy release hypothesis, which states essentially that when introduced species come in, they don't potentially come in with their specialized herbivores or pathogens or insects. So this allows them to essentially grow freely and without any competition and do as they please. However, most research focuses on insects and pathogens. And despite the ecological importance of large herbivores, which we fortunately still have a lot of in African ecosystems, there's very much information on the interactions between alien plants and large herbivores themselves. And this raises questions for me, like, large herbivores have such a generalistic diet. Could they potentially be used to suppress alien plants by simply doing what they naturally do, which is move, feed, and disturb ecosystems? And this brings me to the aim of my chapter, where I look at the role of large herbivores and how, if they can reduce the abundance of young and small alien species, and if they can suppress seedlings or saplings. I did this by looking at reserves in the Waterberg as well as in Pretoria. I had 3 different treatments where I had reserves that had animals up to mega herbivore size. I had some that had large herbivores, and then I had some that had small herbivores. For both adult and youngs, I measured herbivory, trampling or damage, as well as recruitment in these reserves. And for the adults, in terms of trampling and browsing pressure, I looked at 20 different individuals per invasive species. I measured percentage browsing or trampling using a score, so we'd see how much a species or an individual is potentially browsed or trampled. And for the seedlings, I did 3 5x5 plots where we'd have a parent plant as a corner, and that's how we'd start moving towards to 5 meters and create a plot. Inside the plot, we'd measure the number of seedlings or saplings, as well as if those seedlings or saplings had any measures of browsing or trampling, which is similar to what we did for the adults as well. And in terms of my data analysis, I did a hurdle model where I looked at the browsing occurrence as well as intensity of how much they were browsed or trampled, as well as Poisson GLLMs for the recruitment. So when we get to the fun stuff, which are my results, in terms of browsing and trampling occurrence for both reserves, this means if they come across an alien plant, would they browse it or not? We find there's a significant difference between mega herbivores and large herbivores as compared to small animals, but only in Pretoria, well, not between Pretoria and Waterberg. However, when it comes to trampling, there's a difference between both as well, mega herbivores and large herbivores from small reserves that have up to small herbivores. So when it comes to the intensity of trampling, so given that they've occurred and they've come across invasive species, how much are they browsing? There's no significant difference between browsing and trampling. So they do come across, they do browse, but they're not doing any much damage to essentially suppress or affect these plants. So most of my models were there's an unexplained variation that comes to the species themselves. So when we look at the impact of mega herbivores or large herbivores on the alien plants themselves, there's variations between species. So definitely some species are more prone to browsing, others are more prone to trampling, and it's not uniform. So the impact is not uniform. It varies from species to species. And there's certain species that are not affected at all. There could be various reasons. That's the functional traits or the traits of the invasive species themselves, what they're secreting, or thorns, or maybe they're not even palatable for large herbivores. Then when we could look at the seedlings, we see a similar trend as well as the parents, where there is a difference when it comes to trampling and browsing occurrence. However, again, with intensity, there wasn't significant difference. Significant difference. And when we look at the recruitment, there isn't significant difference. So presence or absence of large herbivores does not impact the recruitment of seedlings. So seedlings are still able to escape and establish regardless of their presence. So in summary, what we are seeing here is that suppression is possible through browsing. There's a bit, but only in occurrence, they don't damage the species as much. Trampling also occurs, but only for seedlings. But we do see that there's definitely differences between species. So it's not a one-size-fits-all thing. We have to now consider looking at different specific alien plants and interactions between those two. Which brings me back to my first question. Can large herbivores suppress invasive plants? And the simple answer to this is only in adults and on incidents, but the effect could not even, is still so small. It could just be specific species that have this impact from large herbivores. And an amazing example of this is one of my chapters where I look at the actual diet of mega herbivores, if they include invasive plants, and I do eDNA analysis. I collect dung, and 8 out of 10 of black rhino mittens, I find Datura ferox, either the seedlings or established adults that are either trampled or browsed. So there's a clear relationship between Datura and black rhino. And it also poses something that I haven't discussed in this presentation, which is the dispersal effect of mega herbivores when it comes to alien plants. So they could not potentially be suppressing all of them, and some could be spread by mega herbivores. So the effects are very small, but with the right plant and the right mega herbivore, we could see some interaction. We could see something, especially with mega herbivores just eating, feeding, and defecating. But again, this is not a silver bullet solution. So we have to be realistic and think about what it actually means. This is one of my sites in KZN in Itala, where everything green there is actually lantana. And I can't fathom any amount of large herbivores that could go in there and trample and cause significant difference in these systems. So we have to think about the resilience of a certain threshold where it might be beyond this impact. But going after Julie's talk, there's a bit more hope now, seeing that there's some insects that could potentially help in these situations, but I'm not seeing as much when it comes to mega herbivores as yet, but there's still a lot more that I am looking at and building into this study. I would like to thank my amazing field assistants that made this research possible, my amazing supervisors, collaborators, as well as funding that allowed for this work to happen. Thank you so much.
Thanks very much for a very interesting talk. Are there any questions? We have a bit of time.
Hi, thanks for that. And did you only find the Datura in the black rhino mittens or in white rhino mittens too?
Initially it was black rhino, but I just came back from Marekele a week ago and I found it in white rhino. I was surprised because I think white rhinos are also grazers. I didn't expect to find Datura there, but for the first time I found it in white rhino as well.
Okay, yeah, I've seen it a lot in Plantsburg too, and all the white rhino mittens too.
Any other questions? So I have a question. Presumably in many of these reserves, or there would have been a full complement of mega herbivores or small herbivores that you tested. So these were introduced through some mechanism, but they were still able to overcome the effect of these suppressing herbivores. Do you think that there's some other mechanisms or things that allow them to get through this if there's some effect shown on some of the plants?
On some of the alien plants. I definitely think there's a relationship between the functional traits and what makes some of these alien plants suppressed or dispersed. So it's definitely a lot more than just the mega herbivores that allow them. What it is yet, I'm still in the process of trying to figure out.
Okay, great. Thanks very much. Okay, we're going to change tack a bit, and we're moving on to the freshwater side of invasions. And Josie South will talk about freshwater crayfish.
Lovely, thanks, Llewellyn. Hi, everyone. Yeah, so as Llewellyn said, we're going to shift onto the crayfish side. So I'll initially take the chance to apologize for the deluge of crayfish talks you're going to get this morning. I'm going to be sharing with you the effects of non-native invasive red claw crayfish in the Brotsi floodplain in Zambia. So, loss of biodiversity is very much a national security problem. Anything that is affecting biodiversity change has ripple-on impacts to food security, people's livelihoods, well-beings, and all of those cascading impacts that come from this. I'm specifically going to be talking about crayfish, and what I want to tell you right now is that crayfish invasions globally cost around £5.6 million a year. This is a really small underestimate. This data doesn't include any information from Africa. It is solely from the American continent as well as Europe. So crayfish are really interesting. There are no native crayfish in continental Africa. You only find them in Madagascar. So this means that crayfish are entirely phylogenetically novel. This means we don't really understand how they may or may not fit into the current African freshwater ecosystems. What African freshwaters have instead are bottom-and-ortered crabs. So bottom-and-ortered crabs, as well as crayfish, are large-bodied shredders. They're technically functionally analogous, but we don't actually know whether they truly are functionally analogous. So this gives us a really good opportunity to actually understand the ecology of freshwater systems in Africa a little bit better. You have two main crayfish species invading southern Africa. These are the red swamp crayfish, which are coming from the southern part of the States, and the Australian red-clawed crayfish, which are coming from northern Australia as well as Papua New Guinea. So red-clawed crayfish are invasive throughout the majority of southern Africa currently. I'm going to focus only on the invasion in the Upper Zambezi. They were introduced into the Upper Zambezi initially to the Kafue Flats and Lake Kruger in around 2001-2002. In 2014, another invasion was found in the Brotsi floodplain. The Brotsi floodplain is this part of northwestern Zambia. It's an Upper Zambezi floodplain. It's absolutely enormous. It's an awesome system to work in. However, it's also incredibly challenging. What you're looking at is a series of flooded grasslands, as well as main stem river, as well as canals, ponds, all sorts. And the invasion is currently spread throughout this entire floodplain. So the floodplain runs from Lekulu in the north to Sanangra in the south. This study was done in 2019, which is 5 years after it was found in the Brotsi floodplain. So the introduction site is right in the middle in Mongu. It was initially introduced by road workers building the Mongu-Kalabo floodplain road. They were cultured in a culvert. As soon as the floodplain was flooded, as it does every year, everything was washed out. So our job was initially to find out where they were, what they are doing to humans as well as ecology. So we did an assessment across the entire floodplain to understand exactly how they're affecting people's livelihoods. So the crayfish will come and they'll scavenge on the gill nets. They'll eat parts of the fish then become not able to be sold because no one wants to buy something that's been hashed up. So we're estimating that just from direct damage alone, the crayfish cause around $20,000 US worth of damage. This is incredibly significant because a fish's annual income every year in the Barotse floodplain is below $300, and this is for a household. So we also wanted to understand whether there are sublethal effects on fisheries. The Barotse floodplain is a fisheries-reliant area. People need this directly to eat. It is one of the most important sources of protein. We need to understand how they're changing the food web. So we did a stable isotope assessment. We went from the bottom to the top using the top and the bottom, Lukulu and Sananga, as our upstream and downstream controls, compared to Mongo, which was the only invaded area at the time. We want to understand, is there an empty niche? Do crayfish fit into a different part of the food web that the crabs aren't occupying? Or actually is there direct competition? So this is an example set of isotope plots that show what may happen if there is competition between crabs and crayfish in the Barotse floodplain. So this shows a stable state without invasion. Then the invader comes in, which would be this orange niche. This shows direct competition between the crabs and the crayfish. What we expect to see after this is then the invasive species may expand its niche, start to feed on a wider variety of things to avoid direct competition with the crabs. However, then in response, the crabs may shrink their trophic niche, which then may put them in a vulnerable position. Afterwards, if there is some adaptive plastic potential here, they may then re-expand their niche to offset some of the competition by the crabs. So this is a hypothetical scenario, which would see if there is not an empty niche. So this kind of hellscape of a graph, what you need to see is the yellow point here in the middle, which is Mongo, the invaded area, represents the crayfish. If you look at the upstream and downstream controls, especially from Lukulu, you can see that the crayfish do seem to fit into an empty niche just above the crabs, but between the fish. So that would suggest that maybe actually there is a slight empty niche where the crayfish can fit in, which would suggest that the native freshwater crabs are not directly competing with them. This isn't the entire bit of the story, however. This kind of insinuates maybe the crayfish aren't causing so much damage. So we looked a little bit further. There's no difference in the size of the crayfish and where they're feeding across the trophic niche. So the trophic position does not change from a small crayfish to a large crayfish. This was kind of surprising, but I'll come back to this. So what we also looked at was compared to the up and the downstream control, is there any difference in Lukulu versus Sananga? You're looking at a 200-kilometer gradient, which is quite big. So there's a little bit of difference. The niches of your species, of your fish species, are a little bit larger upstream, but not so much that we need to pay too much attention to this. This shows that we've got quite a good upstream and downstream control. The important thing that we're seeing is actually between the up and downstream controls and the invaded site, the fish's niches are shrinking. They are contracting what they're feeding on and restricting the diversity of foods that they're intaking. The crabs, on the other hand, are expanding their trophic niche. So this starts to look a bit more like what our hypothetical invasion scenario would look like. To go into this a little bit deeper, looking at niche sharing within the invaded site, we can see about 40% niche sharing between the mormyrids, the freshwater crabs, and your cichlids. Some of this was expected between the crabs and the crayfish. The heavy niche sharing between the mormyrids wasn't expected, and this really highlights that this is a group of species that we need to be more concerned about. So I've just shown you that there's around 40% niche sharing between these species. However, if we look at a study that was done in the Komati River in South Africa, there is a much higher degree of niche overlap between red claw crayfish and freshwater crabs. This is likely because we're dealing with two very different systems. The Barotse floodplain is incredibly intact still, and it's also got a lot of resources. In comparison, the highly damaged Komati system probably doesn't have enough resources, so they're all having to overlap. We also don't see very much change in trophic positions between the fish. This shows that the ecosystem in the Barotse floodplain is still relatively intact. We're not seeing too much restructuring of the food web. However, we are seeing a slight increase in crab trophic position. This means as they're expanding their niche, they're also becoming more predatory. They're feeding higher up in the food chain. This is quite interesting to dig into because we don't know what the outcome of this might end up being. In total, we're dealing with increased complexity within the food web. We're getting an insertion into the lower part of the food web by the crayfish. We're showing some adaptive plasticity in the crabs where they're increasing their resource intake. However, your fish are decreasing their niche, which also indicates that maybe in the future, this is making them more vulnerable to other pressures. We don't know how this will progress. This was done at the 5-year timestamp, so we're going back this year in October to redo this study, which will give us, I think, a 12-year timestamp. Now we know both the upstream and the downstream areas are invaded as well, so we have a before and after. So I'll leave that there, and I'd like to thank SIAB, WWF, also the Barotse Royal Establishment, and my wonderful field team. Thanks.
Thanks, Josie. And we have some time for questions.
Thanks for a very interesting talk. I was just wondering, we were in Madagascar quite a few years back, and one of the delicacies, should I say, was freshwater crayfish. So I was wondering if there's any reason why nobody's eating these invasive species.
Sure, of course. So actually people are eating them. However, the fishery's taken around 20 years to develop. So the Kafui now has a small fishery for red claw crayfish. But the problem is, like, crayfish aren't indigenous, and also when you're dealing with landlocked countries such as Zambia, Zimbabwe, it's not a common thing for people to want to be eating. People find it incredibly alien. So we've just finished a survey actually around Kruger and also the Barotse floodplain. The main reason for people not eating them is superstition. We also don't really want people to be having a massive fishery for this species. People have an affinity for the native breams. We need to be working on some sort of nature-based solution to bring back that fishery while reducing the crayfish fishery. It is, however, really good for fertilizer or animal feed. Thanks.
Any other question? Okay.
It's a little hair-raising. My understanding, and I may be incorrect, is that there were similar invasions in Kenya and East Africa, and that otters had a huge impact on the invasive crayfish. So are there any natural predators in other systems?
So can you say the last bit again? I can't quite hear you.
I said, apparently otters had a huge influence on the invasive crayfish, and native predators were limiting their spread. Is that possible elsewhere in Africa?
Yeah, things will eat them. However, at the abundance that we're working with, so you can sort of walk across the Barotse floodplain and pick them out. You know, you can pick up 50 in a 10-meter stretch without really trying. I don't think native biocontrol is going to help, unfortunately.
Okay, thanks, Josie.
Thank you.
To the next. The next presentation stays in the theme, but predicting the spread and management. Thanks, Victoria.
Thank you. Hi, everyone. I'm Vicky, coming from the University of Southampton to present you how we may use predictive approaches to better understand an invasion and simulate some management strategies. We have done this on the invasive red claw crayfish. You're going to hear quite a lot about the characteristics of the species in the next talk. So I'm going to go and pretty much talk about the potential that these predicted approaches may have when dealing with invasion, in terms of like talking about prevention, talking about management, even learning from the characteristics of your model to understand which traits of your species may be driving a successful invasion, sort of feeding back in those hypothesis testing that then you can sort of do in the field, and that will come into that feedback loop, feeding back into the model, making it better. So what you have on the screen, it's a map of the Invertebrate basin with the two initial sources where we had the red-clawed crayfish getting into the system. The red dot represents the earlier invasion that happened in 2016, so they escaped. And then the orange dot at the top, we hypothesize that had happened slightly later in time, around 2022. And let's see what the model can basically tell us about that. So our aim in this exercise, which is still ongoing, so it's very preliminary at the moment, but what we are trying to understand is we have two sources where that crayfish enter into the system. We have data, monitoring data from a couple of years, and can we recreate a model that is able to show what we have now in the Decomposition basin as per 2026? Can we then predict the future, and can we do anything about it? Can we manage and control the invasion? How we have done this, we have used what is called individual-based models. So we are looking at the population from the individual perspective. So we are recreating the life cycle of that individual crayfish from the birth. They will have that sort of growth until they reach maturity. They may die at some point in life. And then you also represent the actual movement from that sort of decision of leaving the natal patch to that actual movement until they find whatever side that may lead that invasion front. And the answer to the first question about can we actually represent the invasion, we actually did this exercise simulating a couple of scenarios. One was with using just the very first introduction in 2016 and see if we could actually represent the spread that happened in that sort of upstream bit of the river as well. And the model was really struggling to be able to get from that point in background all the way up to here if we only had one sort of introduction. So it was already pointing us that that hypothetical second introduction probably happened, otherwise we wouldn't have the crayfish up there. And the map what you see on the screen now is what we think is the current distribution of those invasive populations. So they are pretty much restricted to those two sources. So anything in red is where we are pretty confident that we have a good established invasive population. How is the future going to look like? And again, this is ongoing work, but it's looking quite horrific. It sort of follows a pretty usual type of invasion where you have a very steady initial stages as with any logistic growth sort of function. So in 20 years time, we may still be quite restricted. So maybe only around 10–14% of the catchment is actually invaded. But in 50 years, we may be having a horrific situation. So that's what we want to avoid. And now we're going to do the management. So what do we do? Can we simulate some strategies? And I'm going to explain a little bit of how we actually did this. We pretty much work towards two types of a scenario, and this is all based on cooling. So we are targeting the removal of the individuals in selected sites. And the first strategy is try to pre-identify sites where we may be able to engage with community to lead the removal efforts. So you can see that we sort of selected few towns around the Kruger National Park. And the initial simulation, it's just assuming they are gonna just go once out there to remove the crayfish per year. So not a lot of resources being spent for this particular scenario. You then parameterize the type of cooling that you may want to have depending on the confidence that you have about your method, whether that is trapping, whether that's electric fishing. You will have to adjust that probability of catching, but it's pretty much going to follow a logistic function where if there is not much in the system, the chances that you are actually trapping something is quite low, while if it's so fully invaded, you would expect to have much more chances to remove, but you are never going to remove like the whole lot from that side. This is the result from pre-identified sites. So actually there is not a lot of changes compared to that first simulated do nothing strategy that we saw earlier. This could be linked to many things. So basically if you just have one site once a year that don't do anything, and you may also have the species learning about that sort of trapping that you may do, so they may avoid the areas that you are gonna yearly try to cool. The second type of strategy is where we are adding some sort of random component. So we are going out into the field and we are trying to randomly change the sites that we are trying to remove crayfish from every year. I have done that. This time we are using 10 sites a year to increase a little bit more the resources that we're spending. And then we have done 3 type of strategies. One where we just go random across the whole system. One where we are focusing on cooling randomly but on the invasion front. And one where we are aiming to actually do a more sort of conservation approach and just try to protect the Kruger National Park in this case. And what we get is that the invasion front and the trying to protect the Kruger don't really have a lot of difference in 50 years, but the random cooling, it seems to be behaving slightly better. This is interesting, and it's a first step to try to understand how we may simulate better strategies. And I'm hoping through this meeting I'm going to be able to have some conversation with you to understand resources, to understand management a little bit better, and then we can do a better job. As I said, this is exploratory as it is at the moment. But so far, that secondary introduction in the area of exotic weir has been supported by the modeling, as we saw earlier. Currently, we are in a situation where we are, the invasion is very much constrained into 2 areas. So it's time to be thinking, we should do something or start to think about doing something now before we get into that step growth that may happen as the year goes. Localized effort so far seems to be not very effective. We will keep exploring those scenarios. We will keep improving the model to try to better understand how to do this. But it seems that random cooling, it has some sort of effect on that population dynamic at the way that they are able to build back that establishment on those sites that we have cool, with that first scenario actually reducing the occupancy of the crayfish by nearly 60%. So effort matters, but the spatial distribution of that effort is quite important. And that's it from me. Thank you very much.
Thanks very much. We have a time for a question. Tertia?
Thank you so much, Vicky. I was wondering, with the systems further north of the Sabi, because the sand driven makes sense. They'll come through Exeter on the west, but with Nwatitsonso and Nwanetsi systems, are you thinking that they're going to establish a lot in the Mozambique side of the Komati and migrate upwards? Is that how the model predicted their spread to those systems in the north?
You mean upstream of the system, isn't it? So as the model is built at the moment is using a non-directional sort of kernel type of movement approach. So it will go in either direction, doesn't matter what we are building on top of that. Hopefully in the next few months, is to have that sort of sense of directionality where we are able to take into account high floods, barriers, and so on, so we can mimic better that reality scenarios. So I would say the upstream sort of like movement as it is now shown there, we need to take it cautiously because obviously they are gonna spend much more energy to try to go up than what they would do trying to go down of the system.
Okay, thanks very much, Victoria. Okay, and we move on to our next presentation, also on the spread of red claw crayfish. Thanks, Danny.
Perfect. Hi, everyone. This is my first network meeting, so I'm very excited to be here today, especially because my talk will be focusing within the borders of the park. So you've already heard quite a bit about the threat of red claw crayfish in Southern Africa from Josie, and also what the future might look like with the crayfish moving out in and out of Kruger National Park from Vicky. So I will be taking you a step back and give you some information on the species based on some work that we've been doing in the last couple of years in the park. So the red claw crayfish, as Josie mentioned, that originally native to Australia and New Guinea. They are invasive in many parts of the world, including Southern Africa. They have a very characteristic morphology, which gives them their name. They've got this red patch on their claw. They're also omnivores and detritivores. They're highly fertile, and they're a very popular aquaculture species, which is a main pathway by which they've been introduced in other parts of the world. So what is the problem? So Josie has already talked about this a little bit. They are omnivores, so they will interact, feed, compete with many levels of the aquatic food web, and in areas of special importance, protected areas like Kruger National Park, where there's high biodiversity, this can be very, very problematic. So Josie mentioned the threats to fisheries and all the socioeconomic costs that are associated with that. And this is a major threat, but in places like Kruger, perhaps the more important threat is what they're doing to native biodiversity, how they alter the ecosystem, and the potential to transfer novel pathogens. So I don't think I need to stress how important it is to care for national parks and areas such as the Kruger. You are all here. And I'm also very happy to tell you that you are also halfway through the crayfish talks, so bear with. But where are the crayfish in the park? Where are they hiding? That is the million-dollar question. So the original effort, led by Josie, was published last year. So there was this major effort to sample the entirety of Kruger National Park, so starting from the top, going all the way down to the bottom to find out where the crayfish are in the system. And thankfully, the crayfish were only found in the lower part of the Kruger, which is why last year we went back and resurveyed the Sabi Sand rivers and the crocodile to see how fast are they spreading, how are they changing, and get some more insight as to what they were doing and what might be driving their spread. So to give you a little bit of a timeline, Vicky briefly mentioned this. For Crocodile River, we have an introduction date of approximately 2016 in the Van Graan Dam, which was the first record. And since then, we've seen crayfish move both upstream and downstream of the river. And for the Sabi Sand rivers, we believe now that there has been a secondary introduction next to Weir just outside in the Sabi Sand Reserve in 2022. And we have since seen the crayfish move downstream all the way to Lower Sabi last year. And I believe Vicky also mentioned the word front and core. This is what we refer to as the invasion gradient, and this is what it looks like for the Kruger National Park. So we have marked our introduction sites as the invasion core. Sites that are immediately upstream or downstream are our intermediate sites, and then the invasion front is basically the furthest point that we've detected crayfish downstream of the river. So how do we actually know where the crayfish are? How do we record them? We normally set traps in the evening. We're going to collect them in the morning and see if we've got any crayfish. The two important metrics that I want you to keep in mind are the catch per unit effort and the detection probability. These are proxies for abundance, catch per unit effort similar to what is done for fish, to give us an idea of how many crayfish we've got and to be able to standardize and compare between sites. Once we have a crayfish at hand, we determine the sex, the mass. We take some key morphological measurements that you see here, mainly the carapace, claw, and leg lengths. And we also take some tissue samples for some future genetic testing, which is a work in progress. So, where are the crayfish? In 2024, this is what the situation looked like for the Lower Kruger. And in 2025, when we extended our survey into the Sabi Sand Reserve as well, we also detected crayfish in two further sites, Exeter Weir and the Lower Sabi, where they weren't present previously. Now, a very interesting result that came out of our monitoring effort was the difference between catching crayfish the way we intended, which was the trapping, and the bycatch of crayfish when we were electrofishing for the standard monitoring surveys to look at fish biodiversity. So in some cases, you can see here that we actually caught a lot more crayfish accidentally by electrofishing than we did with our trap. And that can have serious implications about how we manage and potentially remove crayfish and the ways, the solutions that Vicky shared within her presentation about the different scenarios. The first important difference is the bias that we see in the size between the two methods. So we seem to be missing out on a lot of those smaller crayfish that are running around and hiding if we're just looking at the traps. And the other very important bias is the difference in sex. So we're missing out on a lot of females that could be breeding and we're not catching because we're seeing a lot more males in the traps. So how fast are they spreading? Vicky showed you the hypothetical scenarios, but what do we know from our 2 years of surveying? So we've estimated a spread rate based on where they were found and how they could move a path of like least resistance through the path, through the park. For the crocodile, we've estimated a spread rate of approximately 4.5 kilometers a year. That changes for the Sabi Sand to 22.9 kilometers, so almost 23 kilometers downstream a year. And if we look at the difference just between 2024 and 2025, where there's been a lot of rain, and you saw the flooding this year as well, so I'm too scared to look at what it looks like in 2026, they actually made it 35 kilometers downstream in just that 1 year. So in terms of catch per unit effort, how many crayfish we caught, this is the difference between 2024 and 2025. You can already see that we've got a lot more crayfish. And just to give you some numbers for the crocodile, that is a 3 to 5 times increase in the catch per unit effort of crayfish just within a year. And that is true that throughout the time that the crayfish have been present in the park, we've seen a steady increase in both the catch per unit effort and the detection probability. But how are the crayfish changing? Are we seeing any change in the crayfish themselves? Interestingly, we saw a massive increase in size, and that's true both when we're looking at their mass and their length. You can see the gray bars are the other invasion cores. The one immediately before the Kruger is the Barocci that Josie presented earlier, but you can still see how much of the difference we see in Kruger compared to the other cores. Even though we're seeing a similar pattern of increase from one year to the next, this is much higher than anywhere else. And that is very evident in the earlier parts of the invasion, so mainly in the core and the intermediate. And that gives us some clues as to what might be driving this spread. So if we look at data from the Kruger and the Barocci, there is some small evidence of intraspecific competition, perhaps, that might be driving these changes in size and why we're seeing different sized crayfish along the invasion gradient. So for the core and intermediate sites, the claw length and leg length and carapace length seem to be more important. Well, when we move to the invasion front, we're seeing those bigger individuals driving the invasion. And two other very important factors were the distance from the introduction site. So the further away we are from the core site, the less crayfish we see. But more importantly, the average rainfall. So if we had more rain previously, we are seeing more crayfish, which again has implications about how they might be spreading with those intensifying rainy seasons. So overall, crayfish cast a large shadow on the conservation of the parks, the biodiversity, and I'm going to leave you with some key messages. We are seeing a rapid spread and establishment within Kruger National Park in just a few years, which combined with Vicky's models is a terrifying scenario. We think that extreme weather may further facilitate their spread, and there is some evidence for competition and body size increase along the invasion gradient. So ultimately, there is excellent monitoring work being done in the park, but there's an urgent need for further monitoring of the crayfish, impact assessment, and management. And you'll be hearing a lot more about the impacts from my colleagues that are speaking right after me. So I think this is time. So a huge thank you to everyone that has contributed to this presentation. It has been a tremendous team effort to get the data that you saw up here. So thank you very much.
Thanks very much. We have time for a question.
Hi, great talk. I don't know much about crayfish. Can you say, so when you have the differences in the electric versus your traps, is it because that female crayfish are usually smaller, or is there like a behavioral component, do you think?
Were you asking about the crabs? Or the traps, sorry.
The trap, like
the
the electric trapping versus the actual trapping that you were doing.
We're not sure, so we are actually looking into it. The one explanation about why we're getting more is because we use baited traps to catch them, so it makes sense that you've got those bigger males competing for the bait and entering the traps. But I actually started noticing some differences when I was playing with all of our data, and when I incorporated the electrofishing data, I actually saw what bigger difference it is. So it's something that we will be looking at, because we are seeing a similar pattern in other sites where we're monitoring crayfish, including the UK, for example. So I'm personally very curious to see if that is widespread as a pattern and what it could mean. But my main concern is just the implications for removing the crayfish and getting the full picture.
Okay, thanks very much. We'll move on to the next one. Our next presentation will be by James, and look at the impacts of the crayfish.
Yeah, hiya guys. Ooh, hello, sorry about that. Sussing this out a little bit. My name's James, I'm from the University of Leeds, and I'm part of the South Aquatic Interactions Lab. So, yep, like's been sort of talked about a little bit before. I'm going to be talking about the impact of the red-clawed crayfish on fish within the Kruger National Park. So, provide a bit of background. This is going to be brief because we've had it done quite a lot already. We've got the red-clawed crayfish, they're native to Australia, introduced into the park in 2016 in the Crocodile River. They've been seen in the Sabi in 2022, and they are polytrophic omnivores. Essentially, this just means that they feed on everything, on plants, organic matter, hippo dung, crabs, and also predate, consume, and compete essentially with all of the levels of the food chain that they can get their grubby little claws on. I'll be focusing on fish for the moment, and this is particularly important within the national park because it falls within the low-veld biodiversity freshwater ecoregion, which has a particularly high native fish biodiversity. I think it's about 63. Please correct me if I'm wrong later. It's also, and we think that they're competing with these fish and potentially predating on their young. As well as this, and as you've seen from Josie, they also have a really negative impact on humans, whether that's through subsistence fisheries or economic sort of fisheries. Although since such an issue in the park, there are fisheries in community land and in buffer zones, and there are fisheries both upstream and downstream of the park. So it's worth bearing in mind. So this project aims to look at what species are being affected. As first, sorry, I'm really struggling. I'm moving around too much. So we want to know what species are being affected, whether communities are different between different sites depending on the abundance. So we're predicting that a higher abundance of crayfish will see different communities of fish. We also want to know why these species are being affected. So this involves using functional traits. If you're not familiar with that, it's just about how an individual interacts with its environment. In the case of fish, it's what level it is in the water column. Is it sort of open water? Does it mainly spend its time in the bottom? Or how it reproduces? Does it scatter its eggs along the river floor? Does it build nests which it guards? Or does it brood in its mouth? So we're predicting that there will be less clutch tending, nesting, and guarding species with more crayfish. And this is because the crayfish represent an additional predator and there's more energy expenditure used to fend from them. And then finally, we want to know when these fish are being affected. So when in their life cycle? Is it when they're adults, or is it when they're juveniles, or even as eggs? So we predicted that, again, with higher crayfish abundance, we would see less recruitment, so less young fish. So methodology, very briefly, sampling took place across 2 years. We're presenting results from multiple sites across 3 rivers, the Sabi and its tributary, the Sand, and the Crocodile River. And we collected crayfish fire trapping and fish through electrofishing. If you want to know anything more about that, please ask me later. So moving on to the why. Why are fish being affected? So just to provide a little bit of context here, you can look at individual functional traits, and you can also look at these functional metrics of like ecosystem health. And a healthy functional ecosystem is one that's resilient to stresses. It provides ecosystem services like water purification, and it's quite productive. So obviously, if we've got a less healthy, less functional ecosystem, that's a bad thing. Fortunately for us, at the moment, we see no significant effect of crayfish on the functional community metric. So currently, the functional health of systems, particularly fish-based systems, is not being affected, which is really, really good news. However, we also need to think about, and we can also, whilst there's nothing at a community level, it's worth thinking about it at individual levels, and we can pick that out as I go through and I talk about what species are being affected, which brings me nicely on to what. So what species are being affected? Obviously, there's an odd one out on this slide. But moving on, again, at the community level, on taxonomic composition, so the collection of species we're seeing no significant effect of crayfish, which is interesting. So at a higher level, we're not seeing anything. However, for individual species, such as the elephant fish, this Marcasinus pongalensis, which is my personal favorite, so this is a bit gutting for me, and Tilapia sparmani, we see that their presence or absence is significantly associated with the presence or absence of crayfish. Very basically put, when crayfish are present, they're rare, and when crayfish are absent, they're a lot more common. It's important to note that both of these species guard their young. The elephant fish hides their clutch, and the Tilapia sparmani builds a nest that it guards. So just bear that in mind for a little bit later. So finally, moving on to when. So this is when fish species are being affected in their life cycle. And to remind you, we're expecting the young to be eaten. So I'm going to present a hypothetical data set here, and this is going to be explored using length frequency histograms. So on the left here, you can see we've got count of sort of different size classes of fish from small to large, and the density plots is exactly the same data, but it just looks a bit nicer. So this is what we would expect to see if we see no effect of an invasive crayfish on fish recruitment. So you can see that the invaded site in red has very similar patterns to the uninvaded site in blue. However, if we do expect to see some predation, if you look there, look where this arrow is, we'd expect to see a drop-off in smaller size classes and a change in the shape of the density plot. Really, with the density plots, when it's less nice simulated data, there's a bit more of a dramatic change, and you're wanting to look for where the peaks are occurring. So moving on to real data, and I'm going to start with a couple of examples. Or my first example is one where we don't see an effect. So this is the Mozambique tilapia, or Oreochromis mozambicus. And you can see here that across the invaded sites, so Antallista, Lower Sabi, and the sand, we see that they have very similar length frequency distributions to the uninvaded sites, which are in blue here. And again, you can sort of see the peaks are all at roughly the same locations. So we see not much of an effect on Oreochromis mozambicus. The story starts to change when we shift on to redbreast or Coptodon rendali. You can see that in the two invaded sites, we're missing a size category that is represented in the uninvaded site, and you can see this peak here that represents that. So there's some loss of this smaller size category here. A very similar pattern was seen for the yellowfish, which is obviously a common angling fish here. And yep, lost my train of thought there for a second. Moving on to Enteromius radiatus. So this is a nice, cute little fish. And actually, unfortunately, it's one of the ones which we see one of the most dramatic effects on. So you can see this whole size class here that's not represented at all in any of the invaded, well, not represented in this invaded site and represented much less in this site. And there's this massive drop-off in these smaller sizes, which is indicative of quite a high level of predation of the younger fish. What's also interesting is that sand has about 4 times as many crayfish as Antallista does. And you can see that represented in the complete loss of these site classes here, whereas there are still a couple knocking around here at Antallista, which is interesting to bear in mind. So, to summarize the length frequency bit, because obviously that's the meatiest and the juiciest bit, what we see is that egg scattering species and guarding species seem to be seeing a loss of recruitment across the sampled sites. So the young are dropping out there, which is not a good thing. However, things like Pseudochronablis philander, or the southern mouth brooder, and the Mozambique tilapia, which is also a mouth brooder, are not being affected at all, which is really, really cool, because what we're seeing is the things that have really well-defended fry or young protected in their mouth are not being affected by the crayfish much at all. But guarding species, which have to defend on the regular from these voracious sort of little guys, are having a lot more of a negative effect. So there's hope as well, because one of the endangered species in the park, Oreochromis meridianus, is, based on a brief Google I did this morning, also a mouth brooder. So that's positive, at least in the moment. I'm getting nodded up by Dummy, so that's encouraging. So, to summarize, we currently see no effects at a community level, which is great news. We see 2 species at particular effect, one of which is my favorite. This is much worse news. And there's a loss of recruitment across a range of different species, particularly egg scattering species and guarding species. So either egg scatterers are just predated, and guarders, there's probably a level of predation going on the young, and also increased energy expenditure due to needing to stop the crayfish from getting in. So there's subtle effects of a crayfish that have not yet become dramatic ones. And actually, that's a really, really positive note. I know we've had all these scary figures of spread and sort of possible abundances, and there is a predicted increase in abundance, but actually right now is a really, really good time to act, because they're relatively low in abundance and the effects haven't become dramatic yet. So if we act now, we can have a really positive impact and protect these absolutely gorgeous rivers and this sort of gem of hidden biodiversity within the park. Thank you very much for listening, and thank you for everyone who's contributed to this, including the guards, and Isaac and Wilson.
Thanks, James. We have some time for questions.
We actually are we're fighting over the mic, Ben and I. So I'm taking it first. You were telling us about fish, but I was just wondering about crabs. Are you also? I mean, is anyone looking at the size of crabs in these invaded and uninvaded areas? Because it sounded from Josie's talk as if, you know, there'll be real competition.
Can I just pause you there? You're going to love the next talk.
My question was really around management options. You say we need to act now, but what can we actually do?
Yeah, I wondered if this might come up. So this is an ongoing conversation at the moment. We've got a workshop at the end of this week to talk about it. But like we talked, Elizabeth, Vicky's covered some of the things. So there's culling, there's trapping. There's also part of this research is identifying where impact might be greatest. So there's potential to prioritise stuff there. And also thinking about monitoring things. If we know that currently the upper rivers aren't being influenced, if there's regular monitoring, if we do detect them, that's a time where you can get in and quickly remove them. So there's things like that. But obviously, it's not in set in stone yet, and it's a very, very complex problem. And that's why we've got like 25 different people coming to talk about it at the end of the week. So it's ongoing. We're working on it. And the crab question as well. I think it's covered a little bit, but I'm sure we can talk more about it later if you'd like to.
Any other questions? Quick question?
Well, I can bore you with more fisheries graphs, but I don't think we want that.
All right, thanks very much. Our next presentation is by Emma Baker. This is looking at predicting impacts using macroinvertebrates. Thank you.
Last graphic talk, I promise. So, how do I do this? Oh, there we go, that's better. Hi everyone, my name's Emma and today I'm here to talk to you about the impact of red claw crayfish. Sorry, I'm a bit short for this microphone. I'm here to talk about the... Let's start this again. So my name's Emma and I'm here to talk about the impact of red claw crayfish on macroinvertebrate communities in Kruger National Park. So macroinvertebrates are things that you can see on the right-hand side. They're things like dragonflies, shrimps, snails, and all of these things are the foundation of aquatic food webs. Lots of things rely on them for food, and they're also really important for things like nutrient cycling in freshwater environments. And these things can also really indicate river health because the composition of the macroinvertebrate communities can tell us things about the anthropogenic stresses that they might be experiencing. And a way that we do this is in South Africa, there's a standardized biomonitoring protocol of macroinvertebrates called the South African Scoring System, or SAS. And basically, different types of macroinvertebrates or families have different scores based on their sensitivity to things like organic pollution, but also gives us a really nice community composition for monitoring sites as well. And importantly, monitoring using this SAS protocol started in Kruger National Park in 2010. And as you've already heard, crayfish are potentially detrimental invaders. From a macroinvertebrate perspective, they can be directly competed with, they can be predated by crayfish, but the crayfish can also modify their habitat. And as we've already heard from the other talks, the first record of red claw crayfish was on the Crocodile River in Kruger National Park, and this was in 2016. And since then, we've found them on the Sabi and Sand rivers in 2022. So this project aimed to identify hotspots of macroinvertebrate diversity across the national park, because this is going to be really important for prioritization of management. Determine whether macroinvertebrate communities are responding to the crayfish invasion, and also identify which taxa are maybe being disproportionately impacted by the crayfish. Hello. So let's first look at the diversity of macroinvertebrates in the park. So here's a map that I produced using the biomonitoring data that I just mentioned, and each point on this map represents a different monitoring site. And the colour of them represents the richness or the different number of macroinvertebrates that are present. So a lighter colour indicates lower richness, and a darker red colour represents a higher richness. And we can zoom in on the rivers in the park. So this is a Lavuvu at the top of the park. And now the circles are different sizes to represent the sampling effort or the number of years that that's represented in the biomonitoring data set. And we can see this sort of has quite a medium richness, quite high richness. And then we move down to the Oliphants and the Letaba rivers. They have quite medium richness too. But if we move down to the Sabi River, we can see a lot of red circles, which is good because that means that's a real hotspot for diversity across the park. And if we look at the Crocodile as well, that's also got quite high richness. And importantly, both of these lower rivers have been invaded by red claw crayfish, as you've already heard. And just to quickly point out, work has already been done on this biomonitoring dataset. Satole et al. Looked at the SAS score, so that pollution monitoring data that I mentioned, and they found that SAS scores are different between the rivers, but not over time, and I think my richness graphs really echo this as well. So we've looked at the biomonitoring and the richness, but how are communities responding to crayfish invasion? So to answer this question, I've set up the following experimental design. So we know that the Crocodile River was invaded in 2016, so I'm treating anything pre-2016 as uninvaded and anything post as invaded. And similarly for the Sabi and Sand rivers, we have the 2022 threshold, so pre-22 is uninvaded and anything post is invaded. Oh. Hopefully that's going to come back. So we want to be sure that anything that we do see happening on those rivers is due to the crayfish and not any other environmental factors that might be influencing the communities. So to tackle this, I've used the Letaba and Oliphants rivers as controls because these rivers don't currently have any crayfish on them as far as we're aware. And I've used a threshold of 2019, which is an average introduction date in the park, to provide a baseline for community shifts. So let's look at some of the monitoring sites. It's not updating. Okay, so this is Lonely Bull on the Letaba, and all of the points represent the composition of communities, and what we see is the green are the pre-crayfish in the park, and the orange is post-crayfish in the park, and the takeaway message here is that there's still quite a lot of overlap between pre-crayfish and post-crayfish communities. And this is something that we see at multiple sites on the control rivers that I mentioned as well, so there's no significant shifts with non-crayfish related invariables. But now let's look at an invaded site. So we know crayfish are on Antallista, on the Sabi and Sand rivers, and this time we see quite a big separation between invaded and uninvaded communities. And this is a significant difference as well. And who's driving these shifts? Well, the short answer is there's lots of contributions by lots of taxa. So what we're seeing is sort of like quite a big change in the community composition. We're not gaining or losing specific taxa. And however, I did also find that elmid beetles and leptophlebid mayflies are significantly associated with uninvaded communities. I think this is because they're algae scrapers and the crayfish might be messing up that habitat for them. But if anyone has any thoughts on this, they would be gratefully received. And moving on to a second site on the Crocodile this time, again, we see this big shift between uninvaded and invaded communities. And this is also significant. And again, we found that lots of small contributions by lots of different taxa are sort of driving this. But we can also potentially use this in a predictive way as well, and that's something I'm working on now. So both of these sites are on invaded rivers, but we currently don't know if they have crayfish or not because they didn't come up on the 24 and 25 surveys that you've already heard about. But we can see that Sekirawani on the Sabi has sort of this big shift in 22, but that's not the same for Ninkongoma on the Crocodile River. So that might indicate that there aren't crayfish at Ninkongoma at the moment, but we'll see how that progresses. So my take-home messages is that Kruger National Park has a lot of really great macroinvertebrate richness, and the Sabi River is a particular hotspot for this. Long-term biomonitoring is also a really essential tool for looking at how aquatic communities respond to all stresses, not just invasive species. And I've provided some evidence of macroinvertebrate community shifts in response to crayfish invasion. But hope is not lost. Previous literature has also shown that macroinvertebrates can recover quite rapidly to other anthropogenic stresses like flooding and drought. So hopefully the same is true for invasive species as well. And next from me, I want to do a functional diversity assessment like you heard about from James to see what traits we're sort of losing maybe in response to crayfish and what consequences this could have for aquatic communities in terms of the ecosystem services that they provide. And we're also trying to implement the SAS protocol into an R package called Biomonitors, which will allow practitioners and scientists to create easily reproducible workflows for using their biomonitoring data. So a huge thank you to all the field teams and everyone who contributed to the long-term data set. And I was lucky enough to come to Kruger National Park and do some field work last year, and I had a fantastic time. So huge thank you for listening and your patience with the crayfish talk.
Thanks, Emma. We have some time for a question or two. Let's see this one behind you.
Thanks for your talk. I was just wondering if you incorporated geomorphic diversity, your river geomorphology, into your analyses to see if there was any effects of that. Because a stationary pool versus fast-flowing water is going to have a different
I'm really
diversity.
sorry, I can't hear you very well.
Your geomorphology, your river geomorphology,
Yes,
did
yeah.
you include that when you looked at your results? Because you're going to have different results with this pool versus fast-flowing water, for example.
Yeah, so obviously that can really affect the macroinvertebrate communities that we see if the rivers are quite different. But the paper I mentioned, the Satole et al. Paper, looked at the composition, because a part of the protocol for SAS is looking at the habitat composition at the different sites, and they found that there was no significant difference between them on all of the rivers, so that increased my confidence in using those as a control to like make comparisons between those rivers. And as you saw as well from the stresses that I put up on the screen, they're also experiencing quite similar like anthropogenic stresses, so that increased my confidence a little bit, yeah. But it's not perfect, but it was the best we could do as a sort of ecological experiment. Yeah.
Great, thanks.
Okay, thanks, Emma. Tertia?
Thanks, Emma. The great cloak crayfish, what type of habitat do they generally like? Because I know the systems in the south, so Sabi Sand, crocodile, has that overhanging vegetation.
Yeah.
But the Letaba, olifants, Levuvu doesn't really have that. They're very sandy rivers with no overhanging vegetation. Do you think that that habitat could support red cork crayfish in future? Obviously, we know in the south they were introduced from outside the park, but if that were to happen up north, would that type of environment support them?
Yeah, that's a really good point. So yeah, they definitely like sort of woody areas, kind of like slower flows would be better for them, and like pools and stuff like that. So yeah, the difference there might be why we haven't found crayfish on the Letaba and the Olifants yet, because the habitat is just a little bit more suitable there. But generally they are quite hardy creatures, so if they end up there, they'll adapt. And
Okay, thanks very much, Emma.
Thank you.
And thanks to the Red Claw Crayfish Group. I just want to draw attention to two posters that are outside. The one is by Molaseng Mushaban, and that's on the environmental and socio-economic impacts on invasive mammals. And the other one is by Riani Nivenes, which is on monitoring the polyphagous shuttle borer beetle. So those will be outside. And then I've just been asked to make a note in the session after tea. The talk by Corley will be moved to the plenary will be moved to the end of the session. And the talks by Sophia and Jonas will be moved to the start of the session. All right, thanks everyone.
Let's try this again. There we are. Good morning, everyone. So thank you for sticking with us for our first morning session. We are now going to do our group photo. So please do not disappear. We do want you to be in our group photo. We are doing things slightly differently this year. You will see on your name tag, you've got a colored dot. And so your colored dot basically represents the year you first started attending these meetings. So basically everyone that's got the same colored dot basically started roughly at the same time in attending these meetings. So what we would like you guys to do for the photo is we're going to ask everyone that's got a red colored dot to come up first. You're going to collect a hat from Sipakazi at the back there. You're going to bring yourself and your hat forward, and we are going to position you in a bit of a pattern. And then after that, we will be working through the other colored dots. So if I can ask, everyone with a red dot, please go and fetch your hat and come forward.
So, sorry everyone. So once you have your hats, can you please start moving to the front? Everyone at the back, everyone at the back. Once you've got your red hat, please move to the front.
Yes. Perfect. Yes, hello.
I'm worried about the picture quality from inside here, and lighting is so poor.
There's no. Have a look at the foyer. There's not enough space in the foyer.
Isn't there?
Yeah.
That's usually where we do it, or on the outside.
Do you think you rather want on the outside?
Rather on the outside. The pictures are going to be such poor quality from in here. You're only going to see people's red eye dots. But I'm also looking at the group, to redirect everybody. Let's try it. Let's see how it works. Because we're going to do the full group anyway afterwards. Let's do the colored.
We are not doing that. No, we are only doing 2 photos. We are only doing the full group, and we are doing. Yeah.
So the full group, can we do outside? Can we do the color codes here and then the full group?
Okay, we're not doing color codes.
Oh, okay.
We're doing the full group, and then we're only doing the first group. Okay.
Why?
Okay, guys. The slight change of plan simply because we are running out of space. And I also think the light is maybe not the best. So we're going to move this to the parking lot. Do not forget your groups. So basically what is going to happen, my red group, you are going to be our frame for everyone else, okay? Because you're the biggest group. So you are going to be the back and the sides. And then we are going to get the other groups to stand in color blocks in front of them. Okay, So, the red group, please make your way out to the parking area. The next group to go and collect the hats is if you've got a blue sticker on your, if you've got a blue sticker, go and collect your hats.
Thanks, everybody. Can everybody else just go to the back and grab the color hat that's on your sticker color on your ticket? If you can just grab your hat and then migrate to the parking lot where Shanae will help us put everybody else into their correct groups. Thank you.
Just here? Hello, hello. 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 Hello.
Sorry, a little bit of confusion there.
Okay, welcome back, everyone. Just a quick update. I've had a couple of questions from people about the hats. Firstly, yes, this is your hat. You keep it. Okay. And then people were asking, what do the various colors mean? And this was, you were assigned a color based on the first time you have attended a network meeting. So it's about when you started, not how many events you have attended. Okay, so everyone with a blue cap first came to the network meeting somewhere between 2003 and 2006. Okay, black caps, 2007 to 2010. Yellow caps, 2011 to 2014. White caps, 2015 to 2018. Green caps, well done. Green caps, 2019 to 2022. And the red caps, 2023 to this year. So just to give you an idea what those caps mean. Okay, enjoy.
Thanks, Sinead, and welcome back, everyone. We've got a nice session coming up with animal-plant interactions. To start us off, we've got Sophia talking about grass competition, functional traits under herbivore exclusion. Over to you.
Okay. Hi, everybody. Great. My slides? Great. Okay, hi everyone. My name is Sophia. I'm a PhD student at the University of Notre Dame. And today I'm going to be talking about how we can use plant functional traits to understand population level dynamics in response to simulated herbivore extinction. So around the globe, large herbivores are declining and in many places have become locally extinct. This has had cascading ecological consequences, including on ecosystem functioning and vegetation dynamics. At broad generalizable scales, we've seen increases in grass biomass and increases in woody cover. However, we still struggle to predict more fine-scale dynamics, including which species will increase and which species will decrease. Essentially, which species are the winners and which ones are losers in response to herbivore extinction. Plant functional traits, which describe the phenological, morphological, and physiological characteristics of individual plants, could provide a promising pathway forward to help us understand the underlying mechanisms and reasons why certain species respond the way they do. We know that plants respond to disturbances like herbivory by changing their functional traits. And we also know that different species have different traits, which means that they also should have variable responses in response to the same disturbance, which may scale up to population level changes. Using this framework, I developed two hypotheses for why we might be able to predict winning species. The first is that a species that increases in response to herbivore extinction could have high trait plasticity, where essentially, after a disturbance, it's able to quickly shift its traits into a new trait space that allows it to continue to persist and reproduce. The other option is that the species could have a broad trait space or high phenotypic variation, where it essentially increases the likelihood that any subset of the population could continue to persist and reproduce after the disturbance. I'm testing both of these hypotheses at Impala Research Center in Laikipia, Kenya, in the Kenya Long-Term Exposure Experiment, also known as CLE. CLE was established in 1995 and has been simulating local herbivore extinction for over 30 years now by using electric fences to fence out wildlife. We can then compare plant responses in plots where herbivores are present as a control to inside herbivore exclosures where herbivores are absent. So I'm working in those two plots across a replication of 3 blocks, although I would note that CLE has many other plot types as well. CLE has a handful of highly dominant grass species in the understory layer, and I chose 4 of these species for my study. I chose them for 2 reasons. The first is that these 4 species collectively make up 74% of the understory layer, so a very large proportion. And then the second is that they have interesting population trends that I'm about to talk about. All right, so before I show you the data, to orient yourselves, this is data from 1999 to 2013. And on the y-axis, we have the % difference in cover between inside and outside wildlife exclosures. So points above the dotted line means that it has a higher cover inside exclosures. Points below the dotted line means that species has a higher cover outside exclosures. So we have 2 species here that decline in response to herbivore extinction, Themata triandra and Lintonia nutans, although for simplicity's sake, the rest of the presentation, I'll call them decliners in pink and purple. We then also have 2 species that maintain population abundances inside wildlife exclosures. So they're able to persist within herbivore extinction. If anything, they do slightly better inside herbivore exclosures. I've labeled these as maintainers, and I'll refer to them as the blue and green species from now on. All right, so this makes it a great system to ask this kind of question because we have 4 highly dominant non-congeneric species with 2 different population trends that we can then analyze for their functional traits. In terms of the actual functional traits, I chose 5 traits that might be responsive to herbivore extinction. The first 2 are leaf to stem ratio and stem diameter. Both of these traits can help indicate how difficult it is for an herbivore to seek out and digest high-quality leaf material on a plant. Internode length can indicate growth as compensation for herbivory. Leaf strength as the mechanical force required to bite or tear a leaf. And then finally, crown depth, where deeper crowns may make it more difficult for herbivores to uproot a grass. So I collected these 5 traits on 60 plants per species per treatment for a total of 480 plants over July 2025. And I'd like to note that these traits have low correlation between them, meaning that they likely represent a spectrum of plant defense and competitive strategies that could be responsive to herbivory. All right, so onto the results. First, I'm going to show you the results for hypothesis 1, or trait plasticity. For this one, I was interested in how much, or the magnitude of change, each trait was able to shift, each species was able to shift its traits in response to herbivore extinction. All right, so this is our first table. Here, each cell or grid represents the % difference for that trait and species combo in response to herbivore extinction. The color represents the % change direction, and the size of the circle is the magnitude of response. And what we can see here is that our decliners actually show a higher magnitude change within their traits than our maintainers. We have one species, LN, that has a very strong response for one trait, crown depth. And then we also have another species, TT, that is fairly responsive across all of its traits, as compared to our maintainers on the left that show us much smaller magnitude of response across their traits. I then also wanted to look at all of these traits together. So this is a PCOA of all of the traits ordinated in space. And each box is a species, and they're colored by treatment. And what we can see here is that from the Permanova results, actually all 4 species were responsive to treatment by shifting their traits in some direction. However, we again see that our decliners on the right show a larger shift or larger relative distance between their centroids than our maintainers on the left. So this is a somewhat surprising result, and that our species that respond negatively to herbivore extinction actually showed higher trait plasticity and a larger magnitude of response with their traits, indicating that trait plasticity does not seem to sufficiently explain our observed population trends here at CLE. So let's move to hypothesis 2 then. So for hypothesis 2, I'm looking at trait space. For this one, I'm interested in phenotypic variation, and I'm going to use the dispersion of points as a proxy for that. So this is the same PCOA here, except for I've now faceted by treatment instead of species. And on the left, we have where herbivores are present, and on the right, we have where herbivores are excluded or absent. Again, what's interesting here is that our maintainers in blue and green actually show a larger dispersion of points, larger ellipses across both treatments than our decliners in orange and pink, which indicates that they have higher phenotypic variation and a broader trait space than our decliners, showing that hypothesis 2, or trait variation, seems to better explain our observed population trends than trait plasticity, or the ability to move those traits within space. The keen eye might have also noticed another interesting trend on this graph, and it's one that I did not expect with my hypotheses, which is that our decliners actually also collapse in trait space in the absence of herbivores. So on the left, we can see here where herbivores are present. All 4 species are able to maintain a different area in space, or essentially they each have their own unique combination of traits. However, when we exclude herbivores on the right, our orange and pink species, their centroids become much closer together, and the orange species almost completely envelops this pink one, which means that their traits are becoming more similar when we exclude herbivores. Comparatively, our maintainers in blue and green are again still able to maintain their own trait space and a separation in traits across both treatments, which means that niche overlap in trait space could also explain our observed trends, even though they're traits in defense response to herbivory as well. All right, so in conclusion, this means that our species that decline in the absence of herbivores show trait plasticity, but they're constrained by a smaller and more overlapping trait space, whereas our species that are able to maintain population levels have a broader non-overlapping trait space. And collectively, these results also indicate that we could use plant functional traits in the future to help us understand and predict which species respond positively versus negatively to disturbances like herbivory. I also want to mention this is the first phase of a multi-year project. I'm really excited because we actually just started the second phase last Wednesday. We took down some of the fences on these herbivore exclosures to allow herbivores back in after 30 years of extinction, simulating rewilding. And then I'm also setting up 3 new exclosure fences at the same time. And that way I'll be able to compare trait responses and species abundances to both of these disturbances simultaneously. And it'll also hopefully give me a more mechanistic understanding of how phenotypic variation can confer resistance and resilience to both herbivore extinction and herbivore rewilding. All right, a big thank you to the entire field team, my funding sources, and thank you all.
Thanks very much for a great talk. We do have a little bit of time for one question. I'm sure there's bound to be one. If not, I've got one. Okay. I was wondering if these plots had burnt at all during your time, your experiment, and whether you think the fire might have impacted some of those traits.
Yeah, that's a great question. CLE does burn. So they do biannual burns and annual burns in control plots, in plots within each of these herbivore exclusions, and then they also have controls. I am not working with the fire burns. I think it's super interesting, but it complicates things just a little bit further. Yeah.
Great, thank you. All right, next up we have Jonas Treppel talking to us about the role of herbivores and how they affect plant diversity.
All right. I think it's quite lucky that you're not the crowd that needs convincing that large animals in general are pretty awesome, which is to a large extent because they look really cool, but it's also because they're really impactful species. You all know that, right? They eat a lot of plants, they move seeds around, they redistribute nutrients, etc. And by doing that, we expect them to really shape ecosystems. And that makes them pretty fascinating. And following that, we've tried a couple of years ago now to identify sort of generalities in large herbivore impacts. So asking the question, when we have large herbivores or large animals in a system, will it react in a certain generalizable way? And to do that, we tried to find all available studies which evaluated herbivore impacts using some sort of experimental control impact design. And as you can see, these were actually quite a lot of studies. Of course, we've got our usual biases in there, but it's overall a decent spread, I would say. And we find overall almost 300 studies doing that, and could analyze responses across like 26 different ecosystem responses. And just to briefly explain this kind of plot, because I'm using it a little bit throughout the presentation, not this particular one, but this type of. It's like if you see these points right off the line, and not crossing this like zero line, it means herbivores increase the variable. If you see these points being left of the line, it means they decrease our response, and if they're like somehow crossing that line, it means they have no overall statistical significant effect. And what you can see here, there's a lot going on, and we do actually find quite a few responses where I say, okay, as we have large herbivores, the system reacts in a certain way. They reduce soil phosphorus, they reduce plant biomass, thereby opening up vegetation systems, they increase bare ground cover and all these things, and that makes very good sense. But one thing that really surprised us, or at least me, was like, if we look at plant diversity, nothing. It's just an overall neutral effect. And it was kind of unexpected. But what you also see is all these little points in the background, they represent the individual effect sizes. That means there's a huge spread in responses. Some respond really negative, some really positive, okay? And we were wondering a little bit what is going on there, why is that? And three sort of potential explanations that came to mind were this kind of data set represents a really mixed bag. So we've got like sites dominated by super small herbivores, sites dominated by elephants. We've got studies measuring, only grasses, studies measuring all plants, studies measuring very small plots, studies measuring very big plots. So we've got like, differences in herbivore regimes, differences in plants measured, and differences in spatial scale, all sort of lumped together, which may explain why there is so much going on. But we didn't know. So we tried to test it, and the best way to do that for us was to go into the field. Or specifically to go into the field to South Africa, the Waterberg region, which is up there. We tried to find 10 different reserves which are sort of close by and share similar-ish land use history, so being as similar as possible, except their herbivore communities. So we've got some in there that sort of have no large herbivores at all, some have like, some zebra and giraffe, and some have like some rhino on top, and then others have like all the big five in there, which should give us a nice gradient of like sort of herbivory pressure. In each of these reserves, we established then a couple of sites, and at each of these sites, we had a couple of plots, and at each of these plots, we measured, we assessed the plant community, we got their composition, we measured a bunch of rather basic functional traits, and we also sort of monitored these plots to get an idea of how much herbivory they actually experience. And that should allow us to then see, okay, how do plants at different scales, like at the plot scale, aggregated at the site scale, or at the reserve scale, respond to this like gradient of herbivory pressure. And if we do that, we find some interesting results actually, because we do see in this case, the more herbivores we do have in a given reserve, the higher our total plant species richness becomes. And it's like sort of this plot is faceted across scales, and you see the strongest effects really seem to emerge at the kind of landscape scale, but the trend overall is pretty positive. If we split it up into the different sort of rough plant functional types, we do see that this increase is really driven by herbaceous plants. Woody plants, they don't tend to respond very strongly in their species richness, and if anything, they're kind of negatively responding to herbivore presence and herbivory pressure itself. So it's really your grasses and your wildflowers that seem to like being eaten for whatever reason. We then were also interested in seeing whether the functional composition of our plants change. So we asked, okay, is functional diversity affected? It was not. But what was affected is the functional redundancy of these places. And functional redundancy essentially is just a metric for the functional overlap in a community. And that's important because that's thought to be one of the key indicators of ecosystem resilience, which means sort of as they have a greater overlap, it's easier for species to compensate for the loss of others. And I do think that makes conceptually kind of good sense. If you have a system which is exposed to more disturbance, it should also be better able to recover from from disturbance, right? And I'm putting another good picture of nice zebras up there to distract you a little bit while I'm telling you this is a cool exercise, but it's all correlational essentially. So we're just like comparing landscapes. We try to be as good as possible in standardizing, but there's so much going on in the background we can't really control for everything. And we are quite aware of that, so we decided to go back the next year and do a follow-up study, this time using actual, again, real experimental setups and try to populate the ends of the gradient. We tried to identify large and long-term exclosures, which exclude intact herbivore communities, essentially. And that brought us out of the Waterberg, and I think that's actually quite important to note, quite far away from the Waterberg, so from our original study site. And we find 6 sites along more or less South African border. 2 of them in Addo, 1 in Pangola Nature Reserve, and 3 of them here in Kruger. And we did a very similar sampling approach. We again identified plants, we measured some functional traits. But in this case, we thought while we were working with experiments, we should also test some more mechanistic hypotheses, trying to disentangle how do large herbivores actually affect these or plant diversity. And then we sort of tried to test 2 main hypotheses here. One is really they predominantly reduce the dominance or like dominant plant species, thereby opening up more niche space for competitively weaker individuals. Or they modify the vegetation structure in a way that more light can reach the ground, which again sort of allows weaker species to persist. And to do that, we also did measure some vegetation structure using terrestrial laser scanning. When it comes to results, I can tell you I was really surprised because we find exactly the same thing that we found in the Waterberg study before. It's again this increase in total plant species richness. It's really driven strongly by increases in graminoids and forbs. And your woody plants, they tend to respond not strongly, but if anything, rather negative. And I don't know, I mean, I'm early in my career, obviously, but I feel like this is too clean for ecology in a way. So this makes me kind of really happy. We also find that sort of initial support for both the mechanistic hypotheses, we find that large herbivores tend to decrease plant dominance, increasing evenness at the same time, and they also, probably not very surprisingly, open up the vegetation structure by decreasing vegetation density and height. When we then do a little bit of an exercise to try to link these 2 impacts together, sort of asking, okay, at the plots where they decrease dominance and where they open up vegetation, which of those is it that's actually driving the diversity response? We do find this seems to be predominantly related to reductions in dominance. So sort of where large herbivores reduce dominance, what they do frequently, that's where we observe the strongest increases in plant species richness. And then just quickly to the functional component of the thing again. In this case, we even find a slight increase in functional diversity, but we again find a very strong increase in functional redundancy. This is a bit confusing maybe because in this case the estimate is negative. That is our fault because we measured traits in a bit more of a continuous way, which allowed us to use a more fancy metric, which is the functional distance between species. But as your functional distance decreases, your functional redundancy and overlap increases. So this is again telling us the exact same thing, which I think is fascinating. And that is essentially what I'm here to tell you, right? Large herbivores tend to increase forb and plant grass diversity, mostly by reducing dominance. And this former landscape scale correlative study has kind of shown this seems to be really consistent across scales, scaling from sort of the plot up to the landscape scale. And with that, thanks so much to everyone who was in the field, who helped conceptualize. There was a huge team effort. And thanks so much for your attention. And if you have questions.
Thanks, Jonas, and thanks for keeping to time. We do have time for maybe 2 questions. We've got the usual suspects, Lawrence and Corley. Should we start with Lawrence?
Fascinating results. One of the things that we find here with similar drivers is the influence of soil nutrient status fundamentally alters the diversity patterns in forbs. Any thoughts on soil traits and soil nutrient status?
Well done, you find a pretty good weak spot. I think this is super important. I don't know, is that thing on? Yeah. I think that is super important, but we just have not measured it. There are these kind of dominant hypotheses that the more nutrient-rich your soil should be, the more resources are available, the more positive you would expect your response to be. But frankly, we just haven't tested that. But it would be super cool to do.
All right, Corley, time for a quick one.
Thanks, Jonas. That was very thought-provoking. I've been thinking for a while the way that we look at these effects of herbivores. We classically use exclosures. So the scale is really small. And we know that when you have large herbivores, they really increase heterogeneity. So I was just wondering, like, once you consider scale, like different scales, I think this is not so much maybe alpha diversity as something broader at the landscape scale. Have you considered it, and do you think it will make a difference?
Well, that's a little bit what we're trying to get in this first study, is that really bringing this like landscape scale or landscape level diversity in there. And this seems to be, and as we've seen, this was by far the strongest response, which you could explain by likely large herbivores are just using the landscape very differently, thereby creating like areas with different herbivore pressure, which allows very different plant communities to exist, which then in some are more than without herbivores. So that would be sort of my thought on that.
All right, we are out of time. Thanks very much, Jonas.
Thank you.
All right, just to note that we're now reverting back to the program as it would be. So for those online, we will follow from where we should be in terms of the program. First up, we've got one of two Evans. We've got Evan Foster talking about plant ranges in Kenyan savannas.
Great. Thanks so much. Hi, everyone. My name's Evan Foster. I'm a PhD candidate with Tyler Coverdale at the University of Notre Dame. And today I'm going to be sharing with you some really exciting research that I've been doing out at Impala Research Center in Kenya. So we can all appreciate that savannas are highly heterogeneous. And we know that both climate and herbivory are two of the main drivers of that heterogeneity, particularly at large continental scales. But we don't understand quite as well how they set individual species distributions at local scales of tens to hundreds of kilometers. And it's those individual species responses that ultimately are what emerge to shape that larger scale heterogeneity and drive species turnover across savanna landscapes. So my research focuses on understanding when and where climate and herbivory each shape local savanna plant distributions, with the aims of further understanding the dynamics shaping savanna plant communities at large. So imagine for any single savanna plant species the following distribution along a rainfall gradient, like what we have here at Kruger. Ecological theory currently predicts that the wet range limit of that species is set by biotic stress, like herbivory or plant-plant competition, while the dry range limit is set by abiotic stress, with the drier conditions acting as an environmental filter. But we know that herbivory plays a critical role in shaping almost every aspect of savanna plant communities, and that potential role is not captured in that environmental filtering perspective. So what I am proposing is that while abiotic stress alone may eventually set the dry range limit at a certain point of abiotic stress, the observed dry range limit that we actually see is due to the combination of both biotic and abiotic stress together. Now, to test this idea, I'm going to tell you about two representative plant species of Barlaria. Barlaria trispinosa with the white flower up at the top, and Barlaria spinosepala with the purple flower at the bottom. Both are physically defended woody subshrubs, and they are both found at Impala Research Center in central Kenya, which, like Kruger, has a steep north-south rainfall gradient, with the southern part of the property receiving roughly 20% more rainfall than the north, but only over roughly a 25-kilometer distance. And it's over that steep rainfall gradient that both of these species have local range limits, with trispinosa being found exclusively within the drier portion of that rainfall gradient, while spinosepala is found predominantly in the wetter portion of the rainfall gradient before reaching its dry range limit further to the north. So what is causing these local range limits? Well, I predict that the wet range limit for trispinosa is set by biotic stress, like herbivory, while the dry range limit of spinosepala is set by both biotic and abiotic stress. To test these predictions, I have utilized the Uhuru experiment, which is a suite of herbivore exclosures that exclude various different size classes of the herbivore community, and that is replicated across the rainfall gradient at Impala, where at both ends of the rainfall gradient, we have 4 different treatments: the open plots, which don't exclude any herbivores, the mega plots, which exclude only mega herbivores, the mesoplots, which exclude meso herbivores and larger, and the total exclusion plots, which exclude all herbivores larger than 5 kilograms. And it's within that experimental setup that I've conducted a reciprocal transplant experiment, where I have dug up wild individuals of both species and transplanted them into exclosures, both within their home ranges, so not changing their climatic conditions at all, and into herbivore exclosures outside of their home ranges, beyond their dry or wet range limits. And after 1.5 years of demographic surveys, we're already getting really key insights into the drivers for these dynamics, and that's what I'm going to be sharing with you today, particularly the survival data, where on the y-axis of these plots, it is the percent of my transplants that are alive, and on the x-axis, it is the number of days since the start of the experiment. So starting with Trispinosa, which as a reminder is the one found exclusively within the drier portion of the rainfall gradient, within its home range, we see very little mortality or evidence of transplant stress, which is good. That's what we would hope for. And then when we transplanted it beyond its wet range limit into wetter conditions, we again see very little differences in mortality or transplant stress. And if anything, survivorship was slightly higher in the wetter conditions than within its home range. Looking now at Spinocephala, which has its home range in the wet part of the rainfall gradient. Again, very little mortality within its home range, which is what we want. But when we transplanted it beyond its dry range limit, without excluding any herbivores, so the complete herbivore community has access to it, we see this dramatic drop in survival. 75% of my transplants within those treatments have died within the last 1.5 years. And one could reasonably look at this and say that this is evidence of an environmental filter. The conditions got drier and the plants started dying. But when we transplanted them into the total exclusion plots, there are no herbivores larger than 5 kilograms that have access to them. The mortality was roughly the same as it was within its home range. And it's the difference between these total exclusion plots that is the true climate-only effect. That's the only thing that's different between these two plots. Whereas the rest of the mortality that we're seeing is due to the interactive effect of climate and herbivory together. So I integrated this with a full suite of demographic data that I'm not going to share with you today due to time, but I used all of that with a population model to produce a population growth rate, or lambda, where a lambda greater than 1 indicates a growing, increasing population, while a lambda less than 1 indicates a shrinking population, which would then ultimately produce range limits. So for Trispinosa, these are the lambda values that we're seeing within its home range in those drier conditions. I want to just briefly note the sub-1 lambda values that we're seeing in the open plots are primarily due to an unusually high amount of elephant activity within the last 6 months specifically. They uprooted and trampled a lot of these specific plants within those plots. But we have long-term abundance data to show that that is more of an artifact of this pulse herbivory event rather than true long-term population declines. And I'm happy to chat more about this afterwards. Regardless, when we increase survivorship ever so slightly or not change it that much at all, the lambda values also do not change. This is mostly just to say that neither rainfall nor herbivory seem to explain the wet range limit for this given species. Looking now at Spinocephala, these are the lambda values within its home range in the wetter conditions. And then when we incorporate the increased mortality beyond its dry range limit in these drier conditions, we can see that the only treatment where it has a lambda value greater than 1 is in the total exclusion plots, thereby showing that it is the combination of both climate and herbivory that is setting that dry range limit. And I also just want to note the two treatments here that are the biggest differences between them is the mesoplot and the total plot. And the only difference experimentally in them is whether or not they exclude dictycs, which is one of the primary herbivores for this given species. So this isn't necessarily about total exclusion of the entire herbivore community, but about the specific herbivores that are eating any given plant species. So, in summary, I've told you that the wet range limit was not set by climate or herbivory, indicating that it is likely plant-plant competition or dispersal limitation, both of which I have ongoing data that I'm happy to talk about afterwards. I've also told you that the dry range limit is set by both climate and herbivory, not an environmental filter. And these are dynamics that are relevant to all savanna plant species because all savanna plant species somewhere have a local range limit along an abiotic and biotic stress gradient. Just the gradients for each species differ, and it might be over different spatial scales. But understanding what is setting these range boundaries is critically important to understanding savanna plant ecology at large. Thank you all so much.
Thanks, Evan. We have got time for a few questions. Yes, we've got one over there, Jason.
Thanks so much. Really fun talk and experiment. I was just wondering, did you include any competition effects in your models, and could the experiment capture that?
Yeah, thanks. Great question. So when we originally controlled for competition. When we transplanted them into the exclosures, we had to remove the surrounding vegetation in order to dig the hole to put them in. So during the first year or so, all the neighboring vegetation was removed. So we've controlled for that, but we've since let it grow back. So as the experiment continues onward, if we see shifts in mortality, especially in these total exclusion plots in the later years, we could expect that that's due to higher plant-plant competition rather than herbivory or climate.
Thank you. Any other questions? Yeah, behind you, Kira.
Thanks. That's a really great talk. Perhaps I missed it, but I was wondering which demographic stage you transplanted these plants. Are we talking about seedlings, in which case over time they're not going to be seedlings anymore, so the competition will happen not when they're seedlings? Or did you transplant adult plants? What demographic stage are you looking at these limits for?
Yeah, great question. I did not mention it during the talk, but we transplanted small individuals, mostly due to the physical constraints of digging it up with its root ball. So none of them were seedlings. They were probably sapling stage or maybe some adults that had really stunted growth. But it is mostly incorporating just the sapling stage. So none of this is including like from the seedling and onward. I hope that answers.
All right, it looks like there are no more. Thanks very much. Next up, we've got Evan Hockridge talking to us about open ecosystems in Congolese forest mosaics.
All right. Hello, everyone. Can you hear me? Yeah. I'm Evan, number 2. And today, so over the last couple of years at this conference, I've talked a lot about how biotic facilitation at very fine scales drives rainforest expansion at incredible rates into the savannas of the Congo Basin. This takes several forms, but most notably, in these thickets of trees that form away from the forest edge, we have plant-plant facilitation, where plants are helping their neighbors grow, which then confers an enormous advantage by making a fire barrier around them that prevents the spread of fire. But of course, these plants also have to get there somehow. And what we found is, I'll make this very simple here, is that across successional time, there is a change of disperser mechanisms from different long-distance dispersers that actually drive this process. And it's very species-specific, both in the plant and the disperser. And I want to pull your attention to one disperser that I'll talk more about today, and that's African forest elephants, which interestingly, unlike savanna elephants, we found no evidence that they are a major disturbance agent in these savannas. That's been found in other studies, but they are an incredibly important seed disperser that allows the succession to take place. But for those of you that are green hats or later in this room, you may recall I've also talked about other open ecosystems in the past, in particular a system called BIs, which are a very unique, they're only in the Congo Basin, very large forest clearing. And even though they can be extremely close to savannas, they are believed, without any quantitative evidence, that to be stable over extraordinarily long time periods. So you effectively have 2 open ecosystems with completely different fates. Also, like savannas, they are filled to the brim with large animals. And it's been very well understood that lots of animals visit these clearings to access salt resources in the soils. And we put out a study a couple years ago that finds, notice the split axis here, that by far the largest visitor is forest elephants. And that's important because when they visit these clearings and mine them for salts, they cause an enormous amount of disturbance, including on the edge, which we'll focus on today. So for a lot of reasons, people believe that these clearings are kept open by forest elephants, and that sort of is the primary mechanism for stability, but that again has never been quantified and never concluded, and there are very convincing alternative hypotheses. From some past work that we've done, we find that hydrology is a major associated feature with these clearings. They are oriented along river lines, they're associated with certain stream orders, certain topographies, and of course they are incredibly exotic sites, so they also could potentially be preventing trees from entering. And understanding what causes them to be stable is critically important. If it turns out a critically endangered species like forest elephant are necessary to maintain these clearings, that's very important for preserving this unique ecosystem. So we wanted to go and actually figure out which of these drivers make the most sense. And to do that, you have to measure change along a vegetative border that happens very slowly. So we used very high resolution light detection and ranging, or LiDAR, to extract the edge of the bay, to calculate lots of different typo-hydrologic models that we could use to look at hydrology and flooding. And then we resurveyed the sites 3 years later to get woody vegetation change. And for each of these little, sorry, it's not very clear, but each of these edge segments, each are 10 meters long, we can look at segment level change in both the positive, which means the bay is getting bigger, or negative displacement. With that, we also paired it up with lots of soil data, both nutrients and moisture, almost a decade now of long-term camera trap monitoring of these sites, and we were extraordinarily fortunate that for completely unrelated reasons, mostly about involving monitoring, the park that I work in, Adzala National Park, wanted to add salt to these bays to see if they could change animal visitation patterns and get a better understanding of how many animals are in the system, and they agreed to add salt to, we convinced them to add them to the bays that we were studying, and they added to half of the bays we were studying over time. So it gives us a really nice, the first ever landscape scale manipulation experiment in the Congo Basin. And to actually look at these different drivers, we took a causal framework approach where we look at a series of different variables with a piecewise structural equation model to get causal linkages, and then take those causal linkages and understand how that influences by edge displacement. Really important here is that each of these sort of nodes have tons of different variables, and I'm going to talk about which ones are actually important. But to go over these, we looked at how topography and hydrology drive systems, how soil nutrient composition drive systems, and then two different versions of elephant visitation. One we're calling chronic elephant visitation. Not all bays are visited by the same number of elephants, so that's like long-term, and then change over our study period, and as well as how the salt experiment potentially influences change, and as well as the bay edge itself. Now, the actual final model is complex, so we're going to go through this step by step. For those of you that are familiar with structural equation models, and for those of you that are not, the structural equation components are extremely well supported. This is like a pretty remarkable level of performance, and we're not missing any causal linkages, so we're quite confident that we've captured sort of the main variables. We'll start here with the chronic elephant component. Something interesting is we found no significant linkage between soil nutrient composition and chronic elephant visitation. That doesn't mean they're not visiting for nutrients. Bays have 3 times higher salt levels in them than outside of them. What it does mean is they're not selecting bays based on soil nutrient variability. Instead, they're basing it on mean soil moisture in the dry season. It's kind of crazy. When you walk around in a very, very wet bay, you'll notice gigantic crater-like holes, which are elephants sinking into the mud. So they're mostly selecting for different features based on access, and then ultimately that moisture is determined by topography. As far as the experiment, something that we were very excited about, the experimental treatment did have elevated visitation patterns of elephants, as well as several other species, which means that was working. And then we get to the top of this model, and it's very complex. I mean, we have non-significant alternative signs on the 2 elephant visitations. We have opposite signs, significant interactions, and we have these metrics. So instead of talking about it this way, I made a little cartoon here, the most mature way I could possibly do my science. So this is the bay, this is the forest, and these are our chronic elephants. And this right here is our salting site. Now, important is the salting treatment on its own is not significant, with no evidence that just adding salt to one place is going to change how the whole bay works. But we did find something very fascinating. Close to the salting site, the bay edge actually retreated inward, and it happens for about 150 meters. What's very important is this is dependent entirely on the chronic elephant visitation interaction, strongly indicating what's happening is elephant disturbance is being pulled off the edge. And instead, they're accessing the salt that's like buried about 1 meter deep and is very easy for them to access. And what further supports this evidence is after 150 meters, what happens, what you'd expect is exactly what happens. More elephants mean more bay edge displacement. These results together lead to something very interesting. First, it does show us that probably what's happening at the edge is that elephants are trying to mine for salt along the roots of the trees, which has been anecdotally noted in other studies. But also that elephants are responsible for stability of the bi-system in both directions. A loss of disturbance leads to retreat, and an increase in disturbance leads to expansion. And this was further supported by the interaction with new elephants, which is probably just the same elephants revisiting more frequently. They do have an interaction with distance. They tend to revisit this spot, and their sign is the opposite. They counteract this force. So more elephant visitation over time leads to more edge retreat. Interestingly, the two topographic variables are probably both related to elephants as well. Forest elephants really don't like slopes. That's very well understood. So an edge that has like a steep rise directly on the edge of the bi has less elephant disturbance, and more roughness equals more expansion. And those rough areas are areas that are extraordinarily highly mined. Now, if we put this in the context of what we understood from our previous study about savannas, where forest elephants disperse the seeds of very specific species, which then prevent the spread of fire, and that converts the system towards these later successional stages. And considering that actually the edge species in both systems are very similar, it's really fascinating that a single nutrient, really essentially sodium in the case of our experiment, completely flips the sign on these relationships, halting succession, showing how keystone interactions can actually drastically change how megafauna interact with their ecosystems. I don't want to take too much time here, so I'll do these takeaways very quickly. First of all, elephants, we found, are the primary determinant of bi-edge dynamics. It is localized, modulated by salt effects. We have some other findings that show this, and I'm happy to talk about them if people are interested. And our evidence suggests that a loss of elephants would likely mean loss of bis, which is a big problem considering the ecosystems of bis are extremely distinct from other open ecosystems in Congo and represent just 1% of the habitat in the park we work in. Really quick, for those of you that are interested in more of the herbaceous realm, I didn't have time to put that all in this talk, but we are also studying that and how these salting experiments and how these bis change over time on the inside. So chat with me if you're interested. So yeah, thank you very much.
Thanks very much for a very fascinating talk. I'm sure there are going to be questions. Yes, Coley, we got one.
I thought there for a moment we're going to have to call the bouncer to throw you out. I thought you were going to say elephants don't matter.
No, they matter.
So I really enjoyed your conclusions. But I was just wondering, so what we see, what we see from just patterns, veg patterns within Kruger, the moment you have a bit of a flay waterlogged, seasonally waterlogged system, trees can survive there if you don't have herbivory. But the moment you throw elephants into the mix, then you lose trees. So I was wondering, like, if you've done any herbivore exclusions, and what did you see in those cases?
Yeah, that's a very good question, and that's sort of why this system, with these alternative hypotheses, was so interesting. Because if you ever get a chance to see a bi, you could quite literally be in a forest, and you take one step forward, and then the vegetation is centimeters tall, which looks sort of like a Martian system. So that's where these ideas come from. And that's what's nice about this study is I think it shows just how important the elephant component is as a disturbance. As far as exclosures, we have had conversations. I think we've actually at the conference last year, we were starting conversations about putting exclosures up. They're not an easy system to put them up in. You can't electrify them, definitely not in this system. And it's also, not all of them and not everywhere, but they're highly, in the wet season, very likely to flood. So there is complexity there, but I think that's something that's on our radar to do, certainly.
Thanks. We've got another question from Tertia.
Thanks, Evan. I'm just wondering to myself why I haven't asked this all the other years you've spoken about bis. But where exactly does the water come from? Is it underground, deep, warm groundwater? You're nodding.
That's a very good question.
Okay.
It's both groundwater and surface flow, and it depends on the season. Some bis quite literally have springs in them, and they're kind of fun. You can put your leg in and your whole things, and it's like you can't hit the bottom, so they're very, very deep. And during the wet season, it's all surface runoff. So we have some other work on like if these sort of flooding effects and congregation effects potentially bring in all the nutrients from external places. But the hydrology of these systems is very complex, and we're the only people, as far as I know, to study it in any capacity. So it's certainly under-researched.
Okay. Yeah, because I'm just wondering if it's deep groundwater, the temperature of that water is also very different. So I wonder what influence, not just nutrients, but also the temperature of the water has on the vegetation. Thank you.
Very good question.
All right, thanks. We are out of time. So anyone else have a question?
There's another question.
Oh, okay, quick one.
Sorry, this is kind of combining your previous work on savanna thickening with this work. Could you put your salt blocks into your savannas near your trees to try and knock those back? Would that work?
I think that'd be very interesting. Ultimately, we didn't get to pick the salting sites. It was under this like other department in the park. It was more like we helped them figure out where to put the sites, which happened to be really convenient for us. With that being said, the park has added, there's one research house in the entire park, and they have added a salt treatment in front of the house. I have no idea how that's going, and it is literally an N of 1. So, how useful that is hard to say, but if there's something interesting there, I think it wouldn't be hard to convince them to do that. But that would be fascinating if that would switch the interaction effect with elephants. That would be, very cool.
All right, thanks very much, Evan. Next up, a couple of blue hats for sure. Starting with Mike, talking about the impact of elephants in the neighboring reserves adjacent to Kruger, where fences have been dropped. A very contentious subject. Looking forward to hearing what you have to say.
Thanks for that introduction, Ben. Yeah, I might need that. Oh, mine's here. Good. Lekker. Yeah. Are we jumping the gun here already? Thanks, Ben, and good morning, everybody. It's a privilege to be standing up here in front of you all. This morning I'm going to be speaking about dropping fences, linking changing elephant densities to trends in woody biomass in private reserves within the Greater Kruger National Park. And Kathy, you spoke about collaboration and co-learning, so I'd like to acknowledge my co-authors here, and particularly the co-learning part. I learned a hang of a lot, and I've still got a hell of a lot to learn about some of this remote sensing. So in particular, Conrad Wessels and Isak Smit. Yeah, so the question of elephants is an emotional one, as you said. But we find that there's a lack of empirical evidence linking density changes to vegetation structural changes, to specific magnitudes of vegetation structure change across extensive conservation areas. So the sequence of fence removal between the private reserves and the Kruger Park has essentially created a macro experiment as elephant access and density changed along known timelines. So just to give you an idea of the study area, if you look at this area that's the Kruger Park there, and this area to the west is all the private nature reserves, and that's a large part of my study area. And just to illustrate that the yellow there, the fences were removed from Kruger, between Kruger and them in about 1994. The Mdhluli fence came down in about 2003. Then we have got two private protected areas here and here that are still fenced, quite large. And then we have two unfenced fenced areas with no elephants in these two. And then these two in red here are the fences came down in 2017 and 2018. So there's quite a nice difference in the sequence of fence removal. And then if you look at all of these little red dots here, those are our study sites. We've got about 500 monitoring plots that we do every year, and that dates back to 1989. That's why I have a blue hat and a lot of gray hair. So that's our study area. Obviously, we're just going to be looking at the data from the last 7 years or so. We'll go straight into the results. We looked at elephant density, that's the green bars. We looked at field canopy cover, that's measured in field on the ground, those are the red bars. And then we looked at SCANSAR-derived woody cover and above ground biomass density, those are the two blue there and the sort of reddish ones there. And as you can see, where we look at where we have no elephants, these two properties, fairly stable, canopy declining and increasing a little bit, just fluctuating with environmental conditions. But overall, almost a bit of an increase there, maybe speaking to increased carbon dioxide and increased tree densities in these areas. And then we have the two fenced areas, protected areas, quite low elephant densities. They're sitting at about 0.5 elephant per square kilometer. And the pattern there, you can see fairly stable, maybe a little bit of a downward trend in the field canopy measurements and in the two SAR data measurements as that elephant density increases. When we go to the area that's been open to Kruger since about 1994, you can see higher elephant densities, green bars, and varying degrees of canopy decline depending on the inclination of the elephant densities. And then we go to the two really interesting ones here, where we had two fenced areas with very low elephant densities. And then the fences came down here between this protected area and these ones, and you had this massive increase to around about 5, just under 6 elephants per square kilometer. And as you can see with our field measured data, you can see it's quite a steep drop in canopy cover, and the two SAR remote sensed results show exactly the same trends. The same here for this reserve, where the fences actually fell into disrepair, and there was an influx of elephants into the area, but the fences were put up with all the elephants still inside. So there's been a steady increase in that, and it's quite an interesting study site there, and you can see the trends of both the remote sense data and the in-field measured data. Okay, so if you look, these are the areas I've just spoken to, but if you look at probably the best is to just look at the maps and the different shades of red, where and this is the change. This is the change in above ground biomass density in the last 7 years, to 2016 to 2023. So you can see in the fenced areas with no elephant, pretty green still. If you look at the areas that have been open for quite a long time, there's not a major change in the coloration or the change in tree density in the last 7 years. The impact has happened and it's fairly stable, although still declining. But then the really interesting ones are where you go into this area here. The fence was removed in 2019. It's the southern Mdhluli. It's hectic, big changes. This area as well, that very red area there was also just the fence was taken down more recently. And then that's the Olifants River there, which has got a lot of elephants along it now. And then if you look at this area here, Thorny Bush and Sandringham, those two very steep inclines over here, that is a major decline in canopy cover. So there you have it there, the field measurements, fairly stable, and then declining drastically, and speaking to the two satellite image data sets, and Thorny Bush over here as well, where you've got this sudden increase of elephants up to just under, as I say, just under 6 elephants per square kilometer, and your canopy declines, and that speaks as well to those two cover and density measures derived from the satellite imagery. Yeah, so there's the picture again of these two sites, and there's the actual the regression of increasing elephant density versus canopy cover and density. So elephant densities above 5 elephants per square kilometer led to AGBD losses of greater than 800 megagrams per square kilometer per annum. That's pretty significant. And the AGBD loss rate is about 105 to 220 megagrams per elephant per square kilometer per year. And it's a fairly novel calculation that's been done here. So it's very interesting what we're finding. And then elephant density versus above ground biomass density over all the reserves. There's a clear negative relationship between AGBD and elephants at a density of greater than 3 per square kilometer, as you can see there. But at densities less than 3 elephants per square kilometer, there's no clear relationship, and that's because there's a lot of interannual variability across diverse reserves. So It's not a one size fits all here. There's a lot happening in this area. But when we look at our field data, it shows that a relationship between elephants at less than 3 elephants per square kilometer and canopy cover. And there we can see it, where we look at field woody cover trends in relation to elephant density in the Sabi sands. And we are here at around about 1 to 1.5 elephants per square kilometer. We're starting to pick up quite significant canopy declines. And then a long-term conceptual model. If you look at the phase 1 here, where you've had long-term exclusion, you get up to 48 megagrams per hectare AGBD. And then the second part is where you have new access to areas by elephant, and you have the steep increase in elephant densities, and you have at the same time a sharp reduction in AGBD, and that happens pretty quickly in just 4 years in this instance. And then as the elephants decline, because they sometimes go into an area, as we know, and then they move out again, as the elephants decline, this AGBD, the rate of decrease decreases, so it slows down. But as elephants start going up again, we pick up that above ground biomass density declines again. Yeah, so management implications and trade-offs. There's obviously cons. Dropping fences after a long period of time can lead to 50 to 60% loss of woody vegetation and above ground carbon stocks. And this could undermine the natural climate solution projects in conservation areas aimed at conserving above ground carbon. So it's something to think about. And we're not advocating planting trees, but we're looking at some sort of balance between your woody and your grass layer. And then the loss of iconic big trees. It could be that, do the loss of them have a disproportional effect and influence on biodiversity? And if you look at the grasses, the productive, nutritious, palatable grasses that grow under these trees, with the loss of them, is there a time frame at which we'll start moving towards a more heat tolerant, smaller leaf grass, and the implications of that for grazing? And then there's also the impact on sort of tree nesting, hole nesting birds. And also, if we have these increases in temperature, which seem to be happening, I think they said 1.5 degrees centigrade for every calendar month in 2024. There are obviously thermoregulation issues in terms of birds and that sort of thing. Also, if you drive in the park here, you don't see it as much, but in some of the private areas that I showed there's a massive decline in big trees, and you can start wondering what's happened to the leopard. And I think there's probably anecdotal evidence, but of these animals not being able to hoist their kills into trees and under pressure from other predators such as lions. The pros, it's not all bad news. They certainly have reduced the shrub encroachment, and they've really opened up areas in these adjacent private nature reserves. And that's really good for improving visibility and game viewing, which is a primary objective in these areas. So in certain instances, it also opened up the area to allow the grasses to compete more vigorously, which is a good thing. The point is, at what point do we reach this where we start losing those good grasses again, and due to the loss of these iconic trees? Yeah, and then just to finish off, I think it's a very emotional subject, and Sam, we've had a few chats about that. And the management around that it's not easy and it's not simple. But also we subscribe to the same philosophies, the same conservation philosophies in the park and adjacent to the park, but our objectives are often very different. And our scale at which we function is very different next door because of there's excess water. So the distribution of elephants is pretty universal, as opposed to here where they move around a lot more. So yeah, from my side, I just say, I think when we start looking at management, we must do the right thing, the right place, at the right time, using the best evidence. Thank you.
Thanks, Mike. Unfortunately, you've used up all your time. I'm sure you would have had lots of questions, which I have for you. I'll catch you at lunch. All right, moving on to another definite blue hat, Tony Swimmer, talking about elephants and floods on riparian trees on the olifants.
Thanks. So when I uploaded my talk at about 3 a.m. this morning, afterwards I realized I'm giving a talk on elephants and trees, and I only have one photo of an elephant in the whole talk. But then I remembered this lovely shirt my wife had given me for Christmas, so I'm covered. Okay, so before I go on, let me just acknowledge Transfrontier Africa, the guys who manage 2 private reserves, Olifants West Nature Reserve and Bush River Lodge, have really been helpful in the study and made it possible. Craig Spencer in the early days and later Paul Allen, and many of their students and assistants who have helped collect data, and also many SAON staff who have helped collect data over the years, too many to mention. Okay, so elephants and trees are an issue. You might have picked that up from Mike's talk. Riparian trees are of particular concern because riparian trees are generally bigger and they often support a whole bunch of species that you just don't get in the tall trees in the terrestrial areas, and so they can make a really disproportionate contribution to biodiversity at this sort of landscape scale. And riparian trees and forests are in decline around the world, and in many sites in Africa and South Africa, and many of those have elephants. So what's the role of elephants in the decline of these massive trees relative to other things there that can eliminate them, such as floods and droughts? So to disentangle the effects of elephants from floods, you ideally want 2 stretches of river right next to each other that are the same in all respects, except one has elephants and one doesn't. And the western edge of Kruger National Park is a nice place for that, except there really aren't many sites that are suitable, because as soon as you go outside the park with no elephants, there's a lot of agricultural impacts. But on the western edge of Olifants West Nature Reserve, that's the boundary there, we have the Olifants River, where elephants have been back since 2003. And then just upstream, we have private conservation areas with no elephants, but all the other herbivores. So it's a nice place to study this. And after floods in 2012, we started monitoring some of the trees along the Olifants. This is pretty simple field work, geotagging trees with a GPS, measuring heights with a clinometer, tape measure for stem diameters, and we've just done that at regular intervals for the past 5 years. So here is the site, Bush River, with no elephants, and you can see a fairly well-developed riparian area. All the little triangles there are geotagged trees that are included in the study. If you go back in time, so this is now 2010, that's 2024. This is 2010, before the 2012 floods and the more recent flood. And you can see there's still quite a lot of trees there. In fact, it even looks like there's been possibly an increase in riparian trees over that time. Then across the fence line to Olifants West, and here's one of our sites there. Again, the triangles are showing our geotagged trees, and it's just more sparse. Density of trees much less. And if we go back in time, you see actually quite a change. There were a lot more riparian trees. There was a whole island full of tall trees, mostly matumis. There's a big strip of trees right on the riverbank there in 2010. And these are all gone by 2024. So the field data just reflects the same thing. The density of adult trees, about half in Olifants West compared to Bush River. Adult trees here being trees that are taller than 10 meters, and species that can grow into really big trees. Interestingly, the trees are taller where the elephants are in Olifants West, but I don't think this reflects more favorable habitat or faster growth rates or anything. It's simply that the elephants have kind of eliminated all the shorter adult trees, and it's only the really big ones that are kind of elephant-proof that are left, whereas there's a lot of emerging juvenile trees coming through and reducing that mean height at Bush River. If you look at the population structure of juvenile trees, you see a big difference as well. So that over there is the graph I just previously shown. That's the adult tree densities. The juvenile trees, 5 to 10 meters, a lot more at Bush River where there's no elephants, and not many at Olifants West. But then interestingly, the smaller trees, less than 5 meters, actually more at Olifants West. So a lot of short little trees. And these are generally sort of medium trees that have been made short by repeated elephant impacts. Okay, so that's the current situation, the difference between the two. There's also a lot of differences in species composition that I'm not going to go into for the sake of time, but some really big differences in the dominant species between those two communities. So we had a survey in 2012 and then a couple of more recent ones. And on Olifants West, there's a clear decline in the abundance of these adult trees. It's not very rapid. Works out to about 1% per year. But at Bush River, almost negligible decline, pretty flat. So there is this gradual attrition of trees. But is that because of elephants? And actually, no, from our data, it seems not. Most of the trees that have been lost over this time period were growing right on the riverbank and appear to have been washed away by floods. There are some lost to elephants. That's an example of marilla. So the more sort of palatable species are getting knocked out. But that didn't actually make up a huge amount of the losses. All right, so elephants having some role there, and certainly amongst the smaller trees. But what about floods? And we're quite fortunate to have a major flooding event very recently. So that's what the Olifants River in this part of the world looks like in the dry season, pretty low. You see just a band or a stripe of riparian trees there. By this December of this summer was already looking like a flood year. The river was really quite high. This is almost flooding conditions, higher than it normally gets in the summer. And then in January, there was about 600 mils, more than 600 mils of rain in 7 days. So this just got added to, and it just got to really crazy flooding conditions. And from anecdotal evidence, this is the highest the river's ever been in 50 years. So what did that do to the trees? Well, in Olifants West, it had a massive impact. We had about a 10% loss from that flooding event. And mostly really big trees growing on the riverbank, close to the river. Bush River lost some trees, but literally a handful, so much less impact. What was the cause of that major difference? Is it elephants or is it something else? So if you look at Olifants West, here's another site more closely. You maybe can't see, but these little dots up here are trees that have died during the course of the study. And basically almost all the trees along the macro channel, along the riverbank are now gone. And there's this massive floodplain sort of grazing lawn area that looks like that. And then only at the outer edge of the riparian do we have any riparian trees left. Incidentally, the blue line there is the flood line we mapped in 2012 after the 2012 flood, and the red one after the more recent flood. And very similar, the red is slightly further out. So really this whole sort of potential riparian forest, which by all accounts it used to be, is now more a grassy floodplain with just a strip of riparian trees at the one edge. So what could explain that? So one caveat maybe is that both sites didn't experience the same intensity of flooding. There is a fairly large tributary that comes in here downstream of Bush River but upstream of our Olifants West sites. It's not a big river, it's seasonal, but with a massive rainfall in the area that might have created a surge, a pulse that caused more tree losses over here than over there. I don't really think that's the cause because in Bush River, the floodline was at least as high. Almost all the riparian trees were underwater for a while. We could see from debris heights on the trees, sometimes up to 4 or 5 meters high. And that was the same at both sites. So the water got really high at Bush River as well. And there were a lot of trees right on the riverbank, completely submerged, and almost no bank erosion, almost no tree loss. So what else? It seems likely that just the density of vegetation at Bush River was enough to cause drag and eddies that would reduce the erosive force of the water. There's been quite a bit of research on this that shows that it does actually make a difference. So maybe just that density of vegetation was enough to save those trees, whereas at Olifants West, it's kind of hit a threshold from earlier floods where some trees have been lost and shrubs, and now everything else is just really vulnerable. That's not entirely satisfying either, because why did that happen in the first place? Already trees were being lost in large numbers in 1996, 2000, and that was before elephants were in the area. So it can't be that elephants had, caused some vegetation change that then made the trees more vulnerable. So definitely something that needs further study. I think we need to look closely at the sort of topography and the flow dynamics of these various sites. Okay, so just to wrap up quickly, so yes, elephants have definitely altered the riparian tree community, but changed species competition, changed structure, but certainly not the only cause for the loss of massive riparian trees. The direct impacts of floods has been way bigger than elephants, but perhaps there is some sort of elephant-flood interaction going on, but that needs further study. Thanks.
Thank you, Tony. You kept well to your time as well. So we do have time for a question or two. Anyone? Tyler, is that a hand up? No? I was wondering if roots, like where the trees are denser at the bush site, would that also maybe have contribute to holding the soil less erosion?
Yeah, I mean, it could be, definitely. But, you know, at some point in the past, the root density was presumably the same in Oliphant's West. So, and then trees started to go. So maybe it's depth of sediment or something. Yeah.
No. All right. Yes, Sam, we do have a question there.
Thanks, Tony, and also thanks for Mike earlier. Tony, I just wonder, how the hell did we get to riparian forest in the first place?
Yeah, I wonder about that too. If there were, you know, in history, if there were a lot of elephants in the landscape, how the hell did these riparian trees actually establish in the first place? Yeah. So, I don't know. Maybe it's a satiation thing, that there were just massive recruitment events with thousands of individuals, and some of them eventually got through and got to elephant-proof size. But
people were there, maybe?
People. But how long have people been there? Yeah. Okay.
Anyone else? Looks like we're all good. Thanks, Tony. Then moving away from elephants totally. Moving on to Emily Weedle, talking about how do savanna trees cope with browsers and droughts? Thanks.
Okay. Great. Good morning, everyone. Thanks, Tony, for adjusting the height. I'm not quite as tall as you are. But Tyler and I have been thinking a lot about how both herbivores and drought interactively shape vegetation in savannas. And so the project I have for you today is work from my postdoc. That's kind of our first bite at answering that question through a physiology lens. And we've talked a lot about exclosures this morning, and a lot of what we know about how herbivores affect savanna vegetation has come from these various herbivore exclusion studies across Africa. And we very consistently see that when you get rid of the animals, you get massive increases in tree cover and grass biomass. But we don't really quite understand what the consequences are of these increases in abundance, and we don't really even have a good handle on what's driving vegetation dynamics inside of these exclosures. And so these plants that were once experiencing a very heavy top-down pressure are now more or less exclusively experiencing bottom-up control if we're ignoring fire. And they're also living in a much denser and presumably more competitive environment. And so the outcomes of these exclosures on plant performance are going to depend on resource availability. In this case, we're thinking about rainfall, as well as species physiological strategies, which is going to determine how sensitive any given species might be to these increases in competition. And so we were interested in these ideas and had two primary research questions on, is there evidence for increased competitive stress inside of exclosures, like water stress? And then are some dominant tree species more sensitive to increased competition than others? And we approached these questions using the Uhuru exclusion experiment in central Kenya. So this is the same system that Evan has already introduced earlier this morning. But just as a reminder, there's exclosures that span a north-south rainfall gradient. So you can see pictures of the open and exclosure plots there. And this exclusion has massive effects on tree cover. So the wetter site tends to have a little higher tree cover than the drier site, but when you exclude herbivores, you get a similar increase in the magnitude of tree cover, and it ultimately reaches around 40 to 50%. And we focused our analyses on the 3 dominant acacia species at this site. They are estimated to cover about 80% of the tree cover here, so they're really highly abundant, but they vary a lot in their morphology in physical defense investment. So Acacia abaca has both long and short spines and is heavily defended. Mellifera has these short hook spines, and then Acacia brevispica has both short prickles and very large leaves. So it's poorly defended. If you were to go out here and stomp around, I'm sure you would think to yourself that this is a very pathetic looking tree. Which is why it's my favorite, because it's also so highly abundant. So it's really tolerant of herbivory. So over the past 20 years or so, there have been permanently tagged individuals of each one of these species within these exclosure plots that span the rainfall gradient in order to assess changes in size. So in all of these figures, the drier site is represented in brown, the blue site represents the wetter site, and then here the dashed and solid lines represent the exclosure treatments. And very unsurprisingly, when you exclude herbivores, the plants get bigger, but there are some interesting site level and species level differences here. The trees within the drier site are reaching larger size classes than the trees in the wetter site, which potentially could be due to lower tree density at the drier site or lower herbaceous biomass. And then we're also seeing that this Acacia abaca species that's very heavily defended is the only one that's actually increasing in size within the presence of herbivores. And you can compare that to Acacia brevispica, which is that poorly defended species where the average size of these tagged individuals is just decreasing over time. So there's some species level differences here that affect their long-term growth trajectories and then may also affect how well they do in the absence of herbivores or how sensitive they are to those increases in competition. So to get at some of the plant stress within these exclosures, we measured both water potential and gas exchange. And water potential is it's an indicator of tension in the xylem. So a lower water potential value means that a plant has more hydraulic stress or more water stress. And these pre-dawn water potential values are taken before the sun rises. So it's before the plant has woken up for the day and has started its gas exchange. And so it's often interpreted as an indicator of soil water availability. So what we see very consistently here is that in the exclosures, these plants have lower pre-dawn water availability, and this occurs most when the system is dry. And so these data would indicate that the soils are drier inside of exclosures and that these exclosure sites tend to be drying down faster than the control plots. And we saw very similar trends with the midday water potentials, which is the same measurement, but it's taken when the plant has woken up and is at its peak activity for the day. And inside of these exclosure plots, we even find where some of the plants are so stressed out that the leaves are basically just shutting down. And then we find that pathetic species that has poor defenses is the most sensitive to this change in water potential. So it didn't really matter if we measured it recently after a rainfall event or after the system had dried down. It always showed lower water potential or higher water stress within these exclosures. We also measured gas exchange. So here we have photosynthetic rate across the top and stomatal conductance on the second row there. And we see these patterns are mimicked here too. So very consistently, gas exchange was lower inside of the exclosures, probably reflecting greater water stress. And then once again, that Acacia brevispica species was the most sensitive to it. So these physiology variables are aligning with what we might think could be happening if competition is affecting their physiology inside of these exclosures. And so I want to wrap up by scaling up a little bit more to the population and community levels, where this is data from a separate herbivore exclusion experiment at Impala called the Glade experiment. And that species that was very sensitive physiologically to exclosures is also showing to be sensitive at like these higher individual and population levels. So this is mean canopy growth for the first 2 years of the experiment. It's heavily targeted by herbivores. When you put the fences up, the tree grows very rapidly. But after about 20 years or so, there's actually negative growth rates inside of the exclosures. And this is also kind of mimicked with the relative abundance, so that same species, Acacia brevispica, would be the blue in this, and that solid line is the exclosure experiment. So it's decreased in abundance by about 15% over the course of about 20 years. So just to wrap up, I've shown you that these net effects of defaunation on tree growth are often very positive. We see increases in tree growth and tree cover, but there's also some physiological consequences that might be happening with increases in competitive stress, particularly when it's dry, and that the magnitude of these effects vary based off of species physiology. So ultimately, some of these indirect effects of increases in plant-plant competition with herbivore exclusion could drive these changes in community composition and ultimately broader ecosystem processes like carbon and water cycling that we're interested in as well. So I will say thank you to you guys for listening, thanks to our field assistants and the curators of the long-term data, and happy to take any questions.
Thanks, Emily, for a very interesting talk. We do have some time for questions. Corley, you got one? Or are you just waving your pen? You do? Okay, it's coming there.
This is the veg session, so I have to have some questions. But this is just a comment. And it's so nice to see your results, because this is also what we are seeing at our Mdhluli exclosures. And where the encouraged species, Dicrostachys, thickened up so much. And then we had a drought, and a lot of those trees died back. And then we had a massive runaway fire that burned our exclosure, and it just managed to open up again, the exclosure. So you'd think once trees are protected from herbivory, they're fine, but there are these other processes, which it is also nice to see that it's reflected in your results.
Thanks.
That's just a comment, yeah, not a question.
A question from me is, did you guys look at whether you think it's the grass or the trees that are competing for that water?
Yeah, that's a good question. I think the species that's most sensitive, I guess anecdotally, it seems like it's a poor competitor both below ground and above ground. But doing some digging up and getting below ground and looking at the roots, I think it's just a very shallow rooted species. And it's likely that herbaceous tree competition it just can't handle.
Great. Any other? We do have time for one more if there is. Tony, yeah?
That species that was increasing outside with herbivory, what's your sort of theory on that? Is that just a cohort or something that's growing up, or why would that be growing so much?
The species that was doing well outside of the exclosures? Yeah, well, I think it's a poor competitive inside of the exclosures. And then also that it's a disturbance effect of these large herbivores that kind of decrease that competition, probably with both herbaceous and trees, and allow it to do really well. And it's just really, really tolerant of herbivory, so it handles it really well.
Great, thanks, Emily. Next up, we have Lisa LaRue, probably also a blue hat, I would guess, but yep. And she's going to talk to us about dung nutrient dynamics.
All right. Hi. So I'm actually going to tell you a story about, it's a continuation of something I spoke about a little bit before, maybe 5 years ago at this meeting. And it's the outcome, or at least preliminary results on the outcome of a scientific disagreement between me and Judith Sitters and Harry Aldeventering some 5 years ago. That we eventually ended up teaming up with this whole group of people, many of whom are in the audience, to get a bunch of data and try and sort this out. And this is what the story I want to show you. Oh, maybe I'll also just say that this was led by Ritwan Jafar, one of our students, who then just, collated all of this data and started making sense of it. So all the heavy lifting is Ritwan's. So the core concept here is ecological stoichiometry. So it's the study of how different elemental ratios drive ecological interactions and ecosystem processes. And the basic idea comes down to the fact that organisms or animal organisms differ quite starkly often from their food source. So there's a mismatch in the elemental composition of plants and herbivores. So plants tend to have quite a bit more carbon than the animal herbivores. And so there's this process of selective retention of the elements that the animal would need and selective release of the elements that they don't need. And so there's a restructuring of this composition of elements from what goes in and what goes out. And this is where you think that animals should be able to shape these nutrient landscapes. And for our purposes, where we are interested in the impact of body size in large mammals. So what the ecological stoichiometry predicts is that as you have changes in body size, your requirement for nitrogen and phosphorus changes, and particularly the requirement for phosphorus changes across a body size gradient with this concave downwards pattern. So what you would expect is larger animals, so things like elephants that have disproportionate large skeleton sizes would end up needing quite a bit of phosphorus. So they would retain a lot of phosphorus from their diet to maintain the skeleton. And so what is left in the fecal material is low in phosphorus. That's the purple line that you see there. But on the other side of the body size gradient, you would expect that smaller animals also have a very high requirement for phosphorus because they have high growth rates and they need it for ribosomal RNA. So what's left in their fecal material should also be quite devoid of phosphorus because they've retained it selectively. And then nitrogen, you would expect to have slightly reductions in fecal nitrogen with body size, mostly because larger animals are less capable of feeding on high nitrogen diets. And so if the phosphorus pattern and if the fecal phosphorus pattern dominates, then you would expect a concave upwards nitrogen to phosphorus ratio change over body size. And so this was what we were interested in. And we first looked at this in Mdhluli and Mdhluli Park, where we probably more looked at the larger body size part of this prediction because our smallest animal was an impala. And we found quite a bit of support for this change in nitrogen to phosphorus ratios in the fecal material across this body size gradient from impala to elephant. So you can see in the first graph, there was a slight reduction in nitrogen, fecal nitrogen with body size increase, and then fecal phosphorus decreased. You can see that because we're only looking at that bigger size gradient component of the prediction graph, you're expecting this reduction in fecal phosphorus, and we found quite a strong reduction. And because of these two patterns, we could see that as body size increased, we had an increase in nitrogen to phosphorus ratios in the fecal material. And if you look at this from the plant perspective, so the site perspective, areas that ended up receiving quite a bit more fecal material from, on average, large species tended to have nutrient input, fecal nutrient input that were high in nitrogen to phosphorus ratios compared to the areas that received fecal material from smaller species on average. They had low nitrogen to phosphorus fecal input. And on the second graph, this is where we actually then went and measured the grass and looked at what is in the grass. And the grass growing in areas that received their fecal fertilization from predominantly large animals ended up having higher nitrogen to phosphorus ratios in the grass leaves themselves. So we were really happy with this result, and this is what we published in 2020. But then Judith and Harry came and published shortly afterwards data sets that they have. They have 2 sites in Africa and 2 sites in Europe, and they were looking at the exact same questions about how body size and herbivores change the nitrogen to phosphorus ratio of fecal inputs, and they were not finding the same patterns as we were. So you can see the bottom series of graphs, maybe phosphorus was showing this kind of similar pattern, but the Kenya site did not show the patterns that we predicted, and their European sites also did not show the predicted patterns. But there's two key differences between our data sets. The one is that Judith and Harry's data set had a lot more small species, so they were really looking at that full gradient of body size that we were not. And also we were mostly looking at grazers. We only had giraffe as the only browser in our data set, and they've got quite a bit of browsing species within their data set. So essentially what it came down to in the published conversation was that our data sets are not big enough to be asking these kinds of questions. We don't have enough species and we don't have enough variation. And so that's when we teamed up with this giant group of people to try and put some more data together and tease apart these patterns. And this is where Ritwan stepped in and the 30 researchers that were willing to contribute their data, because we reached out to all the people that we knew had some kind of nutrient-analyzed dung samples for whichever reason. And we were able to put together a data frame that was more than 10,000 data points. Almost 2,000 of those data points had nitrogen and phosphorus content. And it crossed 34 herbivore species that ranged from 3 kilograms to or more than 4,000 kilograms from 52 protected areas across 10 different African countries. And so this is what Ritwan could show. This is still a lot of variation in this data, but essentially he showed the predicted patterns within this bigger database. You can see nitrogen in the top left, fecal nitrogen decreasing as herbivore body size increases, fecal phosphorus showing the predicted concave downwards pattern, and then nitrogen to phosphorus ratios in the fecal material showing the concave upwards pattern that we predicted. Despite the lots of variation, but keep in mind that this is dry season, wet season across these 10 different countries. So why does this matter? So we believe that it could be really important for ecosystem function because the impact from nutrient mismatches created by dung input can scale up to ecosystem level processes. So it could influence things like decomposition dynamics, nutrient mineralization rates, things like demographic rates. Think about like the possibility for larger seed sets if a plant gets boosted with phosphorus, and things like plant competitive hierarchies. And so there I want to show you an example that is Judith and Harry's work, just to give you a sense of why we think this could be important on an ecosystem level. And that this is a work that they also did in 2020, where they were predicting that browsers, because they eat tree leaves that are high in nitrogen to phosphorus ratios, their fecal material will also reflect that. So you can see in the bottom left graph that shows high nitrogen to phosphorus ratios in browse versus graze, and that's reflected in the dung. So browsers have higher nitrogen to phosphorus ratios than grazers. And so what they then predicted is in systems like savannas, where you have nitrogen fixing trees, that there you might disrupt the competitive relationships because nitrogen between the nitrogen fixing trees and grasses. Because nitrogen fixing trees maybe would benefit a lot more from having a boost in phosphorus, whereas grasses might benefit more from having a boost in nitrogen. So they ended up fertilizing nitrogen fixing trees and grasses in a competitive experiment with either browsers or grazers' dung. And they could show that the nitrogen-fixing trees, when they were planted together with themselves, and then compared to when they're planted with the grasses, if they were fertilized by browser dung, they did worse in competition to the grasses. So they had a competitive knock when they were fertilized with browser dung. But when they were fertilized with grazer dung, they increased their competitiveness relative to grasses, whereas grasses showed the opposite. They benefited more from browser dung, the high nitrogen to phosphorus browser dung, than the low nitrogen to phosphorus grazer dung. So this is just an illustration of how important these kinds of things could be if it scales up to an ecosystem level. So take-home messages is just that herbivore body size and feeding yield systematically shape elemental composition of nutrient return to ecosystems, and that these stoichiometric imbalances is possibly not trivial if it scales up to influence things as big as structuring vegetation, tree-grass ratios. That's it for me.
Thanks very much, Lisa. You scored a bit of time because we were messing with the timer. You have a minute if there is a question. Do we have a quick question? No, looks like you're off the hook. All right, so finally, Corley does have a question. Of course she does.
So Lisa, why do white rhinos put their dung in middens?
Because their fecal material is very high in nitrogen to phosphorus ratio.
Quick. All right, thanks very much. Okay, I think we've seen really, this morning's program has showed really nicely how our thinking of how savannas function, the importance of disturbance has really been acknowledged. And I think, I was actually sitting here thinking, I wonder what the program would have looked like 20 years ago. Then I realized actually I was at this conference 20 years ago, but I can't remember what the program looked like. Some of you other blue hats might. But I think, so the next talk, a plenary by Corley together with Sally, and I think inputs from Carlos Teva, they're going to outline how our thinking of savanna ecology has evolved over the last 20 or so years. And whilst doing that, acknowledging the contributions of William Bond into the field of savanna ecology. So over to you, Corley.
So this talk is really going to be presented by Sally. She unfortunately, it was a bit of musical chairs, and she ended holding the stick when the music stopped. We were, I think all three of us feel very privileged, but also overwhelmed to be reflecting on how our thinking has changed as a result of William Bond spending a lot of time in this room. And I know where Norman is in this room, and I also knew where William was, halfway there on the furthest row. So if you sit in his seat, you better put your hand up and ask some questions, or just shout randomly from the back. But so I'm just going to open this talk, and then Sally's she's gonna explain the serious science to us. So William occupied. We went. Sally mostly went to the internet and looked at what people have said about William, and he occupied multiple niches simultaneously, which I think is hats off to him. But William is just, you can see a world authority, especially on open ecosystems, a globally renowned ecologist. He was nominated as a fellow of the Royal Society. And we must thank Nikki Stevens and, sorry, who is the other English UK? Malvinder. Sorry, I'm just struggling with the name. In any case, we all know that person. And Sally was just saying that, usually when a fellow is nominated, they have to go onto a waiting list. But William's nomination just sailed through, and we all wonder why it took so long. At the time of his nomination, there were only 6 other South Africans that were Royal Fellows. And you can only become a Royal Fellow if you make this really worthwhile and unique contribution to our understanding of the natural world. And William said to me shortly thereafter, he was looking so chuffed, and he was like, now I can put FRS behind my name. And I was a bit slow, so I was like, FRS, what does that mean? But I've subsequently found out. So William, after he did his first degree, He came back to South Africa, met his wife, and then his very first job, he was off to Mozambique to look at the agricultural potential of land in Mozambique. This is him. I thought like such a bond, like a true bond moment on the Cabora Bassa Dam in Mozambique, where he also worked on water weeds. That was one of his first papers. This is how we often used to see William down a soil pit. And I had so many field excursions where William would be walking around with the auger, and we would be augering along on Katinas, and he would be explaining what we're looking at. So that was very influential in shaping him to become the ecologist that he turned out to be. And then I'm going to pass this on to Sally.
Thank you, Corley. I'm the tall one here. Yeah. It's amazing to be able to reflect on a lifetime of an ecologist like William. If you see those pictures, they were taken all about 20 years apart. So more than 60 years of doing the exciting things that all of us in this room do. I think it's really important to reflect a little bit that he started his academic career in the Fynbos, where he did some really fantastic work on ant-plant mutualisms, looking at agents of diversity and extinction. That photograph in 1979, I was 4 years old at that time, and he was already writing exciting papers. But then he, like the grasses that he loved so well, he spread his wings and he moved into savannas. And he started to drive some really important theoretical advances that we'd like to discuss today. And I think just to highlight that it's the combination of these unique perspectives and then amazing storytelling abilities that really helped William to ignite so many of our imaginations and help us to be better scientists. Corley's already said that he was a big part of creating the exciting idea fest that we have here at the network meeting. And it's wonderful to see the hands going up in the rooms, even now today. Don't be intimidated. Put your hand up and ask some questions, guys. It's also really important to notice that, science is a totally collaborative enterprise, and it's really hard to see the contribution of one person, especially one person like William, who was so good at having collaborators and bringing in people to help him create his ideas, challenge his ideas, and move forwards. So we just want to say that if we're focusing on William here today, that doesn't undermine the fact that it's been a group enterprise. And the insights we've got about savannas and the natural world are something that we've all contributed to. I really had a good time looking through his papers from his pre-Savanna period. And I think it was really clear to me that a lot of the really important insights that he brought to savannas, he was already writing about when he was in the Fynbos. His PhD was on the regeneration, all different sorts of regeneration, and basically focusing on demographic processes and how really important it is to understand demographic processes if you want to understand community scale impacts and patterns. And then a very influential paper looking at angiosperms and gymnosperms back 160 million years ago and the fights that they were having to take over the globe. Key insights that were already in his head then were about how plant traits, there's this interaction between resources and disturbance in plant traits that help to determine what patterns emerge. And then finally, this is something that I've really found useful and was very hard. It was hard for him to fight with people about this, is that plants have the capacity to alter ecosystem function, and therefore that can be driven by natural selection. So evolution is involved in driving these abiotic processes. It's not deterministic. And that's going to be a theme for the rest of the talk, I think. So it's really hard to summarize someone's career who's published over 500 papers. We thought maybe we'd try to group it around 3 major innovations that he brought to savanna ecology: demographics in the persistence niche, consumer control of global ecosystems, and then bringing in a deep time perspective and thinking about ancient ecosystems and their conservation value. When it comes to demographics, the sort of paper that really started his contribution here was a paper that he wrote with Jeremy Mdhluli about resprouting and the very importance of resprouting and the persistence niche
for thinking about ecological theory. And to be honest, like how many years later, more than 40 years later, we still haven't managed to bring the persistence niche into our theoretical understanding of plant life history strategies. But everyone realized after this paper that it was important. And he developed this idea and this term Gulliver. Gulliver being a plant, a woody plant that is stuck in the grass layer because the grasses keep fueling these frequent fires and the tree can't grow fast enough to escape the fire. And he used the word Gulliver because of that Gulliver's Travels, which I don't think very many of us in the room have ever read, which was this huge man who went to visit an island where these teeny tiny little people captured him and tied him down to the ground. And so he thought this was a really nice way to illuminate the idea that these trees have capacity to be these huge giants and get constrained by the grass. And he and Steve Higgins built that into a new theoretical model for tree-grass coexistence, a demographic bottleneck model that focused on the non-deterministic nature of these systems, which everyone learns about in undergrad these days, I hope. But key, even in his paper in Tree, was this idea that if it's all about resources and plant traits and disturbance, then if you increase the resources, then you totally shift this tree-grass interactions and the factors that are controlling these ecosystems. And he pushed for many, many years on this idea. If you look at this graph, which many of us have seen, it's from his original paper. It's not very beautiful, but I wanted to put this one in. Basically, if a sapling needs time to grow above the flame zone to escape the fires, if you give the sapling CO2, it's going to grow more quickly and more likely to escape. And that was his paper that he wrote with Guy Mdhluli. There were some pushback on some of these ideas, and William pulled together multiple lines of evidence to try to approach and demonstrate the importance of these ideas. And that's the thing that I, as a scientist, have taken away, is that he wants to ask a research question, he will draw field data modeling, he'll go and speak to an isotope scientist, he'll persuade his master's student to learn how to do dendrochronology, because it's about answering the question and persuading people with as many lines of evidence as you can. I wanted to highlight two papers, neither of which has really been cited as much times as it should have been, I think. This one focuses on the demographics, the fact that you've got these two different plants, one of which has traits for fire and one of which has traits for herbivores, and that you can use dendrochronology to plot how demographic characteristics interacting with plant traits shift the entire community from a herbivore adapted to a fire adapted community over time. And here's one he did with Ben Widgley that just really showed that these Gullivers are needing to have their stored reserves. They really need carbon, which means CO2 might be an important driver of these systems. It wasn't all plain sailing. He got a lot of pushback on the CO2 because he and Barney Hooper and Guy Mdhluli were doing these experiments in these teeny tiny greenhouses with teeny tiny plants, and no one believed him. The whole world was moving away from thinking CO2 was important. But he pushed and he mobilized and he gathered communities and he gathered resources and he persuaded the South African government to fund this huge, big elevated CO2 research facility that we now have at Rhodes, which is pumping out like 2 or 3 papers a year, showing some really fascinating and useful insights about how CO2 is driving our savanna ecosystems. And I think we can be really grateful to him for that. Also, people took his ideas and ran with them. Everyone got into the idea of if you can have a fire trap, then you must be a browse trap. And we've really managed to explore how these demographic bottlenecks drive ecosystems. Certainly, he's really advanced thinking about life history traits in these savanna environments. And that's expanded wonderfully. And people have taken it up to think about management, because if you're managing a system that's non-deterministic, you really have to approach things really differently. Of course, William took these ideas further as well. He'd always been quite uncomfortable with the sort of classic Clemensian view of an ecological climax, that everything's determined by climate and soils, and that if you know the climate and soils, you know what's there. Holdridge and Clements pushed these sorts of ideas. There's a paper by Hairston, Smith, and Slobodkin that sort of challenged this a little bit. They were like, well, what about the herbivores? You know, if there were herbivores around, why is the world still green? But still quite focused on environmental determinism. But William kind of turned this on his head. He asked, well, what if the world isn't actually green? Here he is, staring at you on this really important paper that he wrote back in 2005, I think, telling you that you've basically been asking the wrong question. There's some more interesting questions to be asked. So he highlighted that there are large parts of the world where herbivores and fire are in control. And to do that, he teamed up, multiple lines of evidence again, he teamed up with a global vegetation modeler. And just all he did was put his demographic bottleneck model into a DGVM and publish this map to show, oh my goodness, if you add fire to a DGVM, you actually represent vegetation distributions way better than you did before. Before they put fire in, they thought the whole world was a forest. And this was an incredibly influential paper. But he didn't stop with the modeling. He gathered a bunch of fire exclusion experiments to show that the model actually reflects what you get in reality when you switch fire off. So he coined this term, which he borrowed from Whitaker, ecosystems uncertain. There are a lot of ecosystems that you really just don't know what it's going to be. You have to look at the herbivores and the fire. It was at this stage that with the help of the Mellon funding, he pulled together the Zululand Treegrass program, which was focusing on the brown world, the herbivores, and the herbivore interactions with the fire. This is a fantastic paper where they used the rhino removals that were happening in Hluhluwe and Mdhluli Park to show that when you don't have rhinos in a system, once you've removed rhinos, the fire size just expands. So these two herbivores, these two consumers, fire and herbivory, are actually interacting with each other and suppressing each other. Then on the non-deterministic side, the exclosures that they'd put up, yeah, when you put up an exclosure, the grass grows because it's not being eaten, and the trees grow because they're not being eaten. But isn't it exciting when you take down the exclosure, when you remove the fence, the grass gets eaten again, but those trees did not go down because they had escaped that bottleneck. And so, again, evidence that these systems are not deterministic, and it really depends on the history. Without the history, you don't understand the system that you're looking at. It was very influential. Yet again, other people took his ideas and ran with it. There was a lot of thinking about, okay, so we've got these three different worlds. What type of processes are controlling the dynamics of these ecosystems? We also looked at, well, can we try to map these different types of ecosystems and where they dominate across the globe and across Africa? And one of the most rewarding things for William over the last little while is to have the temperate ecologists who developed these deterministic ideas about climate and soils wake up and read his papers and everyone's papers in the room and be, oh my goodness, maybe here in temperate Europe, our systems are also herbivore controlled. We also get brown world and herbivore controlled ecosystems in temperate forests. So all of this exciting science, and we felt like we'd really made some progress in understanding how things were going. And yet you still have these maps published by the World Resources Institute that look like the DGVM from 1995 that he and Ian Woodward showed was wrong, saying this is the world, this is the potential forest cover, and this is what we should be restoring the world to. So global conservation and restoration continued to view non-forest ecosystems as not natural. Madagascar, just a case in point there. They have like large expansive grasslands that were always termed secondary grasslands. So William, instead of getting disheartened, he just focused his efforts to try to unseat this forest bias, which he sort of highlighted originated 200 years ago with von Humboldt and again, some European ecologists who traveled the world. What did he do? Well, multiple lines of evidence again, of course. He and Nick Salumis did a great job of showing that there's a big difference between different types of grasslands. Secondary grasslands, yeah, that used to be forest, yes, they are species poor, but you get these old growth grasslands that are ancient and have been around for millions of years that are incredibly diverse. And they have all these unique growth forms that would disappear because they require the light. They're plants in the understory needing the light that would basically be excluded and go extinct if you didn't have these open grassy systems. He also took a deep time perspective again, and I think he's done a great job of showing us how we really can, mobilize thinking around long-term timescales to understand what's going on. Here's a data set he pulled together from inertinite in the ocean. It was a crazy idea to demonstrate how fire and these crazy underground trees that require light, they all evolved way before people were even way before any type of human had ever started to roam the plains of Africa. So, like a lot of work showing that novel fire regimes, rather than just climate change, were really instrumental in these big deep time processes where flowering plants and grasslands spread. He also realized that you can't just talk to people here at the Savanna Network meeting. So with his daughter Elizabeth and a couple of other people, Carla, I discover you're also involved, he teamed up to create a really wonderful video, kind of telling the story of grasses, telling the deep time perspective, pulling all the data together into a story. I've put a little QR code. There's 34,000 views, but you can view it as well. So please look at the QR code. He went into doing more sort of proactive activism, talking about science and society, and challenging the afforestation plans for Africa. It all came to a head with the Madagascar story. There was a lot of pushback on his claim that there were ancient grasslands there. It became quite embittered. But again, what William did is he just teamed up with some people who weren't even plant ecologists to gather more lines of evidence to demonstrate that these grasslands were ancient and that forests had always been patchy and around in grasslands, because you could, using information about mammals and mammal evolution, you can demonstrate that forest grassland mosaics were natural in Madagascar. It hasn't always been plain sailing. I just wanted to highlight yet again, there's always been pushback to some of his ideas. An entire new phytologist workshop was organized in Ghana a few years ago to try to debate these things. It's always going to happen. And I think the inspiration for all of us in the room is when you get a reviewer who's not particularly happy with what you've said, is not to, what William did, sure, he would swear a lot, and he would grind a few roots into the ground, but then he would just go out there and do some fun science, because ultimately, if you can find the data and the evidence to persuade people about your story, then you're doing the right thing. And if your data disproves you, then you've got to change your mind, and William did both of those things. It's amazing to have this talk here in this session, because he would have just, the last 2 or 3 hours of talks have already shown how the ideas I presented in this talk are, we're steaming ahead with a bunch of these really exciting questions around tree-grass interactions, around herbivores. I suppose I just want to inspire everyone in this room to keep going. He was a really great mentor and was always interested in listening to your ideas and encouraging you to go out and do great science yourself. So I'd like to maybe just hand over to Corley for the final thoughts around William and his amazing legacy. Thanks.
Thanks so much, Sally. So just what did William also mean just in terms of the Savanna Network meeting? He was one of the group of people who was instrumental in bringing the Mellon Foundation and giving us this wonderful conference. And I have one of the blue hats. I'm so sorry for the American colleagues if you got a red hat. But I was in the room in 2003, and what we now just accept to be, the theory underlying savanna ecology, the thinking was actually quite different in the room back then. And William also celebrated the launch of his open ecosystem books here at the Savanna Network meeting in 2020. We could have no idea that a few weeks later we'll all be in lockdown. William was just so good, as Sally said, to pull different people together. He just had a really good handle on what it would take to answer a question. And he wasn't shy. When I speak to modelers, I get very nervous. Or to mammal ecologists, that's why Sam is sitting on that side of the room and I'm here. But William had no such qualms. He just got everyone together and talking. And we, as one of William's PhD students, we went to the field so much with him, and he was so good. He was constantly inviting you, and it wasn't necessarily anything to do with your work. So I ended up spending a lot of time in Fynbos and in the Cedarburg and in forest and all over, and he was just so generous with his time. And I think he mentored so many more students than just his postgrad students. And then he was Unfortunately, I couldn't find any other photo, but this is Mdhluli, who used to be a lecturer at the University of Venda. I remember also so many field trips with the University of Venda. William was great in just, kind of getting institutes together that traditionally weren't working together. And then he was great in just bringing international scientists to UCT. And I think that, like back at UCT, we just remember so many great names, Peter Vitusek and Simon Levin and all these amazing scientists that you actually just saw their names on paper and then you could meet them in person. And that was really amazing in a country that wasn't out of apartheid that long. So we just want to thank everyone that has been on this journey. And when I was listening to the talks earlier, like Sally said in this session, I thought how almost every one of those talks have been influenced by the work of William. And then I just also want to say thank you so much to his sister Elsa Pooley, who gave me a lot of information about his earlier years and filled in some of the gaps. And thank you to his fantastic family. And then thanks, William. We hope you're still with us in spirit. We will celebrate William later this week in a separate event. Thank you.
Thanks to both Sally and Corley for a very moving talk. And yeah, like she says, there will be another function on Thursday night. So I'm sure you're all aware of that. Just to wrap up the session then, just to also point out that there are 3 very interesting looking posters attached to the session. So please do be sure to go and check them out tomorrow at the poster session. And yeah, I don't want to get in between you and your lunch. So from here we go to lunch. Can we maybe have one last round of applause for everyone in the session?
Hi, afternoon, everybody. Can we settle down so that we can get started, please? So before I hand over to Don, just a quick announcement. So on the program for tonight, we had from 5 o'clock until 6 o'clock, we had the Lightning Connections, one conversation at a time. So that will happen immediately after the formal session this afternoon. So please join us. That's a fun activity, and there will be prizes. And then there will be some time between that and the dinner that starts at 6:30. So enjoy the afternoon session, and I'm handing over to Don. Thank you.
Hello, everybody. Welcome back from lunch, and welcome to the two sessions we have this afternoon. To start things off, we're going to have a session on understanding biodiversity in changing savannas. And first up, we have Lila van Zyl on a talk on exploring elephant functional role in shaping ecosystems.
Hi everyone, I'm Leila, and today I'll be speaking about elephants and ecosystems. So despite populations of savanna elephants in southern Africa showing positive, negative, and stable population growth rates, the IUCN Red List classifies the African savanna elephant as endangered and the African forest elephant as critically endangered. In addition to their conservation concerns, elephants also play an important role in ecosystems. Megaherbivores, such as elephants, have disproportionate impacts on ecosystems by affecting their structure as well as functioning, by reducing fire extent, facilitating seed dispersal, and impacting woody vegetation structure and the associated biodiversity. Elephants have direct effects, such as that on vegetation, as well as indirect or cascading effects, where the initial disturbance propagates through a system, resulting in secondary and tertiary impacts. An example of this is where the vertebrates respond to the vegetation changes induced by elephants. So as we all know, elephants transform thicket and woodland areas into open savanna landscapes, dominated by grasses. This transformation of the landscape changes the spatial diversity and nutrient composition of vegetation. And while this change in the landscape may negatively impact some species, it also benefits others. And this makes the decisions that conservation managers have to make to maintain biodiversity quite tricky. One approach to synthesizing results from various studies is a meta-analysis, which is essentially an analysis of analyses, and allows us to form more precise estimates of the relationships between various variables. So, a meta-analysis by Robert and colleagues in 2008, investigating the impact of elephants, showed that elephants have both positive and negative effects on woody vegetation at low densities, but they only have negative effects at high densities. And this influence was shaped by rainfall and the presence of fences. So just some background on forest plots, which is the standard way in which the results of a meta-analysis is displayed. The mean effect sizes are indicated by the squares, with the horizontal lines representing their confidence interval. The squares are also weighted, with larger squares indicating a larger sample size. Where the confidence interval overlaps 0, which is indicated by the vertical dashed line, these are classified as neutral impacts. And positive or negative effect sizes with confidence intervals excluding 0 are classified as positive or negative effects, respectively. Another meta-analysis by Robert and colleagues in 2017 reevaluated the findings of their first meta-analysis, taking the new literature into account and extending the responses considered to investigate cascading effects. They found that elephants have an overall negative impact on tree structure and abundance, while no overall negative cascading effects on vertebrates or invertebrates were found. So, because studies report such contrasting positive and negative effects of elephants, we revisit the systematic review and meta-analysis by Goldemont et al. 2017, including the enormous amount of new work published since then, to synthesize these findings and assess their net effects across different categories. So, why are we doing this study? Well, understanding the functional role of elephants in ecosystems will help us better understand how they shape these systems, and this may allow conservation managers to make more informed decisions when implementing management strategies. So, to conduct our meta-analysis, we followed this outline where we did a database search, screened the results, extracted the data, and analyzed the captured data in R. So, to give you more detail on our workflow, I searched the search engines listed here for peer-reviewed research articles using Boolean operators to construct these search terms. I had the inclusion criteria listed here and included both savanna and forest elephants, and these will be analyzed separately. We may also explore including correlation data at a later point. For the papers meeting the inclusion criteria, I extracted the data, and then I analyzed the captured data in R using the metaphor package. So to evaluate the impact of elephants, variables measured in their presence were analyzed as treatment observations, and those in their absence as controls. HSG effect sizes were used and calculated for direct and cascading impacts, as this gives higher sample sizes more weighting. Direct effects included responses of vegetation, and indirect impacts, the responses of vertebrates, invertebrates, and soil properties. The effect sizes calculated included for structural changes, abundance, population dynamics, diversity, as well as processes for all of the different categories. Our search returned over 7,000 results, and we are only about through 70 of those, so our results are still very, very preliminary. So, taking you through the results. In these plots, the HSG effect sizes are shown by the squares, and the effect sizes and their confidence intervals are shown on the right side of the plots also. So, in panel A, the effect sizes for the various categories as a whole indicate negative effects on trees and vertebrates, with neutral effects detected for all the other categories. In panel B, no overall negative direct impacts were seen, but negative cascading impacts were detected. Looking at the effect sizes for the various impacts, and we're looking at the direct impacts, we can see neutral, negative, as well as positive effects. While herb structure was not affected, abundance was lowered, but diversity increased. For trees, the effect sizes indicate that elephants change the architecture of savannas by changing tree structure and lowering abundance. So, looking at the indirect or cascading impacts. Neutral effects are seen for vertebrates and abiotic processes, with negative effects seen for vertebrate populations. In conclusion, our preliminary results indicate that elephants change the architecture of savannas by changing tree structure and lowering their abundance. The differences in study design between studies can potentially help us understand the substantial variation we see, and therefore we will include these as covariates in our analyses from now on. So, just a disclaimer, it is important to keep in mind that these results are not an accurate representation of the literature, as we are still in the very early stages of the critical appraisal of our search results. And there's lots more to review and unpack, so stay tuned. In closing, I would like to thank the Conservation Ecology Research Unit at the University of Pretoria, without whom this study would not be possible. Thank you all for listening. Any questions?
We do have some time for a couple questions, if there are any.
Thanks. I was just wondering, when you do your meta-analysis and you put your search terms in, it seems, if you're putting in, you want to find the negative impacts, or you want to find impacts.
Yeah.
Does it bring out any potential, and obviously there's way more negative than positive, but will it bring out those positives so that you're not directing the search down a certain line?
Yeah, so my searches aren't just for negative impacts, it's just impacts, but that should include then the positive and negative impacts of that, if that makes sense.
There's a question on the right.
Thank you very much for that, that present. I'm glad you guys and Rob and them are revisiting his papers, which everybody queries so much nowadays. Where's Rob? But I think one of the things that would be interesting to do is there's certainly some understanding that the consequences of elephants are not necessarily related to whether they're just there or not. And it's also not necessarily related to whether how many of them are there or not. It's actually how the activity is and what they're doing. So how can we complement this analysis with getting on the x-axis something about the indicators of activity of elephants, where they're doing, who's doing it, whatever they're doing. And we may get, I think, some additional insights which are currently buried in all the strange outcomes that we see. Just a suggestion.
Yeah, thank you so much. We will definitely keep that in mind. Thanks.
Okay, well, thank you very much, Leila. Next up, we'll be sticking with elephants and talking about the indirect impact of elephants on bird and bat assemblages by Karina Fouché.
Hello, everyone. I'm Karina Fouché, and today I'll be talking about the indirect impact of elephants on the bird and bat species assemblages. So just a quick background, which I believe most of you would know. Elephants change vegetation structure, and this change in vegetation structure can have an impact on the species of birds and bats that we can find in that environment. So my study site was in Timbavati, and a newly incorporated property named Skoongezicht, which was incorporated in 2024 in March. So just some comparison pictures. On the left, you can see that Timbavati is a much more open landscape. And on the right, Skoongezicht has much more trees, and there's some early signs of elephant impact in those pictures. So the aims of my study is to assess the differences in vegetation structure, as well as the bird and bat species assemblages, and then also to see how elephants act as drivers in changing vegetation structure, and to identify patterns of vegetation structure and functional group composition. So these are my site locations. On the light green is the newly incorporated property Skoongezicht, and the darker green is the Timbavati Nature Reserve. So firstly, vegetation. Elephant impact was, for each site, it was scored from 0 to 7, and we just noted proof of elephant presence. And then for vegetation, we looked at the clutter of vegetation for both herbaceous and woody. But for today, I'll only be presenting on the woody because it was a much more significant difference. So this is just some pictures to show of proof of elephant presence, broken branches and some mud rubbed off on trees. And then for my methods for birds, what we did is at each site, we had a 10-minute observation period where we just stand there and recorded all of the birds that we heard, as well as a recording that was made that afterwards I went through to double check if we missed any species. And after identifying the species, we placed them into their functional groups. Here's just a picture of kaleidoscope, which I used to help identify the different calls to make sure I'm listening to different birds. And then for bats, I used audiomods. They were placed in their most likely flight pathway, and they recorded early mornings and late afternoons. Just a nice picture of how we placed our audiomods. And then our results for the elephant impact. For Timbavati, we had a greater impact in comparison to Skoongezicht. But when I looked a little bit further at my study sites, you can see that there is Timbavati's sites, so the darker green, there's sites much north and there's sites much more south. So I just investigated them a bit further to see if there's a difference in the elephant impact. And I found that Timbavati north was less impacted than Timbavati south. So this gave me a way to investigate it in a finer scale, looking at Timbavati south as a high elephant impact, and Timbavati north is an intermediate elephant impact, and then Skoongezicht as our lowest elephant impact. Here is a nice graph to show what the woody clutter looked like between Timbavati and Skoongezicht. So Timbavati, you can see that the box plot is much more thinner, so it's much more homogenized vegetation. And then with Skoongezicht, there's a broader box plot showing there's a bit more heterogeneity in the clutter of woody vegetation, as well as there's a bit more sites that had more woody clutter. So then here again for woody, but this time I've plotted my box plots to show the difference in the stratum levels. So on the top is like our 7-meter stratum, then 3.5, 1 meter, and on the bottom is our 0.5 stratum level. So in each one of these, we looked at the clutter volume. And then for Timbavati, you can see at the lower stratum levels, there was a higher clutter volume for woody vegetation and lower in the higher stratum levels. And then for Skoongezicht, it was the other way around. So we get this inverted triangle pattern. So this is where I splitted Timbavati north and south. And as you can see, Timbavati north is a little less homogenized than Timbavati south. And then Skoongezicht is the same graph than the previous one. Now I just want you to take a closer look at this graph, because the next one that I'm showing is the new data that the student after me has collected. So in 2025, just to show the difference in the stratum woody clutter, how that changed. So here's 2025. As you can see, it changed quite a lot. And even with Timbavati north and south, they have been more homogenized. And we did sample the same sites. Okay, then results for birds. What we found is Timbavati was more species rich than Skoongezicht. And we also found that Timbavati north was more species rich than Timbavati south. So Timbavati south had the higher elephant impact than Timbavati north. Okay. So for our functional groups for birds, both Timbavati and Skoongezicht, the arboreal functional groups decreased and the terrestrial functional groups has increased. And then looking at our incidence-based richness, Timbavati has decreased in species in richness, but if you look closer, you can see Timbavati south actually increased, which had the higher elephant impact. And then for Skoongezicht, overall the species richness has increased. And then looking closer to just Skoongezicht north and Skoongezicht south, we can see that Skoongezicht south has also increased, where north has remained more or less the same. And from just observations from some of the people in the field, it seemed that Skoongezicht south was also impacted more by elephants. And then for evenness, we can see overall that the evenness has dropped a bit from 2024 to 2025, meaning that some of the species became more abundant and some of the rarer species became less. And then difference between our sites. In 2024, Timbavati, from 2024 to 2025, the sites became less different from one another, so they became more homogeneous when comparing them to each other. And Skoongezicht also became more homogeneous, but not as drastic as Timbavati. And then for bats, we didn't find a clear pattern. We did find mostly exploiter and adapter species, and then Timbavati was definitely more abundant and active when looking at bats. So here, when we looked at the phonic groups of bats, in Skoongezicht, we actually found more open-air feeders than in Timbavati, which was unexpected, because we obviously expect in Timbavati, which was our more open landscape, to find more open-air feeders. But we expect that the open-air feeders just feed above the dense vegetation in Skoongezicht. And they also tend to call louder than the other species, so they are picked up more in the audiomoths. Then for richness, Timbavati's bats actually increased from 2024 to 2025, and then Skoongezicht seemed to have decreased from the 2 years. evenness. There's not drastic difference, but as you can see, some of them up and down. This just shows that I expect that the lever and what's driving behind this is not really in species richness, but more the composition of which species you're finding in there. And then looking again at the difference between the sites. So for Skoongezicht, between the 2 years, became much more homogeneous in what bats we found there, and Timbavati more or less remained the same. Then looking at vegetation, my finding is that at high densities, elephants homogenize vegetation, but at intermediate densities, they increase the vegetation heterogeneity. And then for bird assemblages, at intermediate densities, we had the highest bird diversity, and at high densities, we have more terrestrial and less arboreal birds. And for bats, they were differently impacted than birds. And at high densities, we have less open-air foragers and more clutter-edge foragers. So management implications just shows how we can use birds and bats as indicator species and how to identify drivers in the savanna and just to know what's going on behind the scenes and how we can improve management, especially also for smaller reserves. And then limitations of this study, I do think that the resolution of faunic groups is a bit limiting, and especially with seasons, because some animals or birds even do diet switching between the seasons. So I really think it would be nice to look at different seasons and how that changes the picture. And then all of this for me shows towards the intermediate disturbance hypothesis. We can't really say elephants are bad or good, but intermediate disturbance does give us a higher diversity. It promotes niche diversity, and of course, as always, further investigation would be needed. Okay, thank you. And any questions?
Thanks, Karina. Is there any questions from the group?
Hi, thanks. That was a super presentation. I really enjoyed that. And I think the best slide was the slide at the end where you talked about the intermediate disturbance hypothesis. And so just going back from there through your presentation, your slides were high elephant density, low elephant density, high elephant density, low elephant density. And there were two sort of parts of the disturbance gradient. But at the end, you talked about the intermediate disturbance hypothesis. And I would just suggest that for your publications and so on, you stick with a disturbance gradient as opposed to a comparison of high and low. Mm. And what I would really like is to see more studies such as yours, but which are filling in that disturbance gradient. So we can see where that sweet spot is in terms of biodiversity in relation to elephant impact.
Yes, I definitely agree with you. I also think that would be really nice to investigate that further. But no, this is what we have for now. But for future, it would be nice. Thank you.
We do have time for one more question, if there's... Front.
Thank you for your talk. It was really interesting. I was just wondering, you mentioned that you had more exploiter and adapter species in TIM. Was that right?
Yes, that's correct.
And can I ask, what that is in that species functionally and why that might be the case?
So the thinking behind that is because it's species that are more abundant, so they adapt more easily to these changes in their environment, so they take over the other species that's a bit more sensitive for these kinds of things. So.
I see. So it's not a specific, it's just species which are good at adapting or exploiting the changes in conditions caused by the elephants.
Yes, and exploiter species also refers to species that exploit others as well. So they push them out of that niche. If that makes sense.
So maybe they're sort of muscling in when the elephants are creating chaos. Is that the idea?
Just say that again?
So your exploiting species are taking advantage of the increased sort of heterogeneity or the increased disturbance by elephants to muscle in on other bats.
Yeah, that is. Yeah.
Thanks.
Thanks very much. Thanks for a great talk.
Thank you.
That's all we have time for now. Next, we have Catherine Rosiak, who will be talking about rhinos and rhino middens and their effects in savanna landscapes.
Hi, everyone. My name's Catherine Rezage. I'm a PhD student in Andrew Davies' lab at Harvard University. And for those of you who are unfamiliar with rhino middens, they are communal defecation sites or latrines that play a really important role in olfactory communication. But what I want to talk about today is what I think is an important but perhaps overlooked role that they play in shaping savanna heterogeneity. So we know that in savannas, mega-herbivores disproportionately affect ecosystem functioning. Our rhinos, hippos, and elephants impact vegetation structure, biogeochemistry, and biodiversity. And we know that white rhinos, as the world's largest megagrazer, consume an outsized portion of forage. And in doing so, they engineer these short grass grazing lawns that increase local productivity, alter vegetation structure and fire patterns, and also influence grazing assemblages. And rhinos aren't the only species that are attracted to these high nutritional grazing lawns. Other grazers also utilize them, and this increased grazing intensity also leads to increased grazer dung and urine deposition. So what we have are these species that are replenishing the nutrients they consume and maintaining these biotic nutrient hotspots at grazing lawns. But rhinos don't replenish the lawn nutrients they consume. So if you see on this graph on the right from a 2017 study, we have expected dung output on the x-axis and observed dung production on the y. And what we see is that white rhinos deviate significantly from this. So they're not finding their dung on these lawns like they do with the other species. And that's because rhinos have this unique behavior where they transport their nutrients to rhino middens, these communal latrines that they almost exclusively defecate in. But what we don't know is that if like grazing lawns, these are also biotic nutrient hotspots that concentrate this nutrient-rich dung and increase savanna heterogeneity. And when we visit middens, we just see a lot of these effects already. We see these clear green halos around middens with distinct species compared to those we see in the matrix. We see lawn-like areas of intensive grazing around middens. And we also see non-rhino species using middens, like this baboon and these hornbills foraging in them. So to start quantifying these observations, we focused on an area of high rhino density in the Kruger Intensive Protection Zone. And we have 16 paired midden sites and 8 control sites. And at these sites, we're conducting midden surveys. So we are collecting soil and plant samples to measure nutrient levels. We're measuring grazing intensity and biomass and characterizing species composition at this gradient radiating away from the midden so we can understand how these effects persist into the matrix. And we only just conducted these surveys in February for our first season, so I'm just going to focus on species composition and biomass preliminary results. So we saw that plant community composition differed strongly with distance from midden. You can see that on the midden and immediately adjacent to, the composition of these communities is really distinct and much less varied compared to distances 5 meters and beyond, which is a lot more heterogeneous and matches what we see in our control sites. So you have this distinct separation between the midden footprint that has this transition zone at 2 meters that's really separate from these areas 5 meters and beyond. And we see that the species driving this midden gradient vary as well. So on and adjacent to the midden, you have this clear domination in cynodon. So the mean percent cover is just really, really dominant, but it drops significantly, almost to nothing, at 5 meters, which corresponds to this increase in Eurocloa, Digitaria, and Aragrastis grasses. Although this percent cover is obviously a lot less dominant, and that's due to the corresponding increase in species richness that happens at the same time. But we are seeing a lot of these same grasses that we see on other biotic nutrient hotspots like grazing lawns and on termite mounds as well. So finally, we also saw that distance from midden significantly affects plant biomass. You can see that biomass is a lot lower immediately adjacent to middens, which makes sense with the low-lying cynodon that dominates there, and also could be due to intensive grazing, but we're still in the process of analyzing those data. And we have this distinct peak at 5 meters, which is really different from what we see at 10 meters and at our control sites. And while we know this corresponds definitely with the change in species composition, it also could be due to the increased nutrient enrichment at these distances that promotes productivity in this intermediate space, though we are waiting for our lab results from our soil and plant samples to confirm that. So while we were looking at these effects on ecosystem function and structure, we also wanted to understand how other species, like these grazing impala, were using middens and not just rhinos. So we put up camera trap surveys at our midden sites, and it's taking a while to annotate these videos, but from the first 8 middens that we've analyzed over about a month of time, we recorded 1,000 distinct visits by different species, and a visit is when an animal is interacting with the midden, not just passing by the camera. And this was from 36 species, but you can see that certain species really dominate. So for our mammals, we see a lot of impala, as well as many elephants, and from the 18 bird species we identify, we see a lot of foraging starlings and hornbills. We saw a lot of variation in the behaviors we observed, but grazing and foraging behaviors really did dominate. So that means that these middens are really a resource for food for a lot of these species that are using them. But we also saw really interesting and less frequent behaviors like dust bathing or scent marking in the middens and using them for bedding sites as well. And you can see some of these here. We have our hippos grazing at middens, we have honey badgers foraging in them, these wild dogs investigating and then marking, and this elephant that was rolling or dust bathing or doing something really weird. That was an interesting one. So we are starting to see that middens are having an effect clearly on savannas. So we want to know how long these midden effects persist for, especially since we're losing rhinos at a really rapid rate, of course, due to poaching. So what we're doing is setting up a midden exclosure experiment, where from our 16 sites, we're fencing off 8 of them to make them hopefully impassable to rhinos. And we constructed a trial of this cable fencing that keeps out rhinos last March, and it did do just that. Over the 6 months that these trial fences were up, we didn't have a single rhino breach, which we were really happy by. So although this was just a trial, we have seen that some of these vegetation effects seem to persist 6 and even 9 months post this forced abandonment. But we're really looking forward to start measuring these as our exposures go up this month. So I've talked a lot about these local impacts of middens, but what I also want to quickly show you is how we're scaling up our understanding of midden impacts across the landscape. And we're doing this by using remote sensing to map middens. And we conducted a study showing that we could do just that, again, in the Kruger IPZ. We used RGB thermal and LiDAR imagery to map over 100 middens across 800 hectares. And when we looked at which features influence midden placement, what was actually most interesting for this work are the factors that don't influence midden placement. So we saw that midden location wasn't significantly influenced by slope or relative elevation. And the reason this is so important is because the work we're doing is on these granite soils, and that is characterized by this undulating topography in the Katina sequences where we have this gravitational and rainfall downpull of nutrients, and that creates nutrient-rich foot slopes and nutrient-poor hill crests. So if we have this localized nutrient gradient, but we have midden placement that's not affected by slope or relative elevation, that means rhinos are placing their middens in these areas of nutrient-poor, areas as frequently are in the nutrient-rich areas. So we're hypothesizing that rhinos are transporting nutrients against this biophysical gradient. And that means they are counteracting this gravitational downpull, this gravitational loss of nutrients. And we do have rhinos that are tagged in the IPZ currently. And we see from these these elevation movement profiles that rhinos are indeed using both the crests and the slopes and moving up and down these Katina sequences quite frequently. So our goal is to combine our midden surveys, our midden mapping, and our rhino movement data to really understand how rhino nutrient transport works and counteracts some of these physical gradients we see. So to wrap things up, I just want to emphasize that our goal here is really to quantify effects of middens on savanna heterogeneity over space and time. So we're using these tools like remote sensing. In fact, next week we are going to map over 300,000 hectares in the IPZ, so that will be a lot of midden mapping for me. We're also tagging 40 more rhinos in the coming months, and we plan to link these local measurements from our survey sites and our experiments to really understand these landscape impacts across entire rhino populations. And the reason this is so important is because we know that over time we're losing rhinos at such a rapid rate due to poaching. So what we ultimately hope to show is yet another reason why rhinos are such important ecosystem engineers in our savannas and what our landscapes stand to lose as we continue to lose rhinos. So thanks very much, and really special thanks to Tanya Kuhlman, Rowan Jordan, and Patu Malota, who have done so much of this field work and really keeps this work going for us. Thank you.
Thanks, Sam. Thank you. We do have time for a question or two.
Thank you. That was a lovely presentation.
Thank you.
Going to your slide where you showed grass biomass with distance from the midden, you found that there was a sort of a halo effect of about 5 to 10 meters of high grass biomass around the midden.
Correct.
And that's exactly the same pattern that we're finding around elephant carcasses.
Yes.
The same place. And so what we're finding is that there's an interaction between high nutrients at the middle of the site, which can create toxicity at those very high levels, but also very high trampling at the center of the site, which tapers away. And you're getting that halo of grass where there's nutrients and not too much trampling. And so it'd be really interesting if you could do some soil penetrometer surveys out from the midden and just look at soil compaction.
Yes, I'd love to talk more about that. A lot of the elephant carcass work has inspired how we're analyzing our own data, so that way we can make these similar comparisons and look at that. So it would be great to talk more about your strategies.
Great.
Thanks very much. I think that's all we have time for right now, but thank you very much.
Thank you.
Next up, we'll be looking at Miko Jar talking about nonlinear biodiversity responses to herbivory in African savannas. Thank you.
Good afternoon, everyone. I am Megha, and I'm a postdoctoral researcher at University of Oxford. And today I'll be talking about differential response of taxa to herbivory in East African savannas. Not that this audience need this introduction, but this is where the tropical savannas are located, and that small picture is where I work in Kenya. One thing that unites savannas overall throughout this area is presence of disturbance. Interestingly, also along these same latitudes, we find tropical forests, but this little schematic shows that when we expect to link disturbance and degradation, in tropical forest, one would say, yes, increasing disturbance would lead to degradation. But in savannas, that need not always be the case, because savannas have evolved with these disturbances like fire and herbivory. And therefore, we would think that removing these disturbances or increasing the intensity of these disturbances, both of this will cause degradation in the savannas. And therefore, we need to identify optimal level of disturbance, or is a savanna healthy enough in the disturbance regime that it has? And how do we quantify that? We need to measure ecosystem health. Ecosystem health, which it doesn't matter which ecosystem you're looking at, broadly has three aspects. One is structure, which is looking at your horizontal and vertical heterogeneity, which we are doing in our system through collaborations where we fly drone over these savannas and look at the heterogeneity in the system. We look at functions in our savanna systems, which include things like productivity, but also those are decomposition bags, which also look at decomposition rates in savannas, and something like composition, which is biodiversity. What kind of species are there? What's their richness? What's the composition? And so on and so forth. So I am working in equatorial savannas in Mount Kenya, and these are located 0.2 degrees north. These are extremely fertile systems. These are black cotton soils, volcanic soils, and fires have been suppressed in these systems. So it makes... Sorry.
Wasn't me.
So it makes this an ideal system to test how herbivory is impacting the ecosystem health in these savannas. So this is how we have a gradient of herbivory going from full exclosures where we have no wildlife herbivory happening, then partial exclosure where elephants have been excluded, open to wildlife, which is everything is there but only wildlife, going to open to wildlife and cattle. So along with wildlife herbivory, we also have livestock grazing happening, and then intense livestock grazing, and all this is along a gradient. As you can see already from these pictures, we have differences in the structure of these sites. So Areas where there is no herbivory or partial exclosures, we have higher percent cover, woody percent cover, and whereas areas which are intensely grazed have extremely high bare ground. And just keep this in mind because this will help us interpret a lot of results that we'll talk about today. So now for ecosystem health that we are measuring in these savannas, we are looking at structure function with help of our collaborators, but what I'll be talking today about is focusing on composition. We are assessing biodiversity across these 4 major taxa, but for today's talk, I'll be focusing only on birds and vegetation. So we see that in terms of herbaceous layer, this is only herbaceous vegetation, which means forbs and grasses. We don't see any significant difference across for richness or diversity across this gradient, which is contrary to what we would expect. That we were thinking that if we remove herbivores or we increase herbivory pressure, we would expect a decline, but we are not seeing, there's no significant difference. Interestingly, though, we do see compositional differences. So this over here is a highly, not degraded, but highly intensively grazed site, which is compositionally very different from all these other sites. Although areas where herbivores have been excluded do not really fall apart from open to wildlife. So the difference is only in highly grazed areas. What we wanted to understand is why, what does it mean? Why is this difference coming upon? What is it about the composition itself? And right now I'm presenting a very coarse results of just functional groups. And we see that it's when you go to extremely highly grazed areas, it's the forb abundance increases. So it's not just the number of forb species increases, but their relative abundance also increases as you're going high up the herbivory gradient. And we think that's happening because cattle are grazing, creating a lot of bare ground, but mostly removing a lot of grasses, because of which the forbs which were earlier being outcompeted by grasses can now come up. The second set of taxa that I'll be talking about are birds. In birds, interestingly, contrary to what we are seeing for plants, we do see that there is a non-linear effect, which is for both richness and diversity, we see a decline, but the decline is non-linear. Also, interestingly, we see compositional difference. But here, the interesting bit is the composition is not just different for highly grazed areas, but also for areas which were excluded, where the herbivory has been excluded. And again, we wanted to see, again, this is contrary to what we would expect, but again, we wanted to see that what does this compositional difference means in terms of functional groups. And these are the predominant functional group of birds that are found in the savannas that I work in. And this is basically based on what they feed upon and where they spend most of their times. We see that these are insectivores, arboreal insectivores, which means they prefer trees. And of course, these are more in areas where there is more canopy cover, and therefore there are more arboreal insectivores which decline as the canopy cover goes down. And then terrestrial insectivores, if you see, they increase as the bare ground increases or the open area increases. And yeah. So to conclude, we see that there are taxa-specific response to herbivory in savannas. Removing herbivores does not compromise overall diversity. So for plants, it doesn't matter. For birds, in fact, if you remove herbivores, there's more bird diversity. But increasing intensity of herbivory, although it's not impacting plant diversity, but it leads to decline in bird diversity. And despite stable diversities that we see for the 2 taxa that I presented, but with help of collaborators, we have data across other taxa, there are a lot of taxa just showing stable diversity, but the functional compositions are shifting. And these seem to be linked to changes in habitat structures like canopy cover and bare ground. So if there is one take-home message from my talk, it's that in these systems that I work on, numbers may endure, but identities are evolving, which means that if we want to understand ecosystem health in these savannas and how it is being affected by herbivory, we need to go beyond just looking at biodiversity in terms of species numbers. And that's what we are actually doing. We are looking at functional diversity, phylogenetic diversity, and trying to link functioning itself, which is things like NPP, with biodiversity through energy flows through the system to understand how they are being affected across the herbivory gradient. Thank you. I'm open to questions now.
Thank you very much. We do have some time for questions. So. Okay, well, that brings us to a close for this session on understanding biodiversity in changing savannas. Thank you very much. And I just want to point your attention, there is a poster associated with this session. It's called Hotspots of Reproduction, How Landscape Features Drive Savanna Tree Production. It's presented by Kira Mayer, so please keep a lookout for that poster. Next up, we're going to shift focus slightly and still talking about biodiversity, but talking about the impacts of habitat and transformation, specifically on biodiversity and biodiversity trends. To start things off, Dan Parker is going to be talking about the impacts of habitat management on arthropod and bat communities in an African savanna. Thank you.
Thanks very much, Don. Good afternoon, everybody. Today I'll be presenting some work that was published last year and led by my postdoc, Jess Cumley. And for those of you who'd like to follow along, you can scan the QR code there and read as we go. So, as most of us in the room will know, and as we've heard today, savannas are by their very nature heterogeneous habitats made up of a mix of trees and grasses and support everything from mega herbivores to the tiniest insects. However, due to stresses such as global climate change causing increasing CO2 levels, in many savannas we are seeing what some may describe as woody densification, where the density of trees is increasing. This woody density is in turn leading to a homogenization of sorts, where grass-dominated patches may be taken over by woody vegetation. With this sort of structural homogenization, there's a fear that biodiversity may be lost and that there'll be an overall loss in ecosystem function. Enter the AI-generated habitat management photo approach, like the mechanical clearing and subsequent mowing of woody patches in savannas. The issue, though, is that whilst such habitat manipulation and transformation is ostensibly in the best interests of overall savanna heterogeneity, we have a rather limited understanding of how, one, the vegetation may respond to these interventions, and two, what the cascading effects may be on other trophic levels. As such, we used a landscape-level manipulative experiment to test how habitat management interventions impact the vegetation, the insects that may rely on this vegetation, and then the bats that hunt them. Our study was conducted at Mala Game Reserve, which shares an unfenced boundary with Kruger, as we've heard earlier this morning. And during the wet season of 2022, we used a randomized block design, this is all really quite blarney, across 3 sites to test the effects of 5 habitat management interventions. So before I describe the treatments that we used, and those are denoted by the various letters on the map on the screen, it's important to point out that Mala is somewhat unique in that in the 1960s, reserve management created several grass-dominated areas. So in other words, almost all of the trees were removed from these areas. These grass-dominated areas, some of which are several hectares in size, were maintained by routine mowing in this state up until 2015. In 2019, we established 3 replicated tree-cleared treatments in these grassy areas, and each of these was then subjected to a different mowing approach for our study. Our treatments included a summer mow that was cut about 2 months before our study, a winter mow that was cut about 8 months before our study, a rest treatment, which was historically part of the broader mowed area, but had not been mowed since 2015, an intermediate treatment, which these treatments were unmowed sites that had been exposed to the reserve's sort of routine fire interval of about 3 years. And finally, we had our densified areas. These are areas that we initially called controls, but were vetoed by reviewer 2 during the manuscript review process. Nonetheless, these densified areas were areas that had had no habitat manipulation intervention. In addition to measuring the grassy and woody components across our treatments, we also deployed song meters from Wildlife Acoustics to record the bats present. This was done by sampling for 5 nights at each treatment at each of our replicated sites. We also deployed custom-built insect light traps immediately after the bat sampling and during the darkest phase of the moon. And this was for 1 night across the replicated treatments. And you can see Keenan up there modeling one of our uber high-tech insect light traps. In terms of the vegetation, there were no real surprises here based on our treatments. The densified or control sites had the highest tree density, followed by the intermediate sites, and the mode sites had almost no trees, so no surprises. The grass layer was a little bit more interesting. The rest sites had the highest grass biomass, followed by the intermediate sites, and then the densified and winter mow sites. Yes, it's a lot to remember, but hopefully we can keep that going throughout. The summer mow sites had, as you can see, the lowest grass biomass. Now, if we turn our attention to the other trophic levels that we're interested in, the insects and the bats, the insects appeared to have been avid readers of Began, Harper, and Townsend because their abundances seemed to increase with both grass height and tree density. And this supporting the idea that the more complex habitats provide more food and cover from predators for the insects. The bats, on the other hand, had different ideas, with bat activity being highest at the summer mow sites and decreasing significantly as tree density increased. So the sites that ostensibly had the least amount of insect food available had 7 times more bat activity than the sites with more food. So a bit of a mismatch there. We expected that the bats would, like the insects, spend most of their time where the food was, but this wasn't the case. Well, why is this? Well, we think that it's an issue of abundance and accessibility. For insects, increased woody density means more food and safety, but open areas mean that they are sitting ducks for predators like bats. For the bats, well, at least most of the bats, the open areas make it easier for them to catch prey. However, not all bats hunt in the same way. Our summer mow sites were dominated by the Egyptian free-tailed bat, which is an open-air forager and a very common species, probably driving up the overall activity observed at these sites. Other species like the Cape and Zulu serotine bats are clutter-edge foraging species, and they were found more uniformly across the treatments. By contrast, the sometimes difficult to detect clutter foraging bats like the rhinolophids were essentially the only ones recorded in the denser habitats. So what might the solution be for managers and for conserving biodiversity? The answer probably lies somewhere between the extremes, so that the broader landscape mosaic can be maintained. Not too much cutting, not too much mowing, not too much fire. Well, what constitutes too much? Perhaps then, a little tongue-in-cheek, we should all go and read Robert Southey's Goldilocks to find out. Thank you.
Thank you, Dan. We do have some time for a few questions, if there's any.
Thank you, Dan. That was really super. So you found that in the open areas, there was the lowest insect abundance, but the most bat activity. And that's puzzling. But I might suggest it's not at all puzzling, is that maybe the bats are eating the insects, and that's why the insect abundance is low.
Well, the insect abundance was Yeah, true, fair. Yes, fair point. Thank you. Yes. Agreed.
Do we have any other questions? Corley?
Thanks, Dan. I was just wondering, would they roost and feed maybe in different areas? And I know from experience, because I do a lot of bat studies at the Lake Panic side in the afternoons, and there's usually a lot of bat activity above that water body. So I'm just wondering, like, the types of insects that they are going for, is that different from the type of insects that you maybe had in your traps?
Yeah, so they weren't. What we were catching in the traps were sort of linked to the sort of main prey of the different species. But it is, so they do certainly roost in different areas, and they'll obviously fly and they'll commute to other areas. It's also kind of not necessarily confounded, but an important factor to consider is, and one of our previous speakers spoke about it, how easy it is to record certain species on these detectors, where the species that have lower frequencies, much like elephants, the lower frequencies will travel further and will be picked up by the detectors. And even if they're flying high above, say, the open areas and commuting to another part of the savanna to feed. So we do have to consider that. It is, we did have the same, pretty much the same insects that they were eating in the different areas. So we don't think that's too much of an issue, but it's more of a kind of a ultrasonic detector issue, and then also picking up the bats that are commuting over the site. Yeah.
I think we have time for one more question.
Sorry to hear the counterfactual community came after you. I can promise you it wasn't me. What do you think the bird patterns would have been, considering those insects, obviously a big food source for your biggest animal community?
Yeah, so instead of answering that with a direct answer, I'm going to point you to another study that we did, published in Ecology and Evolution, that looked at the insects and the birds. So yeah, that'll answer it for you. Talk to you afterwards.
Thank you very much. Thanks, Dan. Next up, we have a talk of Muzi Sibiu, who's going to be talking about 2 decades of bush encroachments and the effects on bird communities in African savanna. Thanks, Muzi.
Hi, everyone. Name is Muzi. I was recently a postdoc at the University of Florida. So my talk today is 22 years of shrub encroachment and effects on bird communities. So we continue to lose species at an alarming rate, and a lot of these extinctions are driven by habitat loss or habitat change. One big driver of habitat change, especially in grasslands and savannas, is this problem of shrub encroachment, which is increasing density and distribution of bush vegetation. This problem is intensifying across the African savannas. So what happens is landscapes that are experiencing bush encroachment, they've homogenized the vegetation structure, so a bunch of woody vegetation that result to homogenized animal diversities, displacement of certain species, decline of certain populations, and overall alteration of the system. But shrub encroachment is inherently temporal, but we lack structured surveys that track these impacts over time. Also, a lot of studies that try to understand impacts of shrub encroachment do not necessarily incorporate changes in tree cover. So here we wanted to, we're curious how vegetation has changed in a 22-year period. And we focused on birds, easily detectable, rapid response to environmental change. They've divested of traits that we could use to, assess fine scale responses. So we're interested in changes in bird occupancy and assemblages. Looking at that using occupancy models. So we focus on diet groups and nesting groups to understand trait responses. We conducted the study in Eswatini across four parks in Eswatini. In each park, we had 12 plots that were established in 1998. So in each of these plots, we're interested in assessing changes in open grass patches, shrub cover, and tree cover. We use satellite data to assess changes in vegetation cover. We also conducted point counts in these plots in 4 consecutive days in the summer season across the 3 periods here. To put everything together, we used a multi-species occupancy model. This model did not include grass cover because it was highly correlated with shrub cover. The model also included linear and nonlinear impacts of nonlinear terms of shrub and tree cover because previous studies have shown that wildlife diversity does respond nonlinearly. Then we summarize occupancy responses by diet and nesting groups. So in general, we found that grass patches declined by 39% over the 22-year period in our plots. On the other hand, shrub cover increased by 28%, tree cover increased by 11%, as you can see on the figure. Regarding bird species, we encountered a total of 121 species over this period. Because this model does not deal well with rare species, so we focus on species that were encountered in at least 10% of the plots. That means we ended up working with 64 species that went into the model. And we found that species richness increased from 30 to 36 species in our plots. So looking at plot scale, but there's that huge variation in 2020. So regarding occupancy responses, on the left is response to changing shrub cover, on the right to tree cover. The squiggly line, these are species-specific responses. The thick black line is community average response, with the gray ribbon showing 95% credible interval. So focusing on the figure on the left, we show that at the community level, there's that curvilinear response to increasing shrub that you don't see with increase in tree cover. So you can say shrub and not tree cover limits community occupancy in our system. So regarding species that exhibited this significant nonlinear responses to shrub cover, a total of 15 species had that strong nonlinear response. Here we are showing, the dots show average occupancy with the error bars 95% credible intervals. So we show that at least from here, we can see that a lot of these curvilinear responses, the peak responses, occurred below 50% shrub cover, meaning that with increase in shrub cover, we're going to start seeing declines in the species, including critical groups such as migrants and open country species. Again, regarding responses, these figures here show diet responses. So focusing on the legend, the blue shows none, no response. So the yellow shows that strong curvilinear response. The gray shows positive. What I want you to focus on is the yellow region on the left shows that all 4 diet groups had that strong curvilinear response to shrub cover, meaning shrub cover is limiting all these diet groups. Similarly, shrub cover is limiting occupancy across these nesting groups, but just one. So in general, this was not surprising, this result. Shrub encroachment or shrub cover is increasing in our plots. But what is critical here is that a lot of the species that we show here are woodland specialists, savanna woodland specialists. So if at the community level, this is what is declining in the system, then we should be concerned. Changes in certain species traits in the system will impact savanna resilience. But there's some positive things happening in Eswatini. In a lot of these parks, there's bush clearing going on. These efforts are increasing, and that is already showing some positive effects for wildlife. But we do lack monitoring programs, long-term programs, and there's opportunities now for incorporating citizen science data and structured surveys into integrated models to produce more robust estimates. So thank you. You can grab a copy of the research here.
Thanks, Muzi. Any questions? We do have some time.
Hi, thanks for the nice talk. This is probably the first Savanna Science Conference. I've seen so many bird community ecological projects, which is cool. Those sites of yours that were contiguously placed, those fixed radius radii, how did you account for pseudo-replication, meaning if you counted the birds in the one site that they don't fly over and you double count them in an adjacent site?
Can you repeat the question, please?
Sorry, so those sampling sites of yours, those fixed radii of your sample sites, they touched one another. So how did you prevent double counting bird species in adjacent sites?
Thank you. So we said when we go out doing these point counts, we say we are counting at a 50 or 100 meter radius. That's what we did here. But the truth is there's problems of detectability, especially with increase in woody cover. So these circles are like 100 meters radius. So even if they touch on the figure, what we counted at the center of the plot should have given us something like independent samples, I should say.
Just a quick question. Thank you very much for your talk. Don't go. Come back. Oh, can you hear? Okay, right. So yeah, thanks. Stop laughing, Carla. Thank you very much for your talk. So at the end there you were saying that the woodland species did better. I might have missed it. Did you mean the open savanna species or were there forest-associated species? Could you just confirm?
Yeah, so unfortunately, this analysis, I would say we focused on woodland species a lot because open country species like your carnivores have already displaced from the system. So they are not there. We find very few of them now. So a lot of them are woody adapted species.
Okay, thanks very much.
Thank you very much. I think that's all we have time for now. Next, we'll be switching to the Brazilian savannas. The talk from Adela Medeirosa, looking at soil and fungal diversity in Brazilian savannas.
So, hello everyone. I'm very happy to be here today to present the first chapter of my PhD. This work is developed by the University of Exeter in partnership with the University of Unicamp in São Paulo, Brazil. So it's entitled Soil, Fungal and Bacterial Diversity Across Pastures and Vegetation Types of Brazilian Savannas. So Brazilian savannas, known as Cerrado, is a biodiverse hotspot. So it has thousands of endemic plant species. The soils there facilitate the water infiltration to important rivers in South America. And the soils are also acidic and nutrient poor. However, the Cerrado is being degraded, replaced mainly by pastures. Almost 50% has been lost across these years, and mainly to cultivate this plant, the Urochloa decumbens, which is an African grass, which became invasive in Brazilian savannas, and is a nightmare for restoration in pastures because it easily regrow and re-invade the areas of restoration. So the farmers, to promote the growth of these species, they intensively promote the soil modification and adding lime and fertilizers, for example. And this has potential consequences for microbial communities, soil communities. But what are the patterns of soil microbial communities in Brazilian savannas? We don't actually know very deeply. We have some work done in relation to some variables in the soil, but still have a lot to do. So the soil microbiome patterns there are still poorly understood. That's why the aim of my first chapter of the PhD is to access the soil microbial composition and functional changes across vegetation types to set baselines for restoration and also for conservation. To start in the methods, we selected 5 different vegetation types, from completely open grasslands until more closed ecosystems. And we compared with 2 pastures, with and without trees. We established these plots in the center of Brazil, in the center of Cerrado, in the municipality of Alto Paraíso de Goiás. We have 31 plots of 50 per 20 meters, from where we collected information on plant coverage, plant soil coverage. For example, we flew a drone to classify the images by tree grasses, and we also collected data using soil grids to do a visual estimation on rock, litter, and bare soil percentages. And the soil samples we collected to analyze the nutrients, the organic carbon, the moisture, and we did also a soil microbial data extraction to see the species identity and the functionality of soil microorganisms. So I will explain about this pipeline of the DNA processing. We extracted the DNA from the soil and we did the sequencing using an approach that brings all the DNA regardless the group. So we have no primers. We use a nanopore sequencing. So this first pipeline in blue is regarding the taxonomy and functionality of the soil, mainly related to bacteria, and the other pipeline in green is to composition, to access the composition of fungi and bacteria. Let's keep talking about this first. So the diversity of bacterial species was correlated, negatively correlated with tree abundance, and other parameters like the soil bulk density affected negatively the fungal genome diversity. But the fact is, we saw that the bacterial, both the bacteria and the fungal communities in pastures are a subset of the native ecosystem. So you can see in the diagrams of Venn the proportion of species in native in green and brown in pastures. And we saw a reduction in 24% less bacteria and 18 less fungi compared to the native sites. The composition of these communities, actually, they didn't change too much. So the dominant species are there both for bacterial genus and for fungal genus. We saw that many of these are actually mutualistic species. So the pink one for bacteria, Bradyrhizobium, is a nitrogen-fixing bacteria. And the yellow one for fungi is Rhizophagus, which is a arbuscular mycorrhiza, and can help, both of them can help the plants grow. But there are other groups, obviously, like pathogens and related to the carbon degradation as well. Let me move on. So we found that there is a core microbiome which is not affected by the establishment of pastures. So let's talk about the functionality based on this workflow to build the megs in the soil. So first, this is a not so usual graph, so let me explain. In y-axis, you can see the vegetation types. In the x-axis, you can see the vegetation types, and y-axis, you see the functions. On the top, related to methanogenesis and methanotrophy, and down there, related to nitrogen metabolism. Green is presence, white is absence. So, first of all, methane generation is limited on pastures, and methane consumption is only present in native sites, which indicates for us that there is a higher carbon storage in native soils that allows the methane generation compared to the pasture management. The pasture management lost carbon, probably. So there is an indirect estimation, there is an indirect measurement of the genes related to that functions. So in terms of nitrogen metabolism, we saw that ammonia production is only present in pastures. So ammonia is a form that's directly available for plants, but we observed the opposite in native sites. So there is a competition of bacteria regards to plant in terms of ammonia in soil. Bacteria are consuming consuming ammonia in soil, and these kind of genes from bacteria are only present in native sites, which lead us to understand that the bacteria contribute to nitrogen availability for plants in pastures and for scarcity in native sites. That is an interesting trade-off. As a final take-home message, despite the weak taxonomy change, there are some microbial trade-offs in terms of functionality between the native and pasture sites. And we need to understand the microbial diversity better to improve the savanna restoration, because so far it's a huge challenge, especially in these times that huge fast degradation of natural ecosystems is occurring. So we may lose some insights, we may lose some functions related to these microbial communities. That's all. Thank you.
Thanks very much. We do have time for questions. Questions from the group?
Hi, great talk. I was interested in a bit more of a methods question. And how did you assign functional groups to your sequences?
Yes, thank you for your question. That's a very interesting approach, actually. We do this based on DNA sequences. So I sequenced every single piece of DNA in the soil. So we usually call this eDNA. So from this bunch of sequences, we build a grouping using machine learning and other techniques, and assign, because we grouped as bigger as possible as of one genome of a bacteria, for example. As we have this bigger genome, we have the information, the genetic information of one species. So we have the genes related to that functions. And then we annotate these genes based on global databases. And we relate, for example, 3 genes participating on the nitrogen fixation, the reduction from ammonia to nitrate, for example. There are some genes related to that. And then you can annotate that and you can quantify that. But in my case, I did only presence and absence. Don't know if I answered properly. We can talk more afterwards.
Thank you, Adler. That's all we have time for now, but thanks very much for the talk. Next up, we're going to be back in African savannas, this time the Congolese savannas, with Catherine and Jer talking about spatial patterning and diversity of termites in these savannas.
Testing. Okay. Hello, everybody. My name is Catherine, and I am a PhD student in Andrew Davies' lab. And today I'll be talking to you about the spatial patterning and diversity of termites in shrinking Congolese savannas. Great. Yes, so I assume for any of you here who have done fieldwork in savannas, you might have seen a termite mound or two. They do try to hide sometimes in the vegetation, but they are everywhere. And especially these really large mounds that are built by some groups of termites in the macrotermitiny often, and you can see them very easily on the landscape, and so they're quite well studied. Past work has shown that they influence the heterogeneity of vegetation in savannas, they accumulate nutrients and moisture, animal activities revolve around them, and the termites themselves, whose colonies can number in the millions, are responsible for a large part of the wood decomposition of savannas. Therefore, I think they really do earn the term ecosystem engineers. But there's also another kind of mound that you will see built by termites that's quite abundant in especially the more humid of tropical savannas. And these are made by a group that we can call soil-feeding termites because they eat soil like worms. These are slightly less studied than macrotermes type groups that build these very large mounds, but they're still very abundant, and increasingly we're realizing they're quite cryptically diverse as we get better with sequencing data, for example. So there's a growing body of work that's starting to untangle the ways that these soil-feeding termites act as soil engineers in tropical savannas. And I wanted to dive into a bit more of that in my PhD research, so I mainly focused on them. My study system is Adzala-Kukua National Park in the Republic of Congo. I have my little William Bond shout out here with some theoretical work that was based on a lot of his original thinking. And that's saying that essentially what's really interesting about this part of the Congo Basin is that given the environmental envelope that we see here, you could actually have either rainforest or savanna coexisting on any given landscape, and it's often just maintained by internal feedback loops. And so Adzala-Kukua National Park happens to be right on that boundary in today's climate, where it's a mosaic of savanna and forest ecosystems. And that means that if we look back through geological time, you see a lot of change at this boundary area, with glacial periods seeing an expansion of savannas and interglacials an expansion of rainforest. And that's what we're going through right now. This really great recent study by Evan Hawkridge, who spoke earlier today, showed definitively that the savanna patches that are remaining in Adzala-Kukua National Park are shrinking very rapidly. And so another thing that I find really interesting about this study system is the opportunity to get an understanding of these communities that have faced a lot of dynamic ecosystem change and what that might mean for the future. So this is part of a much larger study that looked at termite communities across the park and in a range of habitats. Some of these include buy sites that were also talked about earlier today. So if you're interested in the interactions between decomposers and sodium availability, please talk to me afterwards. But for today, we're going to focus on comparing savanna and forest habitats. And I had around 6 sites each that I was comparing the communities using standardized transects. Once I had that foundational information, I wanted to specialize and focus in on these ones that build mounds in the savannas and ask what's their abundance there and how are they distributed across the landscape, with the idea of starting to get an appreciation for how they might impact the ecosystem at a landscape scale. So one of the first really surprising findings of the transect sampling in savanna and forest sites was that the abundance and richness of soil-feeding termites was essentially the same. This was especially surprising because soil-feeding termites are often thought to be quite prone to desiccation and they're relatively sensitive. So I really did not expect this result. To get a better sort of appreciation for what the forest does have over the savanna is that it has really high beta diversity. So the turnover that we saw between forest sites was very high, whereas the savanna communities, we started to see them sort of stabilizing and getting kind of fully accounted for by 5 or 6 transects. We're nowhere near knowing all the termites that are living in the forests. But regardless, I think this points out that we often assume rainforests are just going to be more diverse than savannas, but on a site-by-site basis, this is not always true. So a really obvious reason that beta diversity was going to be higher in the forest than the savannas was that the savanna patches that I was sampling in are quite small. So that means that these sites are all within a few kilometers of each other in the savannas, and they're much further apart in the forests. So to get kind of a bigger picture of what beta diversity might actually look like for savanna termites, I'm starting to integrate other studies that have been done in the region. I'll just mention briefly today this really fantastic study by Fidel in several sites of savannas in Gabon. And I basically have been reprocessing his data, integrating their sequencing data with mine, building phylogenies, delimiting species, and trying to figure out what kind of overlap we see across these 2 countries. So far, it seems like on a per transect basis, there's around the same average number of soil-feeding species as we see in Congo, but per site, it's maybe a little bit higher than we were detecting. But most surprisingly is the beta diversity is still pretty low here. We see most species over 50% are shared even at these distances. So still processing that and excited for more to come, but that shows it's not all just about the distance between the sites. So next, I wanted to ask how abundant and how are these mounds distributed on the savanna landscape. This is because I feel like a lot of the ways that we were able to appreciate the large termite mounds as important ecosystem engineers has come from being able to remotely sense them. Andrew Davies has led a lot of this work. And I wanted to see if we could kind of bring these termite mounds also into that landscape scale perspective. Usually this would have been quite impractical, but we got lucky in that there was a controlled burn in one of the savannas, or 2 parts of the savannas, and then we did a drone flight immediately afterwards, so I was looking at that data. The drone had really fine-scale resolution light detection and ranging data, but unfortunately, despite that, I was not able to structurally differentiate the mounds from the terrain. But interestingly, this is where the sort of mushroom shape of these mounds comes into play, because they have this very round cap and it's actually quite reflective, especially against the recently burned vegetation. So through the RGB data, that's essentially just nice photos that the drone also took, I was able to actually start identifying these on a landscape scale. So I decided to just give it a shot, see how it compares to the in-field plot data that I had collected of these 20 by 20 meter plots. Unfortunately, it was still a little bit on the low side. It's about 60-fold less detectable remotely than it is in the field. But I am still working on filtering the data a little bit better for grass content. And this leads to a pretty wide estimate of the number of mounds that we might have in this park, between 5 and 350 million mounds. But for comparison, there's around 700,000 mounds of the larger type. And the way I see this as becoming useful as a metric is from experiments that look at mound level impacts of these termites, which has been done in some other species. I somewhat replicated one that had been done on a forest species and looked at how the termites might be altering soils, and found that over the course of only 2 weeks, they could increase nitrogen and carbon in soils by around 10%. So this is pretty small changes, but you can imagine that when scaling it up, this could actually have a large impact on the nutrient landscape. So to summarize, the termite diversity in Congolese savannas is actually quite high when we're looking at alpha diversity, but has pretty low beta diversity compared to forest sites, even when we look at a regional scale. And the mounds are extremely densely distributed, but we still have some challenges in measuring them remotely. Thank you so much to everybody and my funding sources and my lab and the people who are not pictured in this lab photo but are here today, Lucas and Catherine, and to my field team. So yes, thanks.
Thank you. We do have a couple minutes for questions from the group.
I was wondering why did you expect to find more species in the grassland of these soil-feeding termites? If forests have a more ancient origin and they were, if not now, once more widely distributed, then you wouldn't expect there to be more grass-associated species. So I was just wondering, like, has there been work looking at what that landscape looked like into kind of geological past?
So there are probably two main reasons. One is probably just my inherent bias that I feel like many of us are taught that, you know, rainforest, species rich. And it's true, that's one of the reasons I'm interested in this very dynamic region, is because presumably there's a lot of recolonization that constantly has to happen. And so it's not the same kind of stability that we might see from more stable forests. But the other is that the Congo Basin is posited to be the evolutionary origin of this group of termites. And so even with the struggles of recolonizing this very dynamic area, it's still been around for about 50 million years worth of diversification. And so I just assumed that this is the biodiversity hotspot for soil feeding termites. So they're probably going to be very diverse there. And colonization of savannas did happen, 30 million years ago, but I just had this sort of general temporal bias there. Yeah, there has been some isotope work on soil feeding termites, but I'd say it's probably one of the biggest mysteries of, we still don't know for sure what exactly they're eating out of the soil. Because there are so many species of them that coexist within like meters squared of each other, presumably there's some niche differentiation going on, but it's still pretty controversial. Some of the latest research that I think is more enzyme based is suggesting that they're eating microbial matter off of soil particles. But I don't think it's been conclusively proven yet. But people have tried isotopes. I just think it hasn't been able to distinguish it perfectly.
The last question.
Thank you so much. I think Corley's follow-up question might have reflected what I was going to ask. But it was really about this amazing species richness and species diversity, and whether you've got any insights about whether that's really, there are differences in the functions of these different species. Like, I think perhaps you've answered it, but have you got any idea whether more species means more different types of soil turnover and respiration?
Yeah, so this was kind of my dream goal that I wanted to answer with my PhD, which is why are there so many different species of soil feeders in this place? The first step to that was to thoroughly sample and figure out kind of the extent of the problem. So like, how much coexistence do we really have to explain? I was hoping that doing pretty fine scale sampling, the problem wouldn't be so bad, but it's still bad. There's a lot to sort through. So I've done a lot of like soil experiments and, moisture measurements, and we have LiDAR data of grass volume. And this is all going to be thrown into a model that I'm hoping will start to explain some of those community differences. But the truth is, on a transect by transect basis, we still see way too much coexistence, and it's not explained by vertical profile, and they're all eating various forms of soil as far as we can tell. So I'd say this is a very open question. People should get more interested in soil feeders. What are they doing? Yeah, thanks.
Thanks, Catherine. We're going to be sticking with termites, looking at with a talk from Kate Parr on the hyperdominance of termites in deadwood decomposition across the Afrotropics.
Thank you, Kat. That was a really great introduction. So now you know all about termites. You learned about the soil feeders. I'm going to tell you about the wood-feeding termites. Before I start, I just want to do a big shout out to the team of people who are involved in this work, particularly our PhD students who worked across different African countries. So this is Kath, Fidel, whose data were actually shown in Kath's talk as well, and Gabriel as well. So decomposition is a really important part of the global carbon cycle and is thought to be controlled principally by climate and litter quality. So decomposer organisms is up there and it's a bit smaller, so people have sort of not paid that much attention to them, but if they have, they've really focused on microbes. So microbes are this sort of attention that they've received is probably because most of the work's been done in temperate areas where microbes are really dominant. We don't have termites. But there's growing recognition that invertebrates can play a role. And then there was this lovely study that illustrated this relatively recently that showed, first of all, the decomposition rates was greatest in the tropics, but actually the net insect effect was also greatest here. And as Kat has introduced, the termites, those are the insects that are most important in tropical decomposition. So we've done a bit of work on termites already. This was some work that Amy Zan led, and this was a global study. And here we put out wood blocks across, well, it was a global study, so all kinds of biomes. And this shows the decomposition rate per year. And what we found really was that these biomes that are hot, so rainforest and savanna and even subtropical desert, that's where the decomposition rates are highest, and it's driven by the termites. So if we look at how microbes respond to temperature, the decomposition rate with microbes, they show sort of a slight increase with temperature, but it's really when you add the termites in at the bottom here that that's where you get this strong effect. So they're highly sensitive to increases in temperature. But you'll see there's quite a lot of variation here when it gets very hot. So we wanted to look at what might be causing that. So then we thought, well, maybe water availability might also be important for decomposition. And as a shout out to William, there was this paper that William Bond and Huli Pausas produced in 2020 that was looking at the major pathways of recycling of vegetation. So what happens to all the green stuff? So if it's dry, they said it could get eaten. If it's intermediate rainfall or moisture, then it burns. And then they said if it's really wet, it decomposes. That's the main thing. So I read this paper and I was like, oh, this is interesting. And William was talking all about microbes. And decomposition, he said, was all the microbes. And he did mention insects, and then he went and lumped all the insects with the microbes. And I was like, no, come on, this is not right. Even if they operate at the same scale, which he thinks they do, it's like they're not equivalent. We know that microbes really do need moisture in order to operate, but actually termites don't. Many termite groups can decouple their environment themselves from the environment, and they can engineer their own microclimate. And we know that termites do occur in dry places. So then, inspired by this, I thought maybe we should think about these two agents, microbes and termites, and what they might look like. So then I thought maybe it looks a bit like this. Perhaps microbes do better when it's wetter, and maybe termites do better when it's drier, quite simply. So we set out to test this. And we asked the question, across the Afrotropics, where temperature is relatively high, so we'd expect termites to be important, do termites and microbes have contrasting responses to rainfall? So do we get this kind of response? And then overall, what's the net effect? So if if one is replacing the other, perhaps it actually looks like this, and perhaps across the rainfall gradient, decomposition is actually fairly constant. We also asked, does the dominant agent change with rainfall? So of the decomposition that happens, how much is by microbes and termites? And then does decomposition and the agent differ with the vegetation type? So whether you're then in savanna or if you're in forest. And to do this, we had our amazing students and big field teams of people, and we did an experiment across the Afrotropics. So we had 35 sites across Africa that were in savanna and in forest, and covered a gradient of rainfall gradient from actually less than 500, it was quite a lot lower, but Mike Wilklim says it's higher, to 1900 millimeters. And all the sites were pretty hot. So our sites were in South Africa here, across Ghana, and also in Gabon. And at each of our sites, we used a wood block experiment. So we put out blocks of wood that we enclosed in mesh bags and sealed them. And if they're sealed, there's very fine mesh so the microbes can get in. So those are our closed bags. But then we punched some holes to allow the termites to get in, and those were our open bags. And if we subtract one from the other, we can work out what the termite contribution is. So we left them in our sites for 12 months, and then we came back and we looked how much mass had been removed. So sometimes there wasn't very much removed. It was always a bit disappointing when there was a full wood block. But actually, sometimes quite a lot had been removed, as you can see here. So sometimes there was one particular area, this might be Wits-Rural, the bag was like, the wood block was entirely gone, really. Right, so let's have a look at those agents then to start with. So what we found was the termites and microbes did actually have contrasting responses to rainfall. So this just shows our gradient here, and this is the absolute decomposition for termites and microbes. But the effect was much stronger of the rainfall gradient for microbes than it was for termites. So this kind of works, but maybe it ought to look a little bit more like this. So then if we put those two together, what does our overall decomposition look like? Well, actually, there was no change then across the rainfall gradient. So decomposition was unaffected by rainfall. But what about who's doing the decomposition in terms of the proportions? So what this shows is the rainfall again, but this is now the proportion of termites or the proportion of microbes. They're the exact inverse of each other because it's proportion. And we've got this little line across here, which is the 50% line. So what we find is that the termites decline with rainfall, and you think, ooh, the termites aren't doing so well when it's wet. But actually, they're the dominant agent at 34 of our 35 sites, right across that rainfall gradient. So this whole paradigm that microbes are really important, we're going to have to start questioning this a bit. Let's look at the habitats. So here we've got the termite and microbe agents and the savanna and forest and our absolute decomposition rates. And again, termites were decomposing, of course, more regardless of the vegetation type. So whether you're in savanna or forest, it's all about termites. And I should add, there's no effect of the habitat, and there's no interaction either. Now, the contribution from microbes is actually really, really small. So for everyone banging on about how important microbes are in decomposition, at least across our gradient in our experiment, they weren't. You're talking only 7% down here in the savannas. That's really tiny. So why are they so dominant in Africa? What's going on with this termite power? Well, it could well be that actually lots of these termites are fungus growers. So they belong to the Macrotermitinae. So they're things like the big Macrotermes mounds that you see. And so they grow their own fungus. And that means that decomposition rates are particularly efficient. And it also means that the colonies of these termites can actually be really, really big. So to think about this, whether it might be something to do with fungus growers, what I'm going to do now is show you where some of our sites fitted relative to a location where there are no fungus growers. So these are some data from a study by Baptiste Fijas et al. And these are sites from wet rainforest all the way to dry savanna. And these are in Australia. So if it's green, this is basically microbe decomposition, and that funny orangey brown means there are termites. So the first thing to notice is they did their study for 4 years. If we'd have done our study for 4 years, there would have been nothing left at the end. So let's see, we had 2 sites that we could pick out that were comparable. Let's first look at the dry savanna. So this is how much of our wood blocks were left after 1 year. And they basically hardly had anything gone. And then this is in the wet rainforest. So again, our rates of decomposition in Africa are much, much higher. I should add, they also use the exact same method we did and exact same wood. So the data are comparable. So what do we conclude? Well, this wood decomposition is actually constant, so rainfall isn't playing a role in Africa, particularly when you get these high temperatures, consistently high temperatures. And these termites are this major player, and this microbe contribution is really teeny tiny, and we need to start changing this paradigm, at least for Africa. So we need to consider termites more in global carbon models. We need to consider them more when we start thinking about soil carbon. And then we need to do a lot more work on this, because though it sounds like we know everything now, we don't. We need to think about wood density, and of course we need to think about other substrates. So I was just talking about wood, but what about all the grass and the leaf litter and the dung? I suspect the patterns are probably different for that, but we just need some more money for another continent-wide study. So with that, I'm going to say thank you to our incredible field and lab teams, and to our funders, and for you for your attention. Thank you.
Thank you, Kate. We do have time for one quick question, if there's one.
Thanks, Kate, and yay to insects. I was just wondering, a methodological, I'm not sure if I missed it, how did you separate the combination of termites and microbes in your bags that allow the termites in?
Yeah, so we don't. So we've got some bags just with microbes and some bags with termites. So if we want to know the microbe contribution, we just look at the microbe wood blocks. If we want to work out the termite contribution, we subtract the microbe bags from the microbe and termite bag. Cool.
Thank you very much. Thanks, Kate. Then this will be the last talk for this session, presented by Mdhluli. He'll be talking about the influence of bird behavioral traits and habitat on predicting hemoparasite infections. Thank you.
Okay, good afternoon, everyone. You know, the beauty about presenting last, it means you've got all the time left that everyone else left over. So settle up. Okay, my name is Mdhluli. I'm at the University of Mpumalanga, and today I'm actually going to be presenting something quite a little bit different, not really looking at the actual parasite itself, but we're going to be looking at some of those things that actually drive the infections in the host itself, which is the bird. All right, let me get that. Okay, so I think the general idea is that, you know, when you look into basically our birds found in the savanna systems, you know, they are quite rarely sampled and poorly documented in terms of the infections that you find in some of our birds. And therefore, we've been trying to close that gap quite a lot by doing a lot of work where we sampled most of these birds and be able to document basically the diversity as well as basically the type of infections that you find from your arboviruses to your parasites and all other types of parasites as well. But what we begin to understand, you know, besides that this system is poorly studied and poorly actually sampled, we're beginning to understand that there might be far much more bigger drivers of these infection and prevalences that we find in these birds. And therefore, in this particular aspect, what we were quite interested in looking at was basically looking into these host vector contact rates that might actually influence and actually, you know, be responsible for the spread of the vector-borne infections that we might actually see. So in this particular study, we had a very simple hypothesis where we looked at the birds' behavioral traits, and we tried to link that to the bird's habitat influence and the chances of infection. So, you know, I'll sort of take everybody a little bit back to say, you know, there's something very quite clear and established, and this is the fact that for a bird, you know, virus infections, be it viral infections, be it protozoans, bacterial, you know, most of these that we understand, especially in this part of the world, is that there is a vector that actually transmits those infections to a bird. And from the bird, obviously, you know, depending on different distributions and diversity and different also species of birds, you will get responses, and those responses could actually affect the bird's body condition, which ultimately might actually also manifest in its own immunity, as well as maybe manifest some really quite clear clinical signs, if not mortality. But over and above all of that that's happening, we are so much now appreciate the fact that, it's the systems in this particular area, things like your rainfall, when it occurs, that actually is going to line up and actually influence the rates and when we get these infections. And furthermore, we begin to also understand that the different habitats that we have in our systems are actually important because they will harbor the vectors that will be the link between the actual pathogen and the host. And therefore, if they are there in the system and that is the particular habitat that favors them, and when that happens, then it actually does translate to infections. All right, but what about the behavior of the bird? We sort of thought to ourselves, what really does that do and how does it contribute to infection prevalence, as well as, the chances of you actually getting infected? And so to look into that, we were very quite, thought in a very exploratory work and said, how do these life history traits actually influence infections? We know birds are found in different, heights. They will feed and forage in different heights, be it some will be ground foragers, others are going to be middle, foragers, others will actually, move in between up and down, others are way up in the stories of the canopies. And then I think I'd just like to bring your attention to the middle picture. That is actually a contrast between the Kruger and just outside. And what that shows is a habitat that looks minimally disturbed being inside the park, and that is outside, and the amount and basically the intensity of settlements that you will actually find in these systems. But birds, remember, they're not limited by fences, and they can actually quite transcend between those two systems quite easily and probably on a daily basis. But some birds have chosen to live and exist in these human habitats, whereas others will persist in these systems that are in protected areas, as we can see. But most of the birds that we also have here, as much of them can actually be endemic, there are some birds that are actually migrants. They will move between continents, and then we do have some, not only residents, but they tend to do this very interesting movement in the region, and that we would term as being just nomadic within the region, and they move about tracking all this availability of resources, as it were. All right, so what did we do? We followed a very simple system where we sampled our birds initially to understand what the prevalence was and what they had. With this particular focus, it was at us looking basically into our Hemoproteus prevalence, as well as our Plasmodium prevalence and leukocytosol prevalence. These are your blood parasites, pretty much. Some people then will say, your Plasmodium is quite similar to what we term as our malaria. Therefore, birds also do get malaria. And looking into things like Hemoproteus, they fall somewhere quite close to what we might term as well as malaria. So if you hear me using those terms of malaria, just remember I'm talking about your Plasmodium infections and probably some of the Hemoproteus ones. All right, so we sampled inside and outside the parks because we've got all of that variation. We sampled over, dry and wet seasons, a period of about 3 years. We were able to get about just under 1,200 birds. That actually represented about 87 species of birds. And then we screened them for infection using molecular methods. I'm not going to go deep into that, and went on to basically do some genomic sequencing to know who's who and who's infecting what. But as I mentioned earlier on, we wanted to understand what is it about the life history traits of these actual birds that actually influences their infection. One of the first things that we asked ourselves was, some birds hang in big groups. They're gregarious. Other birds, you find them in solitary. So does infection actually differ in these different types of groupings that we have? And what we found was that with Plasmodium, yes, it did matter in the sense that mostly birds that were gregarious in nature were the ones that had the highest infection of Plasmodium related infections. Whereas the other two, which is your Hemoproteus and leukocytosol, that was actually quite higher, interestingly, with birds that actually hang around alone. So it brings about that very question, do we really know what the vectors are for some of these blood parasites, as it were, because, we would all think, if we hang out together, then probably the chances of the two hosts through the vector is going to be quite high, and therefore infection and prevalence is going to be quite high, but that's not the same. It's actually all basically infection genus actually dependent. Then the next thing we looked at was basically, the issue of where exactly are you found in the strata? Are you feeding on the ground? Are you a middle feeder? Do you mix the feeding up and down? Or are you actually found up in, feeding in the canopies like most of our silos would do? And what we found overall was that actually it was the mixed feeders mostly that actually had the highest prevalence of infection. So they have an opportunity to actually interact with a wide variety of vectors, and therefore it also increases their chances of actually getting infected as well. Well, there was a little bit, not significant, but you could see that leukocytosomes, for some reason, birds that were actually aerial feeders, most of your swallows, tend to have the higher prevalency of actually leukocytosome. And then we then looked into basically how does this all relate to birds, those that stay and those that decide to leave in terms of migrants, nomadic birds, and the ones that just hang around in the area. And what we found is that actually residents, were overall, if we think about it, residents actually harbored most of the infections and therefore had the highest prevalence, with the exception of leukocytosomes. So for some reason, leukocytosomes seems to be doing something totally different. We've also sort of hypothesized that maybe we don't really understand exactly the whole transmission routes of leukocytosome. But with malaria, your Plasmodium and Hemoproteus, it was quite expected in the sense that, our resident birds being here exposed 24/7 to conditions that probably, bring about malaria, therefore they would have the highest prevalence, as it were. Then last year, we looked into this whole idea of birds that have chosen to be so much associated with people, and then those that actually are found in the wild and the ones that translate between the two, what would that prevalence look like? And what we found was the fact that for leukocytosol, then the ones that are actually found with people had the highest prevalence, whereas for the other ones with Plasmodium, we found that basically it was the ones with the mixed and then the wild that actually harbored the most infection. There was nothing really quite significant to report with Hemoproteus, and overall, well, there wasn't really a pattern to actually explain exactly how that association was different, whether anthropogenic or wild sides would actually translate to infection prevalence. So I think just to wrap up some of the things that we're beginning to understand is that actually, when it comes to basically having malaria, if you basically have got, you hang out in groups, you're gregarious, you're likely going to be more infected than other hemoparasites appear to actually be quite highly prevalent in the birds that hang around alone. We had another work that we did, which actually this actually confirmed it, was the very fact that, our invasives, that actually for some reason they hang out with the people tend to have lower prevalence, whereas the ones that we found, the native birds, they stick around here and they're actually found mostly in the wild, had the highest prevalence. And, the obvious ones that we were able to confirm, because we know this, with vectors, they're quite high in the summer wet season, and that was quite high in terms of prevalence, whereas in the dry season we had the minimum infections. So to take home, what we can say is that actually infection is not predicted by social organization alone, as it were. There's so many things at interplay here for us to actually understand how these life history traits and behavioral and habits actually translate to risk of infection, as it were. There were some genus-specific analysis that we did and revealed that high infection in gregarious birds, but we take it back and we realize it's only for malaria, but for other things it was totally something else different. And then we looked at the effect of the habit, and we realized that it varied among birds, with malaria showing, the strongest association in all of this. So our findings basically, actually give precedence to the hypothesis that we started with, where we said, we expect that the ecological and behavioral factors alone cannot fully predict infection risk, despite their interaction with vector dynamics and parasite life history, meaning we really need to go on and understand exactly what vectors are the ones that are actually transmitting some of these infections. With that, I would like to thank most of our team and some of the collaborators that came into this project and actually contributed quite a lot to it. And thank you, in case you see one with a ring.
Thanks, Mdu. You have used your time, but it is the last talk of the day, so if there is a quick question from anybody,
Thanks, Don. Thanks, Mdu. That was a really cool talk. The effect of the migrants seems really strong with the malaria prevalence. I think it said on your figure there that there was 0% malaria in migrants. So is that migratory escape? When are you sampling the migratory species? Is it when they just arrive in the summer range or at the end of the wet season?
If you think about it, thanks for the question, Jason, is that we were sampling, pretty much in the middle of when migrants have actually arrived, right? They are here in September, October. We were sampling in November and everything else. So we're expecting they are here. They could have had the opportunity to be susceptible, whatever the case was. But we are not finding a lot of infection rates like we would have expected. And secondly, I think there's always a whole lot of theories about this, the fact that malaria is not so high in birds than we more than think, I think just about 30%, but we think the ones that actually make it and come back over and over, maybe they've developed some form of immunity and we're not picking it up, and just looking into probably prior infection from antibodies is not enough for us to actually reflect and say that actually means active infection. So we're still looking into it, but to answer your question more broadly, we will use other forms of infection, other viral diseases to try and really understand, are they really escaping, or in actual sense, maybe we just missed it with this particular pathogen that we looked at this point.
Thanks, Mdu, and another special thanks to everybody who presented in the sessions from lunch. Thanks a lot. There are two posters associated with this session. There's one from Henrike Lehmann on the forgotten terrestrial gastropods in changing South African savannas, and a second one from Marunwa Motoho on bush encroachment and environmental determinants in Marikela National Park. So please have a look out for those ones. Thank you again to all the presenters of the sessions. Apologies to all the presenters' names who I mispronounced, so apologies to all the presenters. And yes, just a reminder that the Lightning Connection session is going to be right after this, so please stick around for that. It'll be just outside here in the meeting area. It'll be sticking around here. It'll be right in here. Okay, well, thank you, everybody.
Thank you, Don, for taking us through to the end of the formal program. As Don said, we are going to move up our Lightning Connections networking session. So thank you for all of you that are planning to stick around. This is something that we introduced last year to try and get people to chat to someone they've never met before. And so see this more as an icebreaker than anything, hugely work-related. You will see there's not a single work-related question that you will be asking tonight, at least not from our side. So basically what we are going to do is you will be receiving a form that you need to fill out. Every time you ask someone a question, we will give you a question you need an answer for. You will write down the answer and you will write down the person who gave you that answer, auditing purposes, okay? And so what is going to happen is you are going to start a conversation with the person next to you. And part of the conversation needs to include the answer to the question for each little 5-minute interaction. At the end of 5 minutes, we will tell you how to move. You will move to a new person. You will get a new question, which you will write down. At the end of your engagements, we will have bingo. And whoever gets all the answers correct, gets all the answers I'm looking for, then you get a mystery prize. Okay, so Are we all ready to start? Do we all have our pieces of paper? Okay, they should be coming. In preparation, please, if there's an empty seat, I want 2 people per table. So please move if you by yourself. Not yet. Okay, so if you are at a table by yourself, please find a partner so that each table has got 2 people, please. Okay, if everyone can please find a seat. So please make sure there's not an empty seat. Okay, I see empty seats, so please fill empty seats.